Hello students, welcome to NEET Biology. This is Neelavati, your biology lecturer. Today I am going to teach you the second chapter of first unit, that is nothing but biological classification. So again it's a one shot video for NEET. So like this, I have done the classes for NEET for all 32 chapters of both class 11th as well as class 12th. Okay, so if you combine the syllabus of both class 11th and 12th, we are going to get 32 chapters. So for all these chapters, I have done a video for NEET only. All are one-shot videos. So total 32 videos. If you want to watch all these videos, you have to become a channel member by paying rupees 199 per month. Okay, it's a very small amount. So if you just click on the join button, so you will get an option to buy membership by paying rupees 199 per month so that you can become a channel member. If you watch all these videos, which are specially done for NEET and competitive exam, here I am going to assure you 100% guarantee I am going to give you that you can score more than 350 marks for 360. Okay. Many students have got selected to government medical colleges just by watching the videos of our channel. Okay, so without wasting time, let's get into the chapter. Okay, so biological classification. What is the meaning of biological classification? So we all understood about classification, right? Classification means grouping. So here, biological classification refers to grouping biological organism. Biological organism in the sense, nothing but living organism. Okay, so why we have to group the organism? I will make you understand with a simple example. Let's imagine in a box, if I have kept all different color marbles, say for example, I have kept some pink color marbles, white color marbles. So let me take a few more colors like blue color, yellow color, green color. Right? Now, if I ask you to mention the shape or size of any of these marbles, say for example, randomly I will say, take any yellow color marble and tell me its character. Then it is quite time consuming, right? To put your hand inside this box and need to search particularly yellow color marble, which is of spherical in shape, not a rod shape. So like that, it actually time consuming and at the same time, it creates confusion also. Okay, suppose instead of this, if I made into a group, okay, if I kept all this marble in a separate tray, so that if I separate like a green color marble here in one tray, pink color marble in one container, okay, so like that, if I keep the yellow color marble in another container, now it is so easy, right? Your work become easier. If I ask you to pick yellow color, easily you can go to this container, you can pick it and you can study about this. Okay. And within that, once again, if I made further classification, so that we know that here I have kept both different sizes as well as different shape. Now, if I separate these two, again like this. If I kept all spherical shape here in one container and all elongated shape in another container, now work become further easier, correct? So like this, to make our work easier, we are making the animals into classification. That means we have kept the animals into different categories, okay? So that if anybody comes and if they ask us to study about any insect, then easily we can go to that group so that we know all the characters of insect and we can study about it. So like that, if I want to study about any fishes, I can go directly to Pisces classification, that is Pisces group, I can study there. That means the work of an ecologist, okay, or a taxonomist becomes so easier if I do such classification. Okay, so this is what actually about classification. But here in this chapter, we are not just studying about the classification. We need to learn about few kingdoms also. Okay, already you have heard the name about the kingdom in our previous chapter itself. Okay, but let us understand what exactly we are going to study in this chapter. That is about the overview of this chapter. Okay, once you understand the overview, work becomes further easier. See, basically, here in the first part of the chapter, we are going to talk about the classification. Many types of classification came forward. Many scientists came forward to classify the organism, but everybody was having their own limitation. But finally, one classification which had many criteria in order to classify the organism, that got succeeded. So that is nothing but 5 kingdom classification. But before that, many scientists came. They classify the organism just based on very few characters. We call it as an artificial classification. But when you consider large criteria, as I have mentioned just in the previous slide, if you have classified the organism only based on the shape, it is only one criteria. Suppose if I consider many criteria about the shape, about the size, about their texture, if I consider everything and based on their particular character, if I keep separating, then obviously my work becomes so easier, right? So such type of classification, we call it as a natural system of classification, where many criteria has been included. So such work done by a scientist called R.H. Whittaker, we are going to study about that here in this part of the video. Okay, I mean here in this part of the chapter, because it's one short video. I can't say in this video, in that video. Okay, everything has mentioned in this video only. Listen to me carefully. If you just look at the time duration of the video, don't get panic. Oh, this is this much. It is about 5 hours, 6 hours. But remember, we are preparing ourselves for NEET, not for board classes, not for board exam. So that if I just read the thing superficially, I can answer the question. No, it is not like that. If I have to attempt for a NEET exam, we should be perfect enough in all the, in every line of MCRT. Okay? Yes. Now in the second part, R.H. Whittaker said there are total 5 kingdoms. One is Monera, another one is Protista, Plantae, Fungi, Plantae, Animalia. Okay, but here in this chapter, we are not going to give importance for all the kingdom at a time. So mainly 3 kingdoms detailed study we are going to do here. That means we'll talk about kingdom Monera in detail. Many information regarding Monera is not given in our NCRT, but I will tell you, as I said just now, we are preparing for NEET. If any question comes from this topic, we should able to answer. Remember, here if I'm standing here to do the class, it is not just by watching, seeing the question paper of just 1 or 2 years NEET paper. Okay, I have referred 10 to 12 years NEET paper. Based on that experience, I am teaching here. Okay, so don't neglect any part. If I'm writing anything, means just write down your copy. Okay, now the next part, we'll talk about kingdom Protista, and we even cover about kingdom Fungi. Then what about plants and animals, ma'am? Of course it is there, but very little bit information is given. Within one paragraph, they have covered both kingdom Plantae as well as kingdom Animalia. So we won't study here. We won't get more information in this chapter. Let us not focus much on this, because there is a separate chapter, as I have mentioned in the previous chapter itself, as plant kingdom and animal kingdom. There we'll study in detail. Okay. And at last of this chapter, there are few organisms which not kept under any of these 5 kingdoms. Okay. For example, the viruses. They do not included in these 5 kingdom classification. And there are certain agents, we call it as viroids, prions, lichens. So all these things do not come under 5 kingdom classification, but based on this part, you may get a question. So that we are going to cover at last of this chapter. Okay. This is what about the overview. Now, let's enter into the classification system. How classification system came into light and who did it. Overall, the scientists worked hard for the classification process. But earlier, at the time of civilization and all, the people were just classifying the things based on their needs, whether it is a useful things or not. Okay, if they want to see any organism, whether it is a useful, yes, this is one category. If it is not useful, this is another category. So it was not a scientific way of classifying, but to classify the organism which are so present on the earth based on the scientific knowledge has done by a scientist for the first time by Aristotle. So what Aristotle did is, he classified all organisms which are so present on this planet into two groups. That means one as plants and another as animals. So he made, once again, within the plants, if they are very long, he considered as the trees as one group, shrubs as another group, herbs as another group. So like that he classified. And about the animals, he made once again two groups. Animals with red blood, animals without red blood. But if you just look at these things, on which basis he might have classified? Obviously, how he could able to see an object just through its morphological character, he could able to classify. Suppose if I just look a plant, if it is very tall, okay, it is a tree. If it is average, medium height, it is a shrub. It is very small, it is a herb. That means based on the morphological character. Morphology means what? How we could able to see through our naked eyes, that is nothing but morphology. So like that he has classified. When he cut the animal, if it is having red blood, okay, you are one group. If you just cut the cockroach, it was not having red blood, no? Now you are in separate group. So like that, he classified the organism only based on morphological character. But here, it was not, though it is a scientific way of classifying, but it was not convenient. That means it was not so convenient. So that's why later one more scientist came. So his name is Carolus Linnaeus. Carolus Linnaeus. You know about him in previous chapter itself. Carolus Linnaeus, he was the person who introduced binomial nomenclature system. Correct? So remember, like such question may come in your exam. Carolus Linnaeus contribution, binomial nomenclature, Systema Naturae. Yes, Systema Naturae, written by him only. Binomial nomenclature, introduced by him only. Two kingdom classification, done by him only. So all of this could be the answer. So how you need to connect the chapter like this. Okay. Now, what he did, he said, I will make classification. So whatever the organisms are there present on the earth, I will divide into two kingdoms. So he made two kingdom classification. Okay. So in that two kingdom classification, again, he made these two category only. One he considered as Plantae, and in another kingdom, he placed all animals as a kingdom Animalia. So this is kingdom Plantae and this is, kingdom Animalia and this is kingdom Plantae. Now, ma'am, what is the speciality in this now? He also did in the same way, but yes, but he included few more criteria. For example, he did classification based on cell wall nature, not cell wall nature, based on cell wall presence or absence. So he said, if any organism, if do not have any cell wall, then you just go to this Animalia kingdom. If you have cell wall, just come here. So while doing such classification, the bacteria which were having cell wall came here only. Fungus which were having cell wall came here only. Many blue-green algae come under this category only. Plant itself, obviously. Because animals are the only organism, they do not have cell wall. Cell wall means what? If it is a cell, this is the plasma membrane. Around this, there is a one more layer, we call it as a cell wall. So those organisms, they have this, no? Yeah, yes. So they consider as a Plantae kingdom. But it was not so, once again, convenient. Because where the plants are, where the bacteria are, where the fungus are, so there is a huge difference between them. So that's why one more scientist came forward. So who is that? His name is Ernest Haeckel. So Ernest Haeckel came. Carolus Linnaeus, what you did, it is not that much correct. Okay, I will do three kingdom classification. I will group all the organisms in three categories. Okay, so that classification we call it as three kingdom classification. So here, what he did, he made three groups. He kept all plants in kingdom Plantae. He kept all animals, obviously, in kingdom Animalia. But he said, here bacteria or fungus, everything which are so there shouldn't be kept here in the Plantae. So that's why I will make one more kingdom as kingdom Protista. Okay, there I am going to keep all these organisms which are so unicellular, right? So like that he made 3 kingdom classification. But once again, not convenient. Why not convenient? Because just think, bacteria, fungus, how come close together? Because fungus is having total different nature, though it is having a cell wall. In fungus, we are going to see multicellular fungus also. The fungus are going to feed on dead organism. So, we won't feel comfortable here. So, that's why this type of classification also got failed. Then came one more scientist. Oh, here, one more thing I have to inform. Ernest Haeckel. He is the person. If you are a second PU student, class 12 student, if you are watching this video, you have heard the name of Ernest Haeckel, right? So, where we have learned? In evolution chapter. Just recall. In evolution, we were talking about certain evidences. Ontogeny, recapitulates, phylogeny. There, the same person gave the statement. Correct? So, you have to remember, here, the same person only tried to make a classification, a straight kingdom classification. Okay? Yes. Next. One more person came. His name is Copeland. He said, not 3 kingdom. I will make 4 kingdom. Okay? He did 4 kingdom classification. So, he said, you don't have any problem with kingdom Plantae and Animalia, right? Okay, let it keep as it is. Kingdom Plantae, kingdom Animalia, kingdom Protista. But you are having problem with unicellular and multicellular. Correct? You are having problem with prokaryotes and eukaryotes. I will give solution for that. So, there is obviously a major difference between prokaryotic cell and eukaryotic cell. Correct? So, if I say this cell as a prokaryotic cell and this as eukaryotic cell, what is the major difference between them? So, you all very well aware about this difference. Suppose if you don't know that also, I will tell you. Nothing to worry. Imagine this is the DNA. Okay? So, this is DNA. So, this DNA is present in a proper place, but around it, there is no any membrane bound cell organelles. There is no any proper membrane. So that, let me take an example like a bachelor home as well as a normal people home. So how we can able to differentiate? In a bachelor room, within a same room, he made a compartition like this by just putting a cot. Oh, this is my bedroom. Just by placing a gas stove, he'll consider that as a kitchen. So like that, he is going to made a compartment within that room itself. There is no proper wall between that. Okay. No proper compartment or a separate wall to separate those rooms. Whereas in the case of a normal house where you and me are going to live, no? In such houses, how it will be? There is a separate kitchen where wall will be there to bifurcate from hall, dining hall, puja room, kitchen, bathroom, bedroom, everything. Okay. So in the same way, here also in eukaryotic cell, if I am saying this is the DNA, if it has to present here in this particular place, means there is a separate membrane. So that I can give a place, I can give a name to this place as a nucleus. But here I can't say this is a nucleus. Okay, because there is no membrane around it. Just I have put a dot. Okay, there is no such dots, there is no such membrane. We need to think that, oh, this might be the nucleus because where DNA is present. As how we have considered just by looking at a cord, oh, this might be the bedroom of this person. Like that we need to imagine, right? So, such a membrane-bound organelles are absent in prokaryote, whereas it is present in eukaryote. Okay, so like that we can take many organelles, but here I am just giving only these nucleus example, that's it. Now, Copeland said, bacteria is a prokaryote and certain other organisms are eukaryote. How can you group together in Protista? Because prokaryotes, eukaryotes are totally different. It is having a huge difference. So that's why I will make one more kingdom called Kingdom Monera, where I am going to place all your prokaryotes. Okay, somehow it is sounds good. But where you are going to keep your fungus? Because fungus are not unicellular. Fungus are not even completely multicellular. Fungus are not can prepare their own food, though it is having a cell wall. So that's why one more complication arises. Then came our hero. He is nothing but R.H. Whittaker. He made 5 kingdom classification. So in his 5 kingdom classification, he included along with these 4 he included kingdom fungus also. Let me write here just kingdom Plantae, kingdom Animalia. I will write in detail in the next slide. Then kingdom fungus he had included. But as usual, kingdom Protista, kingdom Monera were as usual. Okay. Now let's study about 5 kingdom classification given by R.H. Whittaker. Okay. So on which criteria he made this classification? First, we should know about this, no? And before that, we need to understand one more thing. He gave 5 kingdom classification, R.H. Whittaker, in the year 1969. Very, very important year. Very, very important year. Here, from all this part, what you have learned, no, the chances of asking question will be very, very less. But when it comes to this, no, R.H. Whittaker, there is a more chances of asking question. But still, why I wrote all these things? Because it will be interconnected with other chapters. If any question comes as an option, you should be aware about it. That's why. Okay. Now, what are all the criteria he used for his classification? Okay. So, let me write the criteria. See, these are the main criteria used. So once again, this is a NEET question. By giving any of this, any 3, or it can be asked in the statement based also. By giving all these criteria should be in a 2 sentence. They may ask whether these 2 statements are right or not. So you should know. So he classified based on the cell structure. Okay, whether it is a unicellular or multicellular or body organization. That is what I said. And mode of nutrition, reproduction and phylogenetic relationship. Phylogenetic means, once again, what is the evolutionary relationship is there with this animal with the previous one. So he considered many criteria. Therefore, we'll consider this as a natural system of classification. Okay. So classification part, what is given in NCERT, that much we have completed. That means that part we have completed. And here, if you learn this table, okay, if you learn this table, then you will become 100% perfect. But before going to this table, let us understand few terms which are so given here in this slide. Okay. So what R.H. Whittaker made? 5 kingdom classification. Correct? I won't write kingdom again because already we know that kingdom classification only. So let me write directly as Monera, Protista, Fungi, Plantae and Animalia. So what he did, first, he kept all prokaryotes here in this group. So in the nature, what are all the prokaryotes are there should come here. Then rest of the things are nothing but eukaryotes. He placed here. So this group consists only eukaryotes. Right? And once again, if you just look at these 2 things, Monera and Protista, these 2 consist only unicellular organisms only. Unicellular organism. It means here, this is unicellular prokaryotes and this one, unicellular eukaryote. Correct? So one thing is very clear that these 2 kingdom consist only unicellular. And rest of the things, these rest of the things consist multicellular. Right? Now, one more criteria he took. Apart from cells structure as well as this body organization, he took one more criteria, that mode of nutrition. There is a main thing. So how we are going to eat, man? Because you have to survive means you should have a eating pattern. How we are going to eat? Then Plantae group organism said, we are independent. We are not depending on anybody. We can prepare our own food. So that let us keep in an autotrophic group. So these are nothing but autotrophs. Whereas this fungi as well as this Animalia, they cannot prepare their own food. Why? Because they do not have chloroplast. When the organism have chloroplast, then only they can do certain magic, right? They can capture carbon dioxide and you can use water. With the help of that, they can prepare their food. But here, they do not have such a green color pigment called chloroplast. So that's why they said, no problem. We will just chase the organism. We will hunt it. We will have it. That means we are depending on other organism. Whether you are an animal, we will eat it. Whether you are a plant, we will eat it. No problem. Anyhow, we can fill our stomach. So, such nutrition we call it as what? Heterotrophic nutrition. Heterotrophic. And this fungus also, I also cannot prepare the food because I am also not having chloroplast. So that's why here this one is also heterotrophic nutrition. But here, this heterotrophic is quite different. What is that different mode of nutrition? Even though it is a heterotrophic, such nutrition is nothing but it's a holozoic nutrition. Holozoic. It means how we are going to do. If any food piece, if we get, for example, if any bread piece or a roti or a anything, what we are going to do? First, we are going to ingest. Correct? We are going to ingest as it is. Then we are going to chew it. Then it will get digested in our body. Correct? So the first step is ingestion. We are going to ingest it first. Ingestion is the first step. And after ingestion, it will break into small pieces. That is called digestion. And when the food get digested into simple particle, then it will be get absorbed by the blood. That is called assimilation. Assimilation or absorption. And the last step, the unwanted material which is not required for the body, that will be ejected out, is called egestion in the form of fecal matter. Correct. So when a nutrition is having all these 4 steps, ingestion, digestion, assimilation and egestion, such a nutrition we call it as what? Holozoic nutrition. All Animalia group are having this holozoic nutrition, but there are few organisms that depend on dead organic matter. We call them as saprophytic. I can't write here saprophytic, I don't have enough space. Let me write here because the fungus are also show this mode of nutrition. That is, some are saprophytic and some are parasitic. So first, let me make you understand saprophytic. So how fungus are going to do? If you keep any bread or any vegetable just outside without keeping it in a refrigerator, so after a few days or after a week, how it looks? It will somewhat like a watery, right? So you cannot eat it, you cannot touch even, because it start producing a kind of smell, foul smell, because the organisms start decaying it. So we call it as a dead part. It's a dead and decaying organic matter. So who is going to do that watery liquid like structure or a texture in a food material? This is by this fungus. So such type of nutrition we call it as a saprophytic. Imagine this is a bread piece. A fungus is growing here. So first fungus, what it is going to do? Imagine this is a fungus. It is going to spit everything. Spit in the sense, the enzymes which are so present in its body, that enzymes are going to release out first. But in our case, in holozoic nutrition, the enzymes are present inside. So we say to our enzyme that you just keep inside only. We'll just take the food inside and we will give you. Okay. So enzymes are going to break the food inside our body. Whereas here it is totally reverse of it. Enzymes are going to release out first from their body so that enzymes will be present on the food particle. They are going to decay there itself. They are going to degrade the food particle there itself. Then just like how we are going to take the fluid with the help of a straw. In the same way, here they are going to absorb all the nutrition which are going to get digested outside. Okay. So such type of nutrition is called saprophytic. So in a simple terms, I can write it as it is depending on dead and decaying organic matter. Organic matter. Okay. Then parasitic. Parasitic is not like that. So organism, they are depending on living organism. Say for example, if any organism present inside our body, endoparasite, we are not dead, no, we are living. So inside our body it is present and it is taking nourishment. Or if any organism present on outside our body, they are called ectoparasite. They are taking nourishment from our skin, from our blood. So by situating outside of the host body. So such organisms are called parasite. So in a fungus group, we are going to see the saprophytic nature, parasitic nature. But actually they are not bad. So they are doing their work. They are taking nourishment. You are my food, man. So that's why I'm just present on your body. I'm taking all the food from you. Okay. So like that, they are taking the food. They are depending on living body or living organism for food. Such type is called parasitic. Here in Animalia group also, we can see few organism comes under this saprophytic group. Hope I'm very clear about how he made the classification. But not only this criteria, even he had included cell wall also. Because here, I'll do one thing. Hope we have written all these points. Let us go to this slide where the table is present. Just open your textbook. You can have with you and you can write whatever the points that I'm going to write here. Many points already have wrote there only. See, the first character what he took? Cell type. So you know, Monera are prokaryotes. Rest of the things are eukaryotes. We have understood. And the second criteria, cell wall. So once again, it is important for your NEET point of view. NEET point. See, Monera is having non-cellulosic polysaccharide plus amino acid. That means in a simple word we can write it is having peptidoglycan cell wall. Write down in your copy. Peptidoglycan cell wall. Okay. Whereas in Protista, some may have cell wall, some may not. Some may have silica cell wall. Okay. That you will study while doing this kingdom. Then fungi. It is having a chitinous cell wall. Write down in your copy. And Plantae is having cellulosic cell wall. Whereas in Animalia, this cell wall itself is absent. Okay. Next, the third criteria is presence of nuclear membrane. So if it is a prokaryote, nuclear membrane is absent and the rest of all organisms are eukaryote. Therefore, nuclear membrane is present. Just now we have learnt it. And body organization, both are cellular. See, both these are cellular, made up of single cell. Whereas fungus, it can be unicellular like a yeast or it can be multicellular also. So we can say multicellular, loose tissue, anything. Tissue, organ or organ system. So like such grade also we are going to see as we move further in the classification. Okay. Now let's come here. Look at here. The mode of nutrition. It is quite confusing part for the student. When it comes to Monera, Monera is having diverse group of nutrition. Diverse group means wide variety type of nutrition it can exhibit. Okay. I will tell you about this in detail about the mode of nutrition of bacteria while doing kingdom Monera, because definitely you are going to get a question from this part. But just know the names. It can be even autotroph or it can be even heterotroph. So within autotroph, we are going to see chemosynthetic or photosynthetic. And in heterotroph, saprophytic or parasitic. I will make you understand about these terms little bit later. Okay. Then autotrophic, Protista are autotrophic or it can be heterotrophic. So fungus are heterotrophic. As I said, saprophytic or parasitic, depending on dead organic matter or on living matter. Plantae, of course, they are autotrophic. They said, we don't want your relationship, man. We can prepare our own food. Just don't mess our life. Okay, so this is photosynthetic purely. But when it comes to Animalia, so heterotrophic, but still it is having a holozoic type of nutrition. Or some are saprophytic. Okay, so that is what this table is about. If you learn this table, once again, it will help you to carry 4 marks because any one question definitely may are going to come here. Okay. Now let's move on to the classification part is over. Now let's move on to the first kingdom, that is kingdom Monera. Okay. So here, one point we have to remember in our mind that when the kingdom Monera page opens, that entire kingdom conquered by only bacteria. Okay. So the sole members of this kingdom are bacteria only, because all are what? Prokaryotes. All are prokaryotes. Okay. And here, if I ask you a simple question, where you can find bacteria? So you can't say, ma'am, I can find bacteria only in soil. Don't you find bacteria in water? Of course it is there. Don't you find bacteria on your body? Of course it is there. Don't you find bacteria in a hot spring, thermal hot spring? Of course it is there. That means everywhere you are going to see bacteria. Okay, everywhere. Because they have lot of time to change their body structure, to undergo evolution. Because you know one thing, when Earth formed, almost all around 1 billion years, this bacteria are the only living creatures which are living on this earth. So that's why they were having lot of time to undergo getting various mode of nutrition and showing various level of respiration. So they could adjust themselves for a different environment because they are having such a large amount of time. right? So that's why this is a diverse group of organism where we can find all type of nutrition, all type of respiration, everything. Okay, and here, as I said, it will be having peptidoglycan cell wall. Now, based on the shape of this bacteria, if I have to divide bacteria into different groups, then I can make into 4 groups. Okay, based on shape. So what are those groups? The first group, if they are having like this spherical shape, we call them as cocci. Cocci, this is one group. If the bacteria is having rod shape like this, rod shape, we call it as a bacillus. Plural is bacilli, singular is bacillus. Okay, so here in our NCRT, they are even showing about the spore which is present inside. So this is the endospore. Endospore. Okay, suppose if the bacteria is having something like a spring shape, we call it as what? A spirilla or a spirillum. Spirillum is a singular. So here NCRT is also showing a flagella. Okay, like this, thread-like structures, hair-like structure. Okay, now the third category, if the bacteria are present somewhat like a water drops or a comma shape, we can say, comma shape, it is called Vibrio. Vibrio, it's a comma shape bacteria. So which is going to cause a disease called Vibrio cholerae. Write down, which causes cholera. Its name is Vibrio cholerae. Sometimes we don't know how the questions will be picked, how the questions will be asked. We should have all this information. Okay, so this is spherical shape. This is rod shape. So hope you are writing in your copy along with me, right? So this is spring shape or spiral shape, I can write. Spiral shape, and I have written here the comma shape. Okay, now if I just take any one bacteria, let me take a standard size bacteria, that is a bacillus shaped bacteria. If I mention the structure, how it looks. Okay, let me draw the structure here, bacterial cell. Okay, let us talk about the bacterial cell. See, let's assume this is the plasma membrane of a bacteria. And as I said, it consists cell wall also, correct? So, let me call this structure as a cell wall. So, which cell wall it is having? Peptidoglycan cell wall, correct? So, let me name it as cell wall. Now, above this cell wall, there is one more structure, which is actually giving protection to the bacteria. We call it as a glycocalyx cell wall or glycocalyx layer, okay? Let me consider this as a glycocalyx layer. So this glycocalyx can be of 2 types. In some bacteria, it is very tough and very rough in nature. Okay. So such glycocalyx is called capsule, which are very tough in nature. So the main function of this capsule is to protect the bacteria from the antibiotics and some external agents like temperature, pressure. So from such things, the bacteria can be get protected with the help of this capsule. But in some cases, what happens? Instead of having this tough layer, it will be having some loose layer, loose connected layer. So such a layer of glycocalyx only, but it is called slime layer. Right? It's a loose folds. But together of these 3 envelopes, or together of these 3 layers of plasma membrane, cell wall and glycocalyx is known as what? So this layer together called cell envelope. Or let me write here only cell envelope. Now, so this is just an outer layer. Now, if the bacteria wants to move, if it has to move from one place to other, of course, it requires certain locomotory structure. Just like how it is there for us in the form of a hands and leg. Okay, with the help of hands, we can't move, with the help of legs only. But here, the bacteria are having certain hair-like structure. With the help of those hairs, the bacteria can swim. Okay, so here, certain hairs are very short like this. They are small hair-like structure. They are called cilia. And certain hair-like structure, they are quite bigger, somewhat like this. See. It's very bigger. It is called flagella. So let me write the structure of flagella here. if you consider this as a flagella, the flagella will be having three parts. One is basal body. So this part is called basal body. Above it, one more connection is that this is called hook. And above this, there is a thread-like structure, a proteinaceous layer. It is known as filament. So these three parts together form a locomotory structure called flagella. Okay. Now let's go back. When you just get inside. So these are the structures, hair-like structure. And here one more hair-like structure we could able to see in the bacteria itself. So it is somewhat like this. These are called pilli. The plural form is pilli. The singular one is pillars. I will tell you the meaning of this as well as the function of these pillars in our upcoming part of the bacterial only, in the reproduction part. Okay. But you just know the name. This is called pillars. Plural. Singular. Plural together called pilli. Okay. Now, when you just get inside, of course, cytoplasm is there. But here, as I said, being a prokaryotic cell, it is not having a particular place or a membrane so that the DNA can be stored in that particular part. Okay. So that's why the DNA is simply present like this abruptly. So this DNA is also double-stranded. It's a double-stranded DNA. Double-stranded DNA. But the place where the DNA is confined, where it is present, no, that area, this area particularly, where DNA is present, is called what? This region is called nucleoid. Nucleoid. So usually you heard the term nucleus. Correct? But here it is a nucleus-like structure. It is not a nucleus exactly. Therefore, we call this area as nucleoid. It is also called gonophore. Gonophore. Okay. Now, inside, there is a long thread-like structure is that. This is nothing but the RNA or mRNA. Over this mRNA, just like a pieces of burger, how we are going to see, like that we are going to see a structure which are formed on this. So these are nothing but the ribosomes. So the ribosomes can be present separately also. Like this also they are present. So such ribosomes which are so present freely, they are a 70S ribosome. In the case of eukaryotes, it is called 80S ribosome. You are going to study in detail about all these components in our cell chapter, okay, in class 11th only. But the term or the number 70S indicates just its weight, mass. When you just subject this in a centrifugation process where it is going to get collected, so in that place we are going to give a number. One is 70S, whereas the other one in the eukaryote, what we are going to see, that is the 80S. We'll learn about all those things in detail, but you just don't worry about that. Now, you just learn this much, that bacteria is having 70S ribosome. Okay. Now, there are several ribosomes which are even present like this, attested to this mRNA. Okay. Now, such mRNA with lot of ribosomes are called what? Polysomes, which mainly helps in protein synthesis. Then when you just come here, almost all at the edge of the plasma membrane, we are going to see one more structure, somewhat like an invagulation, which is present like this. So this area is called mesosome. Mesosome. So mesosome is going to do a lot of function. Okay. So the first function performed by it is it helps in reproduction. So how it is going to help in reproduction? What is the nature of it? What is the process? How the process occurs? All those things no need to learn. Okay. So if you learn this much, that is more than enough. And on the surface of the mesosome itself, there are so many enzymes are present like this. With the help of these enzymes only, the respiration process can occur. So therefore, we can say the mesosome can even responsible for carrying respiration. And even it is responsible for cell wall formation. So you may get such question. Therefore, I am writing everything. How in our body the mitochondria is going to carry respiration process. So here that work is done by these enzymes present on the mesosome. Okay. Now inside the cytoplasm only, there is one more structure present, which are simply like this circular form. So the circular, this is also DNA only, genetic material only, but not it's a chromosomal genetic material. It is an extra chromosomal genetic material. So such things we call it as what? A plasmid. So you are going to learn about plasmid in detail in class 12. Those students who are class 12, that means class 12 student, if you are watching this video, you are very well aware about this plasmid, because you are going to learn in biotechnology, principle and process, because the plasmid acts as a vector, just like a vehicle to carry the genes. Okay. Suppose if you are class 11 student, nothing to worry. You just learn this much, that plasmid are the extra chromosomal DNA. Let me write here. So these are the plasmids, no? Extra chromosomal self-replicating DNA. Why self-replicating DNA? Suppose if this is a bacteria, if it is having its own DNA like this, chromosomal DNA, what I have mentioned here, this one, if it has to multiply, then whole cell has to get multiplied. That means the one cell has to become two cells, then only we are going to get two copy like this for the multiplication of this chromosomal DNA. Whereas the extra chromosomal DNA, what you are saying, that is a plasmid, it can multiply of its own. It can multiply of its own. Even though the cell is not multiplied, but within that it can multiply. Even if you transfer it into some other cell, some other bacterial cell, it can multiply. Okay. So that's why we say that it is a self-replicating. Okay. Or autonomously replicating. You can also call autonomously replicating. Replicating structure. So this is one more structure we'll find within the bacteria itself. Now bacteria says, there are few bacteria, they can prepare their own food, because just now in the table we have learned that the bacteria can show diverse group of nutrition, because there are so many bacteria, they can carry different mode of nutrition. Some are autotrophic, some are heterotrophic, right? If I say this bacteria is autotrophic, it must prepare its own food, right? But where is the chloroplast? How it can capture the sunlight? Suppose if it has to prepare the food. That's why it made an idea. So what it will do, it will create a structure. It will create a structure, something like this, something like this. Okay. So in this structure, what it will do, it will just store all the chlorophyll pigment inside. Okay. So such a structure, what you are seeing now here, where chlorophyll pigments are present, is called chromatophore. So inside we can write it is a chlorophyll. Right? Now, within this bacteria, again we are going to see few stored food material. For example, in a human body. Suppose if you are not eating anything, that is even the time duration is over, the time what you have fixed to have a food, that is in around 1 o'clock in the morning, in the noon, or morning 9 o'clock you have to have a breakfast. So like that you might have fixed a time duration, a proper time, that is a biological clock, it might have set in your body. For example, due to some reason, if you exceed that time, say for example, instead of having 1 o'clock, if you have your lunch 4 o'clock, we are not going to get die. Why? Because in our body, there is a food gets stored in our liver in the form of a glycogen, right? So that will make us to survive for time period, for time being. So like that, here also in the case of this bacteria, there are certain stored material will be present in the form of certain granules. Okay. So where shall I write? Let me write here only. Somewhere here, these granules are present. These granules are called inclusion bodies. Inclusion bodies. So let me write in the next slide. So what does this inclusion bodies are going to store? So inclusion bodies are going to store the glycogen. They can even store cyanophycean granules, or they store phosphate, that is in the form of phosphate granules. But together we can say that these are inclusion bodies. So this is what about a basic structure of bacterial cell. Hope I am very clear. If you want to make a notes, please make a note of this. Write down in your copy. It will definitely come to your help at the time of NEET exam. Okay. Now, If you just look at this image, what are all the parts? Just now I have mentioned, all these parts are present here also. Here, fimbriae part I didn't mention. Can you see the hair-like structure? These are called fimbriae. Cilia is okay, that is for locomotion. Fimbriae, it is to attest the bacteria. That means it helps the bacteria to attest on the surface of the host. Say for example, let's assume a bacteria which is causing disease on your lungs. Okay, not on your lungs, somebody's lungs. Right? Now, if it has to cause disease on the lung, it has to feed on the tissue present on the lung, means how it has to get attached there. It has to go and fix over the lungs, right? So that's why it should have some specific structure, so that with the help of those structure, it can hold the lungs or it can hold the tissue of the lungs. So those tissues, those structures are nothing but fimbriae. They are just like a finger-like structure. With the help of that, they can hold the tissues of a organism, right? And cytoplasm, ribosome, nucleoid, this is the place where DNA is present, is called nucleoid. And inclusion bodies, I said inclusion bodies can be Synophysian granule, it can be phosphate granules, it can be glycogen, anything it can be. Can you see all these are nothing but granules only, right? And you can see the extra chromosomal DNA, that is a plasmid. It is a circular in nature. And this one is pilus or pilus. See, it's a long thread-like structure. And this one, a very long thread-like structure, arise from the plasma membrane. Can you see this is the flagellum? And this one they have mentioned the capsule. Can you see the green color, very thick structure? It's a capsule. Inside that present white color layer cell wall, and inside that present a plasma membrane. So these are the basic structure, and I have covered more than these structures also. If you learn this much, that is more than enough in a structure of a bacteria. Okay. Now let's move on to bacterial respiration. Okay. See, as I said just now, few minutes back, the bacteria were having a lot of time to show their diversification. Because 1 billion years, what they can do? They can go for every places. They wanted to adjust to that place. Therefore, they were modifying themselves. They were evolving themselves in all these areas so that they have to survive. Suppose if a bacteria comes to a place where only oxygen is present, then they have to survive in that atmosphere where oxygen is there. Okay? Like that, if any bacteria move into the place where there is no oxygen, because when earth formed, that is in the primitive earth, there was no oxygen at all. Only reducing atmosphere, only volcanoes, only lightning, only the hot temperature, only that much. There was no oxygen at all. By that period also, we could able to see bacteria. Okay, but of course, it took certain period of time for the evolution of these organisms, but immediately oxygen didn't come. It took certain period of time for the evolution of oxygen also, for the formation of oxygen. But by that time, there were certain bacteria that could survive without oxygen. That means they had a lot of time for themselves to evolve to different area and in different environmental conditions. Therefore, whenever we just talk about a bacteria, all type of bacteria where they could able to show different type of respiration can able to see. Okay, so what are those mode of respiration? Let's see here. First of all, what is respiration? If anybody asks you this simple question, what you are going to say? What you are going to tell? Taking in oxygen, giving out carbon dioxide. So that is what your answer. But that is actually breathing. That is breathing. But when it comes to cellular respiration, so what should be your answer? It is nothing but breakdown of starch, breakdown of glucose. Why we are going to inhale oxygen? Do you know that? Of course, to get energy. Okay. So, of course, breathing is nothing but taking in oxygen only. But when you take that oxygen, where it will go? Of course, to burn a fuel. How we are going to burn a paper in the presence of oxygen? Because if you just close that paper with the help of a lid or anything, it won't burn. Because if there is no oxygen, how can you burn a paper, right? So, for everything, if it has to get burned, of course, we require oxygen. Okay. So, here also in a body of an organism, that is in the cells of an organism, when glucose entered, this glucose, it is just like a fuel. Okay. So, for the burning of this fuel, cell require oxygen. So, by combining with oxygen, the glucose is going to get burn in the sense it will undergo oxidation and it produces energy called ATP. So, that is what the energy. Why we are struggling a lot? Because to get this ATP. Okay. So, now There are certain bacteria require oxygen. There are certain bacteria, they say, we will get our energy even without oxygen. Okay. So based on that, we can categorize these organisms into two categories. What are those? One is anaerobic bacteria. Another one is aerobic bacteria. So here name itself is telling aerobic bacteria. So these bacteria says, of course, man, we require oxygen for our survival. Okay. So they need oxygen. They need oxygen. They need oxygen. And here anaerobic bacteria, they survive without oxygen. Without oxygen. Okay. Now, let us enter into the aerobic bacteria. So, within aerobic bacteria, again we found two groups. Some bacteria, see, look at here. This is one category of bacteria. They were simply happy. Why they are happy? Because they are telling that we are aerobic bacteria. We require a lot of oxygen for our survival. Suppose, if you just stop providing oxygen to this, what they will do? They will get die. For example, if a human being is kept in a room without opening any window or without providing any air at all, okay, so that there is no oxygen in that area or in that room. So how is he going to survive? Obviously, he is going to get die, no? Right? So in the same way, here also, if you won't provide oxygen to this bacteria, it will get die. So such bacteria, compulsory it require oxygen for their survival. We call them as what? Obligate aerobe. Obligate means must and should. Compulsory you need. Right? Suppose if you won't take, it will get die. Okay, let me write here. If no oxygen, they won't survive. They won't survive. If you won't provide oxygen, they won't survive. That means they are going to get die. Right? Now, if you come here, before going to this facultative, let me talk about this. Obligate anaerobe. So, obligate anaerobe means they are telling we are actually such creatures that we do not require oxygen. If you provide oxygen, then we are going to get die. That means our nature itself is like that. We should not survive in the presence of oxygen. If you provide oxygen, definitely we are going to get die. That means it's a poisonous to us. Right? So such organisms are called obligate anaerobe. Here, oxygen is toxic. Oxygen is toxic. So if you provide oxygen, they die. If you provide oxygen. So hope I am very clear about these two terms. Now, you will get confused in the rest of these two terms. That is, one is facultative aerobe, where I have written in the case of anaerobe part, and here facultative anaerobe, where I have written in the place of aerobic part. Right? I know you are getting confused, but let me clear your confusion. Listen. Here, this facultative aerobe means these are the organisms. These are the organisms. They are telling we are closing our nose because we do not require oxygen. Here also, let's close this nose. Okay, I have just closed its nose. Here also, I have closed its nose. So that if you provide oxygen, they are saying that we are going to get die. But here, there are certain bacteria which comes under the category of anaerobe only. They are telling, no brother, if you provide oxygen, it is a toxic to us. We will get die. But anyhow, okay, there are certain situations what to do. If oxygen comes all of a sudden, of course, we will survive. Because we have that mindset, we can survive even if any oxygen present. Okay. So, such organisms are called facultative aerobe. See, facultative means it's not compulsory. It's not compulsory. Here, it is compulsory. Compulsory you should not provide. Here, it is not compulsory. We will survive. We have to survive without oxygen, but even if you provide, we can survive. Okay, that is facultative aerobe. Even, let me write clearly, they survive without oxygen, but if oxygen is available, then also they can manage to survive. Manage to survive. That's why they are facultative aerobe. Aerobe means they require oxygen. I mean, the oxygen is there in that area. Oxygen is there in that area. Facultative means okay. If you provide, okay. If you not provide, then also okay. Okay. So like that here, it came to facultative anaerobe. So what it is saying, we are purely aerobic, man. We are purely aerobic. So if you provide oxygen, then only will survive. Suppose if you won't provide for a time being, for 1 hour, 2 hours, or few days, then also we survive. Then also we behave like anaerobe. Here they are telling we are anaerobe actually, but if you provide, we behave like aerobe. Okay, no problem. We can adjust to that. Here, if you provide, we will survive. But if you won't provide, we behave like anaerobe. That means we are flexible to that also. That's why the facultative name. Okay, let me write. So survive in presence of oxygen or survive in oxygen presence. But if you won't provide, then also survive in oxygen absence. Okay. So hope I'm very clear about these two terms, aerobic and anaerobic. So this is about bacterial respiration. Very simple. Okay. Now let's move on to the next topic called bacterial nutrition. Okay. So you have to listen it carefully. So easily you can understand. So what exactly nutrition? Taking food, having food. Correct. So that is nothing but nutrition. So everybody in one or other way, we are depending on others. That means if we are heterotrophic. Suppose if we are heterotroph, what we are going to do? Either we kill any animal to eat the food, or we are depending on any plant source, that is to get the food. Ma'am, we are not depending on any plants, ma'am. I mean, not on the animals, ma'am. We are just eating only plants, ma'am. Then also nothing but we are heterotrophic only, because we are the predator. If we are depending on the plants, means plants are the prey, okay, that we are going to learn in organism and population part. But in general, what we can write for heterotrophic, they cannot prepare their food. They have to depend on other organisms for food, okay. So let me write that. But depends on other organisms, okay. Now, when it comes to autotroph, so the situation itself is different. They are telling we are independent. We are not depending on anybody for the food. Okay, we can prepare our own food. So that's why we are called autotrophic. They prepare their own food. But how they are going to prepare their food? Okay, so preparation of food is also in a different way. Correct? See, how the food preparation process occurs in autotrophic mode of nutrition. So the plants are any organism. Usually the plants are considered as autotrophs. But here we are talking about bacteria. Okay, let us take in general as organism. Okay. So any organism, if they are preparing their own food, means they have to follow certain criteria. That means certain process. What is that process? Very simple. They will take a gas called carbon dioxide. To that gas, they are going to add some water. Right? So now, by taking certain energy, that energy can be of different source, that we are talking little bit later, or now itself let us talk. By taking some energy, they are going to prepare their food called glucose, right? So this is what the process which happens in autotrophic nutrition. But here, how we are going to get energy, man? Whether are you going to get energy from the sun or from some other source? Based on that, again, you are classified into two categories. That means one is photosynthetic bacteria, another one is chemosynthetic bacteria. Okay. So autotrophic means, yes, you are preparing your own food. But for your food preparation, who is providing energy? Okay. Then this photosynthetic bacteria says, this is a photosynthetic bacteria. Okay. So let's assume this is the photosynthetic bacteria. It says it's very simple. Can you see sun? Yes, sun is there. Sun is going to provide energy for us. So by taking the sunlight, we are going to prepare the food by combining this carbon dioxide and water, and glucose will be produced. So this is what our secret. Boost is the secret of my energy. Like that it says, the sun is the secret of our energy, of production of food. Right? Okay, we do agree. That's why your name is photosynthetic. Photo means light. Synthetic means synthesis. But now we ask the same question to this bacteria, that is chemosynthetic bacteria. So what chemosynthetic bacteria is, see. So this is chemosynthetic bacteria. It said, I hate sun. I do not prepare my food with the help of sun. But of course, I am also going to prepare the food. But question is, how? How? Because without energy, you cannot prepare. You cannot combine this carbon dioxide and water without energy to prepare glucose. Now the question is, how we are going to get this energy? Then this bacteria said, very simple, I will use chemical energy. Chemical energy means what? Imagine This bacteria is saying, imagine there is a substance, there is a chemical called A. Okay, let's assume there is A chemical. Now I will convert this chemical A into chemical B. Right? During this conversion, some amount of energy is going to release. That energy I am going to utilize in my food preparation. So that is what my secret. Okay. Then, how we are going to convert the chemicals? Can you please give some example? We are asking to this bacteria. Then it said, just look at the atmosphere. It is having lot of nitrogen, right? So how many? 78% of nitrogen is there. But you people cannot utilize. Even plant cannot utilize. Even animals cannot utilize. But I have that power. I can convert this atmospheric nitrogen, which you people cannot use, no. But I will convert into usable form. I am going to do some social service. Okay. That means here, all the atmospheric nitrogen which are so present, that will be in this form. That means in a triple bond form. No plants, no animals, nobody can utilize this. How we can utilize? We, in the sense, the plants can utilize only when it gets converted into ammonia. Ammonia. So this process is called ammonification. Or sometimes ammonia will be converted into nitrate. We call it as nitrification. Right? So during this process, we are converting from one chemical to another chemical, no? So during this conversion process, of course, some amount of energy is going to release. That energy I am going to utilize. Here I am doing two to work together. One thing is I am converting this chemical, and in other side I am preparing my own food. Okay, I am helping you guys, because without nitrogen you guys cannot survive. Because see, in your DNA nitrogen is there, in your amino acid nitrogen is there, in your body nitrogen is there. Because the protein itself is made up of nitrogen. Then how can you survive without nitrogen? So that's why say a big thanks to me. Okay, we did a salute to this bacteria because it is doing a lot of work. So such bacteria, we call it as what? Chemosynthetic bacteria. Okay. So all your nitrifying bacteria, let me write here. Example, all nitrifying bacteria comes under this category. Comes under this category. Okay. We do agree for that. Fine. We understood the meaning of this photosynthetic and chemosynthetic. Now, when I just enter inside this photosynthetic bacteria, which are preparing their food with the help of this sunlight, again there are two types. Why two types? Because there are certain bacteria, during this process, they release oxygen as their byproduct. But there are certain bacteria, everything will be same to same. Everything will be same to same. But apart from this releasing oxygen, they are going to release sulfur. So that's why we are going to classify them as anoxygenic and oxygenic bacteria. Okay. So here anoxygenic bacteria means they release sulfur. Right. So here the best example is green sulfur bacteria. Here green sulfur bacteria is an example. Whereas it comes to oxygenic bacteria, here what they are doing? They release oxygen. Right? So the best example here we can take blue-green algae. Blue-green algae. So blue-green algae are self-sufficient bacteria. They need not to depend on anybody, because I will tell you why they are so advantageous, why they are so forward in our next slide. Okay? But you just remember the name of this blue-green algae. Example, nostoc, anabina, oscillatoria, like this. Right? So one category of autotrophic bacteria is over. When it comes to heterotrophic bacteria, all bacteria which cause disease, they are nothing but heterotrophic bacteria. Do you think they are so crazy to cause disease in your body? No. But actually they are doing their work. They are taking their food from your body. But during this process, of course, as a side reaction, they are going to, side effect, they are going to cause the disease, because they are going to release certain toxic, that is once again toxic, certain chemicals that will be toxic to our body. Therefore, we are going to suffer from the disease. Okay. So let me write the example here as all the disease causing bacteria. So the disease causing can be in human being, animals, plants, anything. So all these bacteria, wherever you are going to see the disease in these organisms, they are all nothing but heterotrophic bacteria. That is one example. Like that, one more example we can take here. All good bacteria also. So we are going to say the curd is very healthy. The curd is having so many good bacteria in that, right? So what are those? They are nothing but heterotrophic. They are not actually not came inside the curd to help you guys, because they are taking their food. They are curdling means they are actually digesting that milk protein. They are eating that. During that process, they are releasing a gas. Therefore, lactic acid. That acid they are going to release. So that's why it is converting into curd. Okay. So I can say right here, the lactobacillus that we are going to study in your class 12th chapter, microbes in human welfare. And in that chapter itself, we are going to study a lot of bacteria. There are so many bacteria that are actually very helpful to us. Say for example, the bacteria that helps in production of biogas from the cow dung of animal. Okay, so there are so many bacteria that we are going to use in sewage treatment. That means the drainage water, unwanted wastewater, what you are going to consider it, no? All those wastewater will be converted into usable form with the help of this bacteria. Biogas is a usable form of gas produced from this bacteria. Okay, so many chemical agents can be produced from this bacteria. Antibiotics can be produced from this bacteria. Okay, so like that, if you just keep making the list, we can get a lot. But in general, I can write all good bacteria, all bad bacteria. Everything comes under the group of heterotrophy. Okay, so that's why if you just look at here, we can say the bacteria will be having a diverse mode of nutrition, right? In detail, they are not given in NCERT, but if you just read the line, then you will understand why ma'am has told all these things. Because in certain lines, certain word, they have put these information in a unknowing way. So only if you understand the concept, you can able to understand the meaning of the line. Okay. Yes. So bacterial nutrition part also over. We have done. The next is, let's move on to bacterial reproduction. So how bacteria is going to reproduce? So bacteria, they do not have any different sexes like a male and female, how we are going to see in the case of human body or in human being. But still, they are going to reproduce. But that reproduction process in different way. One is binary fission, it is the asexual mode of reproduction. Another one is endospore formation, that is also asexual mode of reproduction. And the last one is genetic recombination. I will tell you about that. So before going to the genetic recombination, let us understand what exactly binary fission means. See, this is NCERT image. Here what they are showing, one bacteria is dividing into two, correct? So how it actually happened? Look at here. So let's consider this as a bacteria, and this bacteria is having its own DNA, right? Now, during the process of reproduction, it will create one more copy of its DNA, so that within itself, the two DNA molecule we are going to see, right? Now, this bacteria, it will just split in the middle, okay? So as a result, what happened here? The two bacterial cell we are going to see, in each bacterial cell, this DNA will enter. So as a result, the two bacteria we are going to got, so these two bacteria are called daughter cells. So once again, after attaining growth, again they become mature, again they are going to divide. So like that, this process keep happen. This is called binary fission. Okay. So the time duration of this bacterial fission can be varies. Some bacteria can take 1 hour to get divided into two. Some bacteria within 20 minutes can divide into two. Okay. So like that, it depends on kinds of bacteria. But binary fission is also a type of reproduction. Right. Next, endospore formation. So what is endospore formation? Let's imagine here is a bacteria. This bacteria, it was so happy in its life. But all of a sudden, it is crying like anything. Why crying? Because it came to know that from external, that means it is getting actually a lot of trouble from external environment. The temperature could be higher, the outer surrounding environment could be so toxic, so that this bacteria might be feeling suffocation. Okay, it is unable to survive. Then what happened? What it will do? It will make an idea. What it will do here? It just doubled its DNA content, so that one copy become two copy now, because it will know anyhow I'm going to get died. At least my genetic material, let it get survive for future generation. So that's why it has doubled its genetic material, and after its doubling, it started creating a wall around it. A thick wall started creating around this bacteria. Around this genetic material. Okay, so it's a very hard layer, hard covering. So this whole structure, what you are seeing now with the covering inside genetic material, it's together called endospore. Endospore. Now, it is not bothering at all, because I can just come back like a phoenix, right? So even if you kill me, my genetic material is there, right? Now, external pressure act on it, this bacteria got killed. But still what happened? Here the endospore is surviving. Even though the bacteria has got killed, endospore, still it is there. It is surviving in the environment. So endospore is so stronger that it can survive even thousands of years. If you boil in a very hot water, then also it survives. Okay, hundreds of years it is going to survive. So that much strong the endospores are. Understood? So that's why it is actually, instead of telling a kind of reproduction, we can say that it's a type of protecting itself. But as it is doubling its genetic material, that's why we have put them into a category of reproduction. Okay. Now, when the surrounding environment, atmosphere become favorable, so automatically what happened? This outer layer, it got burst out, so that this genetic material, anyhow it is having its own information, no? Genetic material means it again start creating itself, so that the new cell has found the genetic material will be lying as it is, and this bacteria again happy. Understood? So this is how it is going to survive in unfavorable condition. If you just look at this image, hope it's not very clear, but still somehow you can manage to see here. See, a blue colored dots. Can you see here in this bacteria? It is somewhat like this. The bacteria, it is red color. Inside this red color, the blue color spores are there. These are endospores. Okay. So endospore is a kind of protection method. Now the last category. Let's come back. So the last category of reproduction is genetic recombination. So let me tell you. As I said few minutes back, it is not having separate sexes like male and female. But still, why we are giving the term genetic recombination? Because here it is going to exchange its genetic material. Imagine this is one bacteria. This is another bacteria. This bacteria is having green color genetic material. This bacteria is having, okay, let me write in different color. So this is the genetic material of this bacteria. Let's assume. This is the genetic material of this bacteria. Now, because of any reason, if the part of genetic material, if come and attach here to this bacterial genetic material, then it is called what? Recombination. So that recombination process can occur in these three different ways. One is transformation. Another one is transduction. The third one is conjugation. Transformation, it happened by chance. Okay, it is by chance process. It is not going to happen purposely. Whereas transduction can take place with the help of virus. And conjugation, it happened with the help of pillars. I said no, I will tell you in our upcoming slide about the pillars or pili. So here it is a process. So let us discuss about these things one by one. Transformation means what? Very simple. All are very, very simple. Okay, what is transformation? Imagine this is a bacteria. This bacteria. It is having its own genetic material. But now this bacteria is not happy because it is knowing that I am going to get died shortly. Okay. So now it got died also. Let's assume it got died also. But when it get died, its genetic material just remain in the environment as it is. Now, here, next to this bacteria, let's assume there is one more bacteria is present. But this bacteria is not facing any such difficulty. It could able to manage its genetic material or its cell easily. Now, what it will do? It is having a tendency, a kind of tendency it is having. So, what is that tendency? Easily to uptake the neighboring DNA, whichever be the DNA which is presenting on its surface, that is in its surrounding, easily it will uptake it. So that tendency of uptaking the neighboring DNA, surrounding DNA, is called what? Transformation. So because of that, this DNA will enter inside this bacterial genome, this bacterial cell. Okay, so that's why what the definition we can say are right. Uptake of naked DNA. Uptake of naked DNA from surrounding by bacteria is called transformation. It need not to be a bacterial genome. Suppose this bacteria, if it has to uptake any DNA, means this DNA need not to be a bacterial DNA. If it is any other fungal DNA, then also it can take. So that power it is having. That process we call it as a transformation. If you are a class 12 student, once again I am giving one more hint. Not hint. I have already done. There is a genetic part. Hope you have remembered about the transformation process where we done in Griffith experiment, right? So what happened during Griffith experiment? R strain bacteria, S strain bacteria, when a process occurs, the R strain become S strain because it uptake the naked DNA of S strain bacteria. So that's why the R strain also become S strain, correct? So if you are a class 11 student, don't worry, you are going to learn this in your class 12, okay? But you just know the process. Understood? This is one category. Here genetic material has got incorporated with this DNA. Okay, so that it will consider this DNA as its own DNA, right? So this is one process. The second process is transduction. This is also very simple. Transduction takes place with the help of virus, I said. Okay, now let's assume here is a bacterial cell. Okay, so this bacterial cell, it is having its own DNA. Now, in the life of bacteria, a villain entered. So this villain is nothing but bacteriophage. So what it will do, soon after the infection, first it will incorporate its DNA. So imagine this is a viral DNA. So this virus itself we call it as a bacteriophage. Why bacteriophage? Because it infects bacteria, that's why. Now, the first thing, what it will do is, first it will break this bacterial DNA into small pieces. This is viral DNA, no? Now, the bacterial DNA become into small pieces like fragments, like this, right? It becomes small pieces. Now, it is so happy. It is start multiplying its own copy. So, as a result, when you just look at here, let me write in this color so that you could be able to distinguish. Now, small baby viruses are start forming inside this bacterial cell only. They start replicating. So within that, this copy of DNA also got doubled, no? They start incorporating inside. But sometimes what happens? Sometimes, by mistake, this DNA along with its own DNA, it also got mixed with this bacterial DNA. Okay? That means viral DNA along with its own DNA, it also got combined with that of the bacterial DNA. Okay. Now, this cell got burst out. These viruses release freely outside. Now they are releasing outside. Now they are targeting for another bacterial cell. Okay. Now imagine this this virus, which is having its own piece of DNA and along with the piece of bacterial DNA also now enter or attacking one more cell or one more bacterial cell. So as a result, this is a bacterial DNA, right? Now it freshly attacking here. So during this process, what happened? So it will incorporate its DNA, but along with that, this viral DNA also came. Along with viral DNA, bacterial DNA also came. Correct? The pieces of bacterial DNA also incorporated here. So now it got mixed with this. Suppose it could be strong enough to kill this viral DNA. Then if this bacteria is surviving, then it sees that, oh, this is somehow looking my neighbor only or my category only, so that it can incorporate here along with its DNA. So that is once again a genetic recombination. So this is nothing but transduction, which happens with the help of virus. Okay. Now the last part of genetic recombination is conjugation. Okay. So here in conjugation, what happens? It won't happen in all bacterial cells. There are few bacteria there are few bacteria which are having that pili, pillus or pili. They will be having a special type of plasmid. Okay, see, listen. Let's imagine this is a special type of bacteria. Of course, there is a chromosomal DNA is there, but along with that, it is having a special type of plasmid in it. So, such plasmids are called F plasmid. F plasmid, and such bacteria are called F plus bacteria. Why F plus bacteria? Because these bacteria are having F plasmid. Here F stands for fertility plasmid. Fertility plasmid. Now what this will do? It will just make a contact with the neighboring cell. So this cell, it is saying, I am having my own DNA, that's it. But I am not having any fertility plasmid like you. Right? So that's why my name is F minus bacteria. F minus bacteria. Because I don't have any fertility plasmid inside me. Then it says, that is F plus bacteria says, don't worry my friend, I am having a special structure. What is that special structure? There is nothing but a pillars or pili. So with the help of that pillars, that is a tube, I am going to insert in your cell. Okay. So I am going to insert my tube into your cell. So that with the help of this tube, I can transfer one of my plasmid into your body. But before transferring, I am going to make another copy of it. Wait, I will make another copy like this. Another copy. Now I will send this copy to your cell. Now what happened? What happened? Now one plasmid is here, one more plasmid can also be present here. Now do you call this as F minus plasmid or F minus bacteria? Of course not. Because it also got F plasmid here. That means the genetic material has entered here in other cell also. That's why it will also become F plus bacteria only. Understood? So this is how, with the help of this pili or plural pili, singular is pillars, a type of bridge which is so formed now, this is called conjugation bridge. Conjugation bridge, with the help of this, the genetic material can be transferred. Understood? Here if you just look at this image, you can understand. Can you see? So here, this is one bacteria, this is another bacteria. Now, you can easily look at the small tube which is just entering inside the other bacteria. This is nothing but pili. So, hope I am very clear, crystal clear about all these processes of nutrition, respiration, reproduction, everything. Now, we are talking about Monera only, kingdom Monera only. Now, let us enter inside what NCERT has given. Because within NCERT, again 2 groups within bacteria has provided. Based on that, the chances of asking question will be more. Let us focus on that. Now, the kingdom Monera, which can be grouped into 3 group of bacteria. But what are those 3 group of bacteria? Let us write down that first. One is Eubacteria. The second group is called Archaebacteria. And third category within Monera, we are going to see as Mycoplasma. Mycoplasma. So before studying Eubacteria and Mycoplasma, let us focus on Archaebacteria. Archaebacteria are most primitive bacteria. They are most primitive group of bacteria. Why they are so primitive? Because they are saying, I am the senior compared to you both. Because when Earth has formed, no, I am the one who conquered the whole Earth. Because do you know how our Earth atmosphere was? It was very harsh. Now you guys are coming that we are saying, oh, the bacteria I can found here, I can found here. But look at the Earth atmosphere when it was 4 billion years ago, 3 billion years ago, not 4 billion. Okay. So by that time, it was so harsh. It was full of acidic in nature. It was full of high temperature, full of saline environment. But still in that condition, I was the one who was able to survive myself. Okay. So that's why I am the primitive bacteria. And here, these Archaebacteria, they are so smart enough that they have few features like Eubacteria. And even they have a features like eukaryotes. Right? So let me write that. Eukaryotes and Eubacteria. So let us talk about this first. Archaebacteria. Archaebacteria, I said they are called primitive bacteria. And we also say that As you said, no, you could able to survive in extreme condition like high temperature, high acid, high salt condition. Therefore, we are going to give a title as extremophiles. Extremophiles. Why extremophiles? Extremo means extreme condition and philes means love. So, you are the lovers of extreme condition. No? That's why we have given a title called extremophiles. Just go and enjoy. Okay, fine. It said, okay, fine, no problem. Just look at my categories. It is saying, just look at the categories. So, we came to know that this bacteria is having 3 categories. So, one category of bacteria, that is nothing but Archaebacteria only, which could able to survive in high salt concentration, very high salt concentration. So, that's why we said, you are Archaebacteria, but as you live in high salt concentration, no? So, that's why your name is halophiles. Halophiles means survive in extreme salt concentration. And another group is there. They could able to survive in very high temperature. Therefore, we have given a title as thermoacidophiles. And another group of bacteria, they are not actually extreme condition, but they could able to release methane gas. That's why we kept a name as methanogens. Again, you are going to see this term in Microbes in Human Welfare chapter. So, in that chapter, we are going to talk about the biogas formation. So, biogas means it is produced from the cow dung. So, all the organisms which are having a part called rumen, we call them as ruminant. For example, cattle, buffalo. So, all these organisms, they have a rumen. Just like how we are going to say kidney, liver, intestine, small intestine, large intestine. In our body, kidney separate. Like that, in the digestive tract of these organisms, rumen is also a part. So, such animals are called ruminants. So, in all those ruminants, these methanogens will be present. Okay. So, what these methanogens are doing? Because we all know that the cattle, buffalo, they are going to take grasses, they are going to eat grass. Do you think that these cow, buffalo itself are going to digest these grasses? Definitely not. They will digest the grass only with the help of this methanogen. Right? So, that's why as they are living in the rumen of these animals, therefore, they are anaerobic in nature. Anaerobic. Remember one thing, any organisms which are present inside the body of other organisms, they are always anaerobic because they could be able to survive without oxygen. And here they produce methane gas. Methane gas is also denoted as CH4. It is also called biogas or marsh gas. Right? So, here a group of bacteria are there. Methanogen is a group of bacteria. In that, the main bacteria name is Methanobacterium. Methanobacterium comes under this category only. Understood? So, these are certain characters, certain points you have to remember regarding Archaebacteria. Now, if any question comes in your NEET exam, it will be asked only from these lines. Halophiles, thermoacidophiles, methanogens, or extremophiles, or even primitive bacteria. Right? So, if such question comes, you should not get panicked. They are asking about Archaebacteria. In certain modules and in certain other certain modules, the question can be asked in that way so that what is the main nature of Archaebacteria so that it is different from some other bacteria. The answer is need not to remember the process, need not to remember why these are surviving in such condition. Only if you learn, it is mainly because of different cell wall nature, different protein nature. So that is more than enough because such questions will not be asked in your NEET exam as it is not given in our NCERT. Understood? Okay, let me write. These are different because of having different cell wall nature and protein nature and protein. So that's enough because it won't be as stressful. Now the next group of bacteria is nothing but Eubacteria. Let us talk about this. Yes, Eubacteria means true bacteria. Here the term Eu refers to true. True bacteria. It's a common sense. Why we call it as a true bacteria? Because we know that they won't prefer extreme environment, extreme condition, just like Archaebacteria. Okay, so they prefer to live in a normal environmental condition. Therefore, we consider it as a true bacteria. Okay, then where they are going to prefer then? That means where they are going to grow or survive. So when you just make a list of their habitat, we are going to found almost all everywhere. Suppose if you ask them to found in terrestrial, they say yes. In terrestrial habitat also, we can found this Eubacteria. Like that, if you just go to aquatic environment, there also we can found this Eubacteria. Aquatic means water environment. Okay, so within aquatic, we are going to found even in fresh water as well as in marine water, sea water. So in everywhere, we are going to found this bacteria. Now, if I just look at this structure, so we'll come to this diagram later. But first, let us understand few general characters regarding this Eubacteria. Okay. Now, if we just examine the structure of Eubacteria, almost, we are going to found in a unicellular, obviously. Some organisms are found in a filament-like structure like this. Filament in the sense, one cell is going to attach with other cell like this somehow. This is called filamentous structure. So like such structure also, we are going to get here. And some bacteria, they occur in a group. We call it as a colony. So it is found even in colonial way. Understood? So this is how certain general character we are going to found in this bacteria. Now, if you get a question, ma'am, can you please give some examples regarding Eubacteria? Then we can make a lot of list. All your disease-causing bacteria are nothing but Eubacteria only. Okay. Here few diseases names also mentioned in NCERT. If you know those diseases, that is well and good. Let me write here. All disease-causing bacteria. Either the disease can be found in plant or even in the animal, but all are nothing but. If the bacterial disease you are going to found, that is nothing but Eubacteria only. For example, we heard about cholera, it is by Eubacteria. We heard about typhoid, again it is a Eubacteria, right? And we heard about citrus canker, which occurs in plant, again by Eubacteria. Okay, so like that, if you just make a list, we can get a lot. Understood? So this is one category within Eubacteria. Then if you come to know about the good bacteria, as I said a few minutes back, so many good bacteria are there because we cannot consider whenever we heard the term bacteria or villain only or culprit only. There are so many good bacteria also there. For example, good bacteria like lactobacillus, which helps in production of curd from the milk, and even, where shall I write? Let me write here only. And the bacteria which help in production of biogas, and bacteria which help in nitrogen fixation. Only few lists I am making, but if you just keep making the list, even in the case of disease causing as well as in the good bacteria, we can get plenty of bacteria we can get. But all these belong to, either it is a good bacteria or harmful bacteria, they belong to the group of Eubacteria. Okay, now we are not focusing here on good bacteria. We are not even focusing on bad bacteria. We are focusing mainly a group of bacteria within Eubacteria. We call them as blue-green algae, right? Because the name itself is telling blue-green algae. They might be having some greenish color or greenish pigment. That's why the name blue-green algae. Let us focus on that only because it is given in our NCERT. Now, let me write here itself. Within Eubacteria, let us talk about what? Blue-green algae. It is also called BGA or cyanobacteria. So first, let me give some example of this blue-green algae. Have you heard the term nostoc? Nostoc comes to this group. Oscillatoria. This is also belongs to this blue-green algae only. Then Anabina. So all belong to this category. And all these are filamentous. As I said, they have diverse mode of nutrition, and especially when it comes to the term of this blue-green algae, they are self-sufficient bacteria. Why self-sufficient bacteria? They need not to depend on anybody. If you take the example of this disease causing, they need to depend on the host, either on the plant or on the animal. That is, of course, if it is causing disease, means the disease tissue which is so present in a body of a host animal, they have to depend on that. If it comes on good bacteria, again it has to depend on the biogas, not biogas. It has to depend on the cellulosic material, that is a plant source which is present in an animal body, or it has to depend on the milk. Suppose if it is a lactobacillus, like that. I can take many examples, I said, no. And even methanogens, they have to depend upon the methane gas. One second, student, one second. Here, let me not take this biogas, because biogas is by methanobacterium that already we included in Archaebacteria. Let me take this lactobacillus nitrogen fixation, like that even in sewage treatment. So like such example we can take. Okay, bacteria and sewage treatment. So one or in other way, they have to depend on other organisms or other source for their food. But here, these blue-green algae, they are self-sufficient because they says that we need not to depend on other, because we have chlorophyll pigment. We can prepare our own food. That means we can say that these are autotrophic because they prepare their food. How come it is possible for the food preparation? Of course, they have a pigment. So here, they have a pigment called chlorophyll A. It's a green color pigment. So that's why it can capture the sunlight, and with the help of that pigment, they can prepare their food. Okay. And here, one more speciality of this is, whenever, if a plant requires nitrogen, they depend on bacteria. Correct? That's what I said in chemosynthetic bacteria. And here, the speciality of this blue-green algae is that they need not to depend even on chemosynthetic bacteria also. That is for nitrogen. This is that we can prepare or we can fix the nitrogen ourselves, because we have certain special enzyme, right? That means here they have a capacity to fix atmospheric nitrogen. So let me talk about the structure of this blue-green algae so that let us understand all these points. And here one more important line which is given in our NCRT about this green pigment. As this blue-green algae, which is having that greenish pigment, no, when they get lot of source, they start multiplying like anything, so that it will cover entire surface of the water. Because I said in the beginning itself, they found even in the fresh water. Okay. Now, if I just draw a tank or a lake or anything which is having water, that is a water body. Now let's assume here is a blue-green algae. It is somewhat greenish in color. Now they got some good food source. Okay, or if they having a sufficient atmosphere, they have certain nutrients, everything is provided, then automatically they start multiplying like anything. So it almost looks like a green cover or a green mat, as if you are going to spread on the surface of a lake. It is known as algal bloom. Algal bloom. It is formed by this cyanobacteria only. Algal bloom means blue-green algae, no? That's why algal bloom is the name given to this. So because of that, many aquatic animals which are so present inside, say for example, if any fish is present or any other animals are present or even plants are present, they won't get a proper sunlight because it will be covered by these blue-green algae which form an algal bloom, so that they start getting died because of lack of oxygen. So there is a separate scenario, there is a separate part, there we are going to study. But here when it comes to the points what I have mentioned here, these points will be get understood only when we understand the structure. Okay, let me draw the structure of this. Let's consider this is one cell, this is another cell, this is another cell. Like that, many cells are present in a filamentous form, right? In all these cells, the process of photosynthesis is happening. How? Because they have a green color pigment in them. Let me show only one cell here. So these are chlorophyll pigment. So like that, which is present in every cell, but except few cells. Imagine this is the cell this is the cell where you can't find the chlorophyll pigment. Why it is not present? I will tell you later. But rest of the cell, here, in everywhere, we are going to see this chlorophyll pigment. With the help of that, what they will do? They take carbon dioxide and combines with water. Then, obviously, they are going to produce food called glucose. And during this process, they are going to release oxygen. So, oxygen gas will be released. Correct? So, this is nothing but photosynthesis process only. They can carry out. That's why we can say that these are autotrophic. Autotrophic. And one more benefit or advantage I said that they can fix their atmospheric nitrogen. How come it is possible? These organisms says you have nitrogen in your atmosphere. And this nitrogen has to get converted into ammonia. Correct? This is nothing but nitrogen fixation. But for this process to done, we need not to depend on anybody. Instead of that, we have certain organisms, or not organisms, we have certain enzymes. So these are enzymes. These are nitrogen fixing enzymes. They can do this work. Okay, they capture atmospheric nitrogen, they will convert it into ammonia so that in my body itself ammonia will be available. Right? So, this process will be done by enzymes. So, that's why they have to bother? Not even for the food, not even for the nitrogen. If everything is happening in their body itself, therefore, obviously they are independent. Correct? Now, here, I said all the cells are doing this process. Every cell is releasing oxygen by producing glucose. Everybody, here also glucose is forming, oxygen is releasing. But why it is not happening here? Because of this enzyme. This enzyme says, if I have to do this work of nitrogen fixation, here you have to prevent, you in the sense the cell has to prevent from this oxygen. Because this is sensitive to oxygen. Sensitive to oxygen. If any oxygen enter or touch this enzyme will get damaged. So that's why it will just get covered inside the cell, a special cell of this nostoc. I mean this filamentous algae. So that's why it even made a plan. It will have a very thick wall also. So cell wall, peptidoglycan. So that cell wall itself becomes so thick. Just to protect itself from this oxygen, because everybody is releasing oxygen. If this oxygen, by chance, if it enter into the cell, it will get damaged. So therefore, it will prevent it. So such a cell, where we can find this nitrogen fixation, as well as the enzymes, which are sensitive for this oxygen, but help in nitrogen fixation. So this special cell is called heterocyst. It could be your NEET question, heterocyst. It helps in nitrogen fixation. Right? Now, this cell, as I said, these are filamentous in nature. Correct? So, to make all these cells to be in a filament-like structure, there is a mucilaginous sheath present like this, so that it can hold all the cells together. Understood? Suppose if it is a cell, if these cells are present in a filamentous form, there is certain mucilaginous sheath is present, like this. This is a mucilaginous sheath, which holds the cells together in a filament structure. Understood? So, this is about Eubacteria. That's it. Now, let us look at this diagram. So, this is NCERT diagram. Here, you can see this is the nostoc. It is a filamentous, once again, blue-green algae only. Here, outside covering, what you are seeing now, here, this outer covering, this is nothing but mucilaginous sheath. A dot structure has given, and inside there is a special cell which has been marked with the different color. This one. These are called heterocyst, which helps in atmospheric nitrogen fixation. And in rest of the cell, small dots are given. Dots are given inside. That is chlorophyll pigment. So this chlorophyll A, the pigment chlorophyll A, which is so present in these organisms, it is a characteristic feature of higher plant. Because in higher plant, green plant, we see the same pigment, chlorophyll A. Okay? So, the same pigment we can find here also. So, that much is enough about Eubacteria, nothing to worry. Rest of the things already have done in the reproduction part, nutrition part, everything I have covered. Now, we are in the last part of kingdom Monera, that is about the last category. Okay, next is Mycoplasma. So, till now, we have studied about Archaebacteria. So here Archaebacteria said, I am totally different. Why totally different? Because I am not having peptidoglycan cell wall, but different cell wall I have. Unique cell wall. And here, different lipid and protein in their plasma membrane also. Because the lipid chain is somewhat branched chain. Whenever we talk about plasma membrane, it is made up of lipid and protein. So that lipid, it is quite look different. In a branched structure, it looks. Where in the case of Archaebacteria. When it comes to Eubacteria said, we are okay with our own peptidoglycan cell wall. Correct? That means in both the cases, cell wall is there, but maybe of different nature. But when it comes to the question of mycoplasma, so here organism is there, but it is not having cell wall at all. So there is no cell wall. No cell wall. So it is very unhappy because nature has cheated. It has given a unique cell wall to this, peptidoglycan cell wall to this, but not any cell, not of any kind cell wall. Okay. So that's why it got upset, and at the same time, it started taking revenge on other organisms. Why it started taking revenge? It became pathogenic. Not all of mycoplasma, majority or most of the mycoplasma become pathogenic. Pathogenic means disease causing in both plants as well as even in the animals like human being. So this mycoplasma, it is the smallest living cell. It is the smallest living cell. So it said, okay, no problem. I don't have cell wall, but I can live without oxygen. So without oxygen also, it could able to survive. And here, we can also called by a term called PPLO. Mycoplasma is also termed by PPLO. PPLO means pleuropneumonia-like organism. Majority of the lungs disease. Pneumonia is a lung disease. Okay. So you will study about pneumonia in your class 12 in human health chapter, human health and disease. Suppose if you are class 12 student, you are aware about this. So this pneumonia-like organism is nothing but mycoplasma only, which is responsible for causing such lung disease. Almost all 1/5 of mycoplasma, what are there in the nature, they are responsible for causing lung disease. Right? So let me write the full form here. Pneumonia-like organism. Understood? So the same information given in our NCERT also. Look at here. Look at here. The mycoplasma are the organism that completely lack cell wall. Again, it's a NEET question. They are the smallest living cell. Again, NEET question. Now, and can survive without oxygen. Right? Many mycoplasma are pathogenic in animals and plants. If you just look at this structure, you can see here the cell wall structure has not shown. Only lipoprotein membrane is there. Lipoprotein membrane in the sense they are talking about plasma membrane only. And it will be having a protein inside, ribosome inside. So this structure anyhow, it is present in every bacterial cell. And DNA, some soluble RNA, everything is there, but not cell wall. Understood, no? So this is about mycoplasma. Now we have completely done the kingdom Monera. Now let us check our knowledge based on the classification part as well as the kingdom Monera. So like this, after every topic, let us discuss few questions which are quite interesting, which are concept based and even applied based. Because the previous year question easily you can solve. But if the questions asked on the concept basis are the questions, if it is on the applied level, then you have to think and answer. So when you start thinking about the concept, when you start applying your knowledge on the question, then it is confirmed that you have understood the concept very well. Right? Now, let's solve the question. Question number 1. Which of the following are absent in Eubacteria? So, Eubacteria in the sense they are talking about true bacteria. So, here 80 is ribosome. Do you find 80 is ribosomes? Whether it is a Eubacteria or Archaebacteria, whatever, whenever the kingdom Monera comes, always it have 70 years ribosome. Remember it. Because it is a prokaryotic cell. So therefore, which of the following is absent means this is absent. Then it says cellulosic cell wall. Correct. This is also absent. Because either it is having peptidoglycan cell wall or some other nature, but not cellulosic cell wall. Cellulosic cell wall found only in plants, not in bacteria. right? So therefore, this is also absent. Then it comes to plasma membrane. Of course, it is present. Of course, it is there. And circular double-stranded DNA means they are talking about plasmid. So plasmid is there in all bacteria. So therefore, for this question, which is our answer? Which option is our answer? Yes. Option A and B is our answer because these two structures are absent. Next question. Oxygen is not produced during photosynthesis by nostoc, green sulfur bacteria, cycas and chara. So you heard about nostoc, which comes in Eubacteria. So being photosynthetic, that means autotrophic, here they could able to release oxygen here. Okay. So therefore, this is not our answer. This is not our answer. Green sulfur bacteria, of course, they can also prepare their food, but they are unoxygenic. Correct? I said they are unoxygenic. That means they release sulfur, not oxygen. So therefore, here itself we got the answer. This is our answer. Then what about the cycas and chara? About these two, we are going to study in plant kingdom. Cycas comes under gymnosperm. It is green color only. It will release oxygen. Chara, it comes under algae. It will also release oxygen. Therefore, these two are not the answer. Next question. Identify true statement about Archaebacteria. Let us check all the statement. They can live in harsh habitat, of course. Therefore, they are primitive in nature. Cell walls do not contain peptidoglycan, but they contain something else of different nature. So, this is also correct. This is also right. Cell membrane contain branch chain lipid. That's what I said. Lipid, protein, that will be branch form. This is also right. Some of them are capable of producing biogas. That means they are talking here about methanogens. Methanogens. They are found in ruminant. That means in the cattle. So all these statements are right. So therefore, it's not an answer. Only A and B is given. Here only A, B, C given. Here B and C given. So therefore, let's go with this, all of this. But every time, all of this, none of this is not the answer. We need to think and answer. Okay. Next question. Assertion and reason based. Many students get confused in solving assertion and reason based question. I will make a separate video for that because if I start explaining about all those things, about the tricks and all, how to identify, it will become 15 hours, 20 hours video. Okay, that's why here I will just explain the answer, but I will make a separate video about this assertion as the following all sort of questions. Assertion. Several ruminant animals contain methanogens within their gut. Correct. Several ruminant in the sense they are talking about cattle, buffalo, like those animals where the rumen part is present. So in that gut, methanogens are found. What is the reason? Methanogens help in the production of methane from the dung of ruminant. Correct. Why they have to present? Because the dung, the undigested waste, what they are going to release after the digestion, from them the methane gas is going to form. That means here, what the assertion is given, it's the right reason for this assertion. Right? So therefore, let us go with option A. Assertion is correct. Reason is correct. Reason is correct explanation for assertion. Whereas here, assertion correct, but reason is not a correct explanation for assertion given. Therefore, it's wrong. Here, assertion correct, reason incorrect given. Wrong. Here, assertion incorrect, reason correct given. Wrong. Next question. Rate the following statements and select the correct option. Sometimes the questions can become in the form of statement. I said no. Like such questions you may see in your exam. So for that, you have to read NCERT. Statement 1. Almost all bacteria possess lipoproteinaceous plasma membrane. Correct? Correct? Almost all. They are not telling every bacteria. Almost all they are telling. That means few bacteria are not having this lipoproteinaceous plasma membrane. Almost everybody. Either it is Archaebacteria, Eubacteria, Mycoplasma. Everybody is having. Statement 2 says, the plasma membrane of Archaebacteria as well as the Eubacteria have same type of lipid. No, this is wrong. Because here they have branched lipid. So therefore, which is the answer? Statement 1 correct and statement 2 is incorrect. Next question. Organisms living in salty areas. Direct question. You can answer. It's not methanogen. It is halophiles. Not even halophytes. thermoacidophiles. Which among the following are the smallest living cell? Smallest living cell means viruses are still smaller compared to bacteria, but viruses are not considered as living. I will tell you why at the end of the chapter. It acts like a boundary between living and non-living. So that's why if the question comes, which is the smallest living cell, just go to Mycoplasma. So I told answer itself, but still let us learn, understand the question. Now, without definite cell wall, they do not have cell wall, pathogenic to plants as well as the animals, and can survive without oxygen. Obviously, it is Mycoplasma. Correct? Bacillus, Pseudomonas, Nostoc, they do not come under this category. Right? Next question. Two kingdom system of classification did not distinguish between all of the given except. Did you understand the question? They are telling we made two kingdom classification. Correct? So in that, which one is considered and which of the following are not considered? That means only one is the answer here. Any one is the answer. Rest of the thing is not included in two kingdom classification. So what are all the things? So they made only Animalia and Plantae in two kingdom classification. So this has done only based on the cell wall presence and absence. It didn't consider the criteria like photosynthetic or non-photosynthetic. If it is like that, it would have made fungus as a separate kingdom. But here in two kingdom classification, we didn't found any fungus as a separate. Therefore, it was not included. And eukaryotes and prokaryotes not included because all your bacteria, fungus, plants, animals, everything has, fungus, protista, everything is included within Plantae only. Therefore, this was also not considered. And unicellular and multicellular, not at all considered because fungus, many fungus are multicellular, many organisms are multicellular, but bacteria is unicellular, but all together placed under kingdom Animalia. Therefore, this one also not considered. Only consideration was plants and animals. These are the plants, these were the animals. So this is the only thing they made. Therefore, this is the answer. Okay. Next question. In which kingdom chemosynthetic organism, chemosynthetic means they can prepare their food with the help of chemical energy, are seen out of 5 kingdoms proposed by Whittaker? If you just recall the chart or the table which is given in the classification part, easily you can notice that only in kingdom Monera it was shown. Rest of the thing was showing either autotrophic or heterotrophic. Within heterotrophic, saprophytic and parasitic, but no one was showing the chemosynthetic. So that's why it belongs to kingdom Monera, where the bacteria shows diverse mode of nutrition, not a protista, fungus and Plantae. Next question. Select the incorrect statement. So let us see what is the means. Which one is the incorrect one? In 3-domain system, all eukaryotes are placed in single domain. Okay, so you have not understood about 3-domain, no? It is given in just a single line in NCERT. Let us learn about that. We understood 2 kingdom classification. Correct? We understood 3 kingdom classification, even 4 kingdom classification and 5 kingdom classification. 5 kingdom classification is the main one done by Whittaker. But after that, so everybody accepted this one only. Yes, 5 kingdom classification is the best. Let us follow it. But after few scientists came, they said we will do 6 kingdom classification. So 6 kingdom in the sense, they said we make 3 domain classification. 3 domain classification. 3 domain are 6 kingdom classification. In that what they did, as usual, all the 5 kingdom they considered. Kingdom Monera, kingdom Protista, fungus, Plantae, Animalia, all 5 considered as usual. But within Monera, what they did, they made a separate group. They considered Archaebacteria as a separate kingdom and Eubacteria as a separate kingdom. Are you getting? But now, in the Whittaker classification, he said Monera itself is having these 2 categories, Archaebacteria and Eubacteria. But these people made these 2 as a separate kingdom only. And that's why they were considered as 6 kingdom classification. And this 6 kingdom itself also called 3-domain. Why 3-domain? So they made 3 domains like this. 1 domain, 2 domain, 3 domain. So these 3 domains were called by different names. So 1 domain, the Archaebacteria itself has kept under 1 domain. That means this is 1 domain and the Eubacteria also another domain. Domain means nothing but a group. And in the 3rd domain, they kept all eukaryotes. They considered it as eukaryota. So total 3 domain. This is 3 domain classification. Are you understood? Archaebacteria, here 1 domain. Eubacteria, another domain. In the 3rd domain, eukaryota, here they said all your fungus, all your Protista, fungus, Plantae and Animalia should come and included in this domain called eukaryota. So such classification called 3-domain classification. Understood? So it is given in our NCERT just in 1 line, but I made it clear. Hope I am very clear, crystal clear about this. Let's move on to the question part. In 3-domain system, all eukaryotes were placed in single domain. That's what I said. It is right, but we need to identify the incorrect. Plants have cellulosic cell wall, correct? So plants are having cellulose in their cell wall. Cell type is prokaryotic in kingdom Monera, correct? In Monera kingdom, all cells which are so present, that is bacteria, they are all prokaryotes only. This is also right. Then which one is incorrect? Mode of nutrition was not the criteria to classify organisms in 5-kingdom classification. This is not correct. This is incorrect. Because Whittaker considered mode of nutrition also as a criteria for classification. That means classifying the organism. Therefore, this is the answer because we need to identify the incorrect one. Okay. Next question. The composition of fungal cell wall. Fungal cell wall is what? Chitin. Chitin. Non-cellulose polysaccharide. It is nothing but peptidoglycan. Cellulose means which is found in plants. Lignin. It is also found in plant. But chitin is found in cell wall of fungus. In the table itself, we got the answer for this. Yes, now, next kingdom, kingdom Protista. Hope I am very clear about kingdom Monera. I have given more information actually which is not given in NCERT. But remember, they are important for your NEET exam. All the points what I have mentioned, what I have did, all are very important for NEET point of view. Okay. Now, when you just look at one more information I have to give here before going to this kingdom. Whenever you just look at the video, though it seems to be very lengthy, the time duration is very long, don't get panic just by looking at the time. But you can have an option. That means when you just press the description box, there the timestamp has been provided. Suppose if you want to watch only Protista, then you just click on the time so that it will just take you to that part only in the video. Then you can watch that video. You can watch that part within the video and you can understand the concept. If you want to watch full video, you have to watch from beginning to end. Suppose after watching, while reading NCERT or while solving any questions, if you get any doubt from kingdom Monera itself or kingdom Protista itself or within the kingdom Monera, if you have any doubt regarding particularly Eubacteria, then nothing to worry. No need to watch from the beginning or no need to search by scrolling it. Just put the just press the timestamp. You can get the direct topic there within the video. So that option you can have, you can watch that, right? Now, let's move on to kingdom Protista. Okay, now let's talk about kingdom Protista. So, whenever we talk about an organism, either they are multicellular or unicellular, right? Correct? So, multicellular means what? Organism which have many cells, multiple cells. Say for example, human being, animals, plants. So, all comes under the category of multicellular. Even there are certain animals, they have single cell also. But I am talking about more than one cell if they have. They are nothing but multicellular. Now, if you just made one more group apart from multicellular, we are going to get one, that means one more group as a unicellular. So here name itself is telling unicellular means they are having single cell. So these organisms, just by having a single cell, only one cell, they can perform all the activities. which could perform by these multicellular organisms by having many cells. Say for example, if you just keep counting the number of cells in human being, we can't count first of all because they are very, very microscopic. But still, even with the help of microscope, if you start counting, it's unbelievable that it will be more than billion, more than trillion. So such a large number of cells are present in our body. So all these cells together join and perform certain activities like a digestion, excretion, circulation, like that. But all these activity can be performed by a single cell also. Okay. So now we ask a question. Okay, we agree. You are great. You are single cell. You can perform all this activity. So is there any difference once again within you people? It says yes. If you just take an example of a kingdom Monera. Of course, this kingdom Monera also consists unicellular organism only, but they are telling we are prokaryotic cell. Prokaryotic cell. So there is one category. But here we are talking about Protista. In Protista, they do not consist any multicellular organism. They are only single cell just like Monera, but they are not prokaryotes. Listen it carefully. In kingdom Protista, all organisms are once again unicellular, but they are eukaryotes. Actually, there is no perfect boundary in this kingdom. Certain organisms, they show even multicellular characters or multicellular even, but majority or most of the organisms are nothing but single cell only. But they are all eukaryotes. Now, if we just allow them to fight together between prokaryotes and eukaryotes, of course, eukaryotes are going to get win. Why? Why? Just think for a while. Because in a prokaryotic cell, we say you do not have membrane-bound cell organelles. If there is a DNA, it lies as it is. If there is any certain other organelles, it lies as it is. Okay? But eukaryote says, we are more advanced than you people. Because, of course, we are a single cell. But if you are saying a DNA that is bounded by a membrane, so that I can call it as a nucleus. But here, such a membrane-bound organelle was not there. So that's why we said it as a nucleoid. Okay. So like that, if you just keep seeing many organelles, we could be able to find here, like Golgi apparatus, mitochondria, chloroplast, everything is present. Then obviously, who is stronger compared to prokaryotes? Eukaryotes only. Correct. So eukaryotes, from this we can come to the conclusion that eukaryotes, they are quite advanced compared to prokaryotes. So how eukaryotes are formed? Once again, we need to go back to evolutionary theory. Okay. So here, there is a theory called endosymbiotic theory. Hope you are making notes along with me. According to this theory, so you will learn in detail about all this theory, about all this process of evolution in your class 12th of evolution chapter. But I will just tell in brief about this. Okay. So earlier, before the formation of organisms on the earth, when single-celled organisms form, of course, they are all prokaryotes only. They are primitive organisms. So when you just looking all the organisms, they are all present in water itself. The first formed organism in water only. So all these organisms, they are prokaryotes. All are prokaryotes. But their size was varied. Some organisms were bigger. Okay. Some organisms were smaller. Like this. Some organisms were having medium size. So like that, their size varies. But remember, all were prokaryotes. Now, certain scientists, according to their according to their study, they said that the big prokaryotes, they engulf the small prokaryotes. For example, here is one prokaryote, correct? So this big prokaryote, it made to ingest, that means it engulfed this. But this smaller one, it resists to get died because it is saying that why you are killing me? I will do a lot of work for you. So this big prokaryote says, what work you can do for me? Then small prokaryote says, I can do a magic. That means I can capture the sunlight. I can prepare my own food. Okay. So because of that, every day you need not to go in search of the food, so that in search of the prey to have a food. Because I am here to prepare the food. Then it somehow got convinced. Because anyhow, it can prepare the food means why it has to search for a prey. Correct. Then it has entered inside its body. Now let me rub this. It got entered inside and it start preparing the food for this bigger one. So this is nothing but the chloroplast. This is nothing but the chloroplast. It means what? Chloroplast was also a prokaryotic cell once upon a time. Okay, so because of this endo means inside, symbiotic. Why symbiotic? Both are in actually compromising nature. That means both are actually helping each other. See, bigger one, it is getting food from this chloroplast because earlier it was a prokaryote. And how the smaller one can able to get a benefit from this bigger one? Because it could able to get a shelter, no? Suppose if it's not going to kill, if it's not going to eat, somebody was there to kill it. Okay, so somehow it got escaped from that scavenger or something else who is attacking on it. So like that, both are mutually getting benefit. So now we could say that which is having a membrane around it, it is a membrane bound organelle, therefore it could be a eukaryotic cell, right? So like that, this was a mitochondria. This was a mitochondria. Now in the same way, how chloroplast was engulfed, so like that a bigger prokaryote engulfed even the smaller one. There is a mitochondria so that it could able to break down all the glucose which are so prepared by this chloroplast, and finally it could able to produce ATP. So at last, why we are going to struggle a lot in our life? To get energy. So that energy itself is provided by this cell. There is a small prokaryotic cell. Earlier it was there as a prokaryotic. So that now it has entered inside its body. So now it is also one of the cell organelle being entered inside the another cell, because it is also having a membrane around it. Okay, so now both are mutually getting benefit. It is saying, I am giving shelter to you, okay, but you have to give energy to me. It agreed. Okay, I will give energy because you are giving shelter. So it is preparing food. It is providing energy. So now it is very happy. Not even to go in search of prey, not even to break the food to get the energy. So everything is done by its subordinate. Okay, so like that, the eukaryotic cell has been evolved. So this is actually endosymbiotic theory. Okay, so like that, all the Protista which are so evolved, they first formed on the water. So hope you are making notes along with me. So if you just make a general characters here, general characters of kingdom Protista, majority of the Protistans are found in water. We can say they are aquatic, but it is not a restricted area. They can even present as a parasite. That means they are even present in the soil. So like that, their habitat can be various. Then, when you ask a question about its nutrition, so it is having a diverse mode of nutrition, just like kingdom Monera. So there, we could able to see the nutrition like chemosynthetic, autotrophic, like that, no? But here, it is moved one step ahead. It means they are telling we can prepare our own food. That means you can keep us in the group of autotroph, prepare own food, right? And another group, of course, if they cannot prepare their own food, of course, they have to depend on others, right? So we call them as heterotrophs. So two category we got, but one more special category that we couldn't able to see such character in other organisms or other kingdom, but those things can be able to see in kingdom Protista. They are nothing but mixotroph. It seems to be quite different. Of course, it is different because these organisms, they can behave either as autotroph and even as heterotroph. That means, if I am an organism, if I will be considered as a mixotroph, means I can prepare my own food in the presence of sunlight. Okay, if the sunlight is there, then I can say, okay, I can prepare my food. Now, if there is no sunlight, then what I will do? Immediately, I will turn myself into heterotroph. That means, if the organism present in my surrounding, I immediately capture those organisms. That means I will consider them as a prey and I will eat them. That means what? I am not even a autotroph purely, not even a heterotroph purely. I am having a mixed character. So that's why it is known as what? Mixotroph. That means they behave So they behave both as autotroph in presence of sunlight and heterotroph in absence of sunlight. So that's why they are called what? Mixotroph. Okay. So now, if you just ask about the reproduction mode, we can say that they can produce or reproduce both sexually and asexually. So asexual reproduction either by producing the small spores, so that you will understand while talking about the different characters, I mean different category within Protista. And about sexual reproduction, it is very simple. Imagine there are two cells. This is one cell, this is another cell. We cannot say this is male or female, but the two separate cells were there. Of course, it will be having a nucleus. When it undergo meiosis, obviously it will be converted into single nothing but haploid cells. Okay. Now these cells, when fused together, let me make it in some other color. So it when fused together, obviously the nucleus also fused and it become a 2n called zygote. So like that, the sexual reproduction can occur by fusion and can form the zygote. So such type of reproduction can also be seen in Protista. So this is about general character. Now let us enter inside the actual category in the sense within the Protista, what are the different category we could able to see. From all these topic, from all these points, the chances of asking question will be high. Please make a note of all this. Right. Now, how you can able to classify the Protista? If you just making a group, we could able to get how many? 5 groups we could get. So what are those? Let me make one by one. First group, second group, third, fourth and fifth. You have to remember the names of this group. The first group is called Chrysophyta. Second group are called Dinoflagellate. Third one, Euglenoids. The fourth one, slime molds. Very easy, all are very easy. Simple. Within 5 minutes, every category will get finished. So only 3 to 4 points, if you remember, that is more than enough. So the information which is so given in our NCERT, that much only I am telling. I am not telling extra information. That means anything else apart from NCERT. Even if I tell, that will be, it is important for NEET. That's why I will be telling. So don't worry, all are easy. You can easily remember. Now the last category is called Protozoa. Now, out of these five, these two categories, that means Chrysophyte and Dinoflagellate, they are saying we are having photosynthetic pigment. Okay? That means we have a capacity to prepare our food. Then obviously, in which category you are going to place them? Yes, you are right. You just be in autotroph group because you are special, you can do magic. And these two, slime molds and protozoa, they are saying we cannot prepare our food because we are lazy fellow. We go and hunt others. That means they are depending on others, means they are heterotrophs. So within heterotrophs, we know that if they feed on dead organic matter, we call them as what? Saprophytes. If they depend on living organic matter, living organism, for example, present on living organism body, within their body or outside the body, they act as a parasite only. So even they are saprophyte or parasitic. So majority of saprophytes comes under this category, that is slime molds, and protozoa. Most of the protozoa are parasite. Some are free living also, but most of them are parasite. Okay. Now obviously the euglenoid, which is the one which left alone, where you are going to place? Yes, you are right. In mixotroph nutrition. Correct? It is not just based on the mode of nutrition we have classified. Even certain other characteristic features are also considered here. Okay? But the main feature here is the nutrition mode. We consider them to group as a different category. Now let's talk about the Chrysophyta. Okay? Yes. Chrysophyta, they are crying. Why they are crying? Can you guess why they might be crying? Obviously, we are exploiting, we are giving torture so that you have to prepare the food. You have to do photosynthesis. That means they are preparing lot of food. They are doing lot of photosynthetic activity in water. Because when you talk about Chrysophyta, They are crying like anything so that their tears made them to fill entire water body so that it become a big ocean. It is just the way to remember. Okay, it is just way to remember as a strategy. Okay, we can remember like that, because sometimes if the question comes whether it is aquatic or marine, sometimes what we'll do, we'll get confused. We'll write aquatic, of course, and many students, they consider it as a fresh water, but majority are marine water. So that's why I said they started crying like anything. That's why the entire water body, so it started feeling like anything, it became an ocean. Let's remember like that. So let me write few characters regarding the Chrysophyta in general. So what are those? The first thing is primarily aquatic. In that particularly, they are found in marine water. Marine water means ocean, sea, that's it. Okay. And how they look, ma'am? How they look? As I said, they are unicellular. How can you see through your naked eyes? If you want to see them, you need a help of a microscope. Correct. So they are very, very small. We can say they are microscopic. Microscopic. Will they move? Yes. They move in the water current. That means they are called phytoplanktons. Phytoplanktons. They say we do not have any legs. We do not have any locomotory organelles. Like that hair-like structure, like a cilia, flagella. You might have heard these structure. Okay. So such a structures we do not have. But as the water is moving along with the water current, we also make our journey. So that's why such microscopic organisms, they behave like a plants. They're not actually plants. They behave like a plants by preparing their own food. No, that's why we say that you are a phytoplanktons. Okay. Now, within this Chrysophyta, we have two categories. One is diatom and another one is desmites. NCERT is not giving much information about desmites, but only one line has given about this. That is nothing but they are considered as golden algae. So obviously the name itself is telling they might be looking in golden color. Why? This is because the pigment. So it is having a brown color, yellowish color pigment in their body. Suppose if I write few organisms, imagine this is one organism. So in its body, let me write in white color. If I start filling a brownish pigment-like structure, so how it looks? It looks like a golden color only, no? Golden only. So that's why as the organisms are having pigments of yellow and brown pigments, therefore they are referred as what? Golden algae. So it could be a NEET question, remember. So everything is of NEET only, because this video is mainly for competitive exam like NEET. But still, why I'm writing in certain line? Because 99% of chances will be there to frame the questions from these lines. That's why. Now, desmite part is over. That's it. Now we need to study a lot. Not a lot. We need to study few things about the diatoms. Just look at this image. Can you say which shape it is? Some are having spindle shape. See, spindle shape, you know, tapering end on both the side. Like this. They are called spindle shape. Some are having ladder-like structure. Correct? And some are having disc-like structure. Okay? And some are here, you can't see, they are having box-like structure. That means, if anybody asks a simple question, what is the shape of diatom? You show your pen. So this is how the diatom looks. You show your drum. So this is how the diatom looks. You take any vessel from your kitchen and show it. Oh, this is how diatom looks. You take any plate. You show them. This is how diatom looks. That means they do not have a proper shape. Instead of telling not a proper shape, we can say that it is present in all shapes. You can get diatoms in different shape. There is no particular shape to describe it. Okay. Now, we have given a credit to these diatoms because you are having a lot of doing a work of photosynthesis, no? So that's why we are giving a credit that don't cry, man. You are the chief producer. Chief producers of the ocean. Because of photosynthesis. And I said, it is present in different shape. If I just take any one particular shape and if I start observing its character, I could able to see, have you seen a soap box? So within a soap box, how a soap is present like that, this organism is present like a soap, but over that soap, how we are going to see two soap box, they are fit together so tightly. Right? So in the same way, the cell wall will be present around this cell. For example, this is the cell wall. One is smaller in size, another one is bigger in size, but they are fit very tightly with each other so that it will enclose the cell inside it. Okay, so now this cell wall is so strong that if you keep this cell below a table and you just call an elephant and just crush it or put your leg on the table will get destroyed, the table will get break, but not this diatom, because that much strong it is. Okay, so that's why we can say that the cell is indestructible. The indestructible cell wall. The cell is having indestructible cell wall. Why? What is such a speciality we could able to see within the cell? See, inside this cell, there is the cell wall which is filled by a special substance called silica. So these are actually cell wall. Within that, we could able to see silica. So silica is such a strong material that nobody can destruct it. Okay, so that's why they are very powerful and they are very strong. Now, obviously, if you are saying a cell, one or other day it has to get died. Now, when it get died, obviously it will lose the mass. It has to get settled in the bottom. Correct. So now in the ocean, imagine it's an ocean. Cells has got died, but cell wall is not destructed easily. It starts settling at the bottom like this. Settled at the bottom. But gradually, it starts getting broken up. It starts getting decay. Right? So, but that should be taken around billion years. Billion of years for the breakage of these cell wall. So finally, it will get convert like a chalk powder. So such a thing we call it as what? Diatomaceous earth. So this, if you just touch this diatomaceous earth powder or this powder of cell wall, it is very gritty in nature. Okay, so that's why because of that property of grittiness, it can be used in polishing and even it can be used in filtering a syrup and even a oil. So once again, this could be a NEET question, and here many students just ignore that by giving option how many years it takes for the formation of diatomaceous earth. Then the options can be given as 1,000 of years, hundreds of years, million years, billion years. So while reading, you just ignore that sentence. Okay, so it's a general sentence. Yeah, it takes billions of years. But when the question asked purposely on that particular word, then you start thinking that, oh, I read somewhere else. It was thousands of years or million years or billion years. So that's why you should be very specific even while reading also. Okay, so just make a note. It is a billion of years. It is given in your NCERT itself. Now this is a diatomaceous earth. It formed after a billion of years. So being gritty, where we are using, what are the uses? It is used in polishing, filtering, syrups and oils. That's it. So the diatoms, the Chrysophyta, one category of Protista over. Now let us enter into the second category. What is the second category? Dinoflagellate. Can you see? They are looking red in color. But when you just keep looking at these dinoflagellate, they are not just red in color. You could able to find green color dinoflagellate, blue color dinoflagellate, yellow color dinoflagellate, red color dinoflagellate. That means they are available in multiple color. Okay. So there is a first thing we have to note. Why? Why it is available in all color? Obviously because of pigments. Because of chlorophyll, how it looks green color. So like that, if there are certain specific pigment like a phycoerythrin that you are going to study in your next chapters. So like that, there are certain specific pigment that makes that organisms to look in that particular color. Okay. So let me write because of pigment. Because of pigments. So here, if you just look at these organisms, they're looking as if they are shining, as if they are having light inside. Correct? So such property we call it as what? Bioluminescence. This word is not given in NCERT. Bioluminescence, but if you know it, this is better. Bioluminescence in the sense, see, imagine this is a pen. In this pen, of course it is a pen only, not imagine. In this pen, there is a, let's assume there is a special protein called luciferin. Okay, so that protein is having a special property. What is that? It says, I can absorb sunlight during the daytime and I will emit as a light during the night. So that's why in the dark, it starts emitting like a light. So that property we call it as what? Bioluminescence. So that property will be exhibited by these organisms. Therefore, they start illuminating in a bright color as if there are so many red light we have spread on the ocean. Of course, they are also marine only. They are marine only. Okay. Now, there, while studying the atoms, we saw that the cell wall was there, but cell wall is filled by the silica. Now, if you just look at these organisms, here also the cell is there, but around this cell wall is present, but this cell wall is made up of cellulosic cell plate. That means like this. Imagine this is one plate, this is another plate, this is one more plate. So like that, in all angles, the cell plates are present. They are filled by cellulose. They act like a stiff plate. I could able to show only 2D diagrams. In 3D diagram, you can imagine, you can see here, 3D diagram. Entire structure has been surrounded by like these plate-like structures only inside the organism present like this. How? If I close my hand in a fist, the entire area has been surrounded by the cellulosic cell plate. Okay, that is nothing but the cell wall. Cell wall made of stiff cellulosic plate. Now, if you just look at the name itself, it is telling dinoflagellate. Di means two. Okay, so here di means two. Two flagella is present. Flagella means a locomotory structure. Look at here, something like this. One flagella is something like this, transversely. Another flagella is present like this, longitudinal. Okay, let me name it as, one is transverse flagella. Another one is longitudinal flagella. So because of this only, the name came as dinoflagellate. Now, as these flagella are right angle like this, right angles to each other, they show movement, but the movement is different. It's not show simply like this. It shows like a whirling movement. Just like if you look at your washing machine, how the clothes are rotating, how the clothes are spinning inside that, in the same way, it starts spinning like this, spinning so fast. So such movement we call it as a whirling movement. So that's why they are also called what? Whirling whip. This word is also not given in NCERT, but if the question comes, you should not get panic. Oh, we didn't read anywhere. See, here I am giving information. Whirling whip means nothing but dinoflagellate. Okay, so they show whirling movement. Okay, now here there is one special dinoflagellate. Everybody will get scared about this dinoflagellate. Its name is Gonyaulax. Why? They will get scary other organisms by this Gonyaulax because it multiply anything and it is red in color. Red in color, it is very important, and multiply very fast. Multiply rapidly. For example, if it is an ocean, here water is there in the ocean, and these are being red in color, they start multiplying like anything, and entire sea started looking like a red. It's not Red Sea. Red Sea is different. But here, the tides are appear red in color. Okay. So these Gonyaulax responsible for red tide formation. Very important information. This Gonyaulax responsible for red tide formation. And why other organisms are getting scary about this? Because along with multiplication, it start producing a chemical. So that chemical is very toxic. So it is a toxic chemical. To whom it is toxic? It is toxic to marine organism like fish, other aquatic organism like a prawn or any other organism. If they present, if they start and eat it, they will get die. Okay, that's it. Even dinoflagellate also over. So here, which are all the points you have to remember? One is it is obtained in all color. Second one is it is marine and it has two flagella. One is transverse flagella, another one is longitudinal flagella. And a stiff cellulosic plate as a cell wall. Whirling whip. Gonyaulax, it is a red dinoflagellate. Give an example for red dinoflagellate. Like such question appeared. It's a Gonyaulax. It produces red tide, helps in red tide formation because of its rapid multiplication, and even produce a toxic chemical because this toxic chemical is very harmful to fishes as well as the marine environment. That's it. Over. Now the third category. Already we have understood few things about that. That is euglenoid. So euglenoid, the first thing we should know that this is a mixotroph. Why mixotroph? You know it. You know about this. Because in sunlight, it acts as autotroph. That means it prepares the food. If you are saying you are preparing your own food, means obviously it should be having a chlorophyll pigment. Correct? Yes, it is having. It is having a chlorophyll pigment. So this chlorophyll pigment, it is a characteristic of higher animals. I mean higher plants. Right? So such a special chlorophyll pigment is responsible for them to prepare its own food. So that's why sometimes it behaves like an autotroph. Chlorophyll pigment like higher plant. Once again, NEET question. Higher plant. Now, the chlorophyll pigment is there, but there is no sunlight. They cannot store their food or they cannot behave like a plants. How we are going to see the nature? In higher plants, they do not behave like that. So that's why, how they are going to satisfy their hunger? They start looking around them. Oh, is there any prey? Yes, I will capture you. Okay, it will start engulfing it. Then obviously, in absence, it captures the prey and acts as heterotroph. Right? Now, if you just look at this organism, let me write here separately. Like this it looks. Let me show some green chlorophyll pigment here. And here it says, don't worry, even I too have flagella. One is small flagella, another one is long flagella. Correct? So what I said? One is short, another one is long flagella. In both the cases, in diatoms, that is in Chrysophyta, as well as in Dinoflagellate, we saw cell wall. Correct? And here we are asking, do you have any cell wall? It said, I am very flexible, ma'am. I have no need of having cell wall. I do not want cell wall. But I am having protein layer. It made them to become a flexible for me. Okay, so that's why it easily move with the help of this flagella and start showing its shape in a different way. That means all these things are formed only because of that protein layer, flexible protein layer. Let me make it in this color. So what is the name of this protein layer? It is called pellicle. Pellicle, and it is very flexible. Very flexible. And where they are available, I mean where they are present, of course in water only, but in a stagnant water. They found in stagnant water. So the best example here in this category is nothing but Euglena. Euglena. Can you see this image? These are all nothing but Euglena. Okay. So you can see only the long flagella. Another one short flagella is there, but it is very, very short. No, that's why it is invisible. Okay. So once again, the third category. So it's not visible. Okay. Third category, Euglenoids also over. So how fast we are moving now? Hope I'm not fast. Hope you are understanding even. So which are all the characters we have covered? We have covered Chrysophyta. We have covered Dinoflagellates. We even covered Euglenoids. Now let's move on to another category, that is slime molds. So now slime molds, they cannot prepare their food. Why? Because they do not have chlorophyll pigment. Right? So obviously they are heterotroph. And even after that, the protozoa which comes, that is also heterotroph. Let's talk about this slime mold. Yes. First, look at this image. Look at this image. Can you see this yellow color, which is actually present on this tree trunk? They are present on the dead leaves. See, even you can imagine, if you just put a gum, okay, glue or a gum, so that should be run slowly, not immediately it should get dry, but it has to run slowly. You can imagine. So like that, when a yellow color gum, if it starts spreading, if it starts moving slowly, how it moves, no? In the same way, this slime mold, when it present on organic matter in the sense the dead leaf, the tree trunk, anything which is not living. So that organic matter will be considered as a food by these organisms. They start engulfing. How they engulf? Not with the help of their hand, by just moving over them. If they start spreading over them, then it is nothing but they start digesting, they start engulfing. That thing we have to understand. So as they are spreading over it, no, so they start engulfing it. So that's the nature of the slime mold. Now let us talk about few things about this. Imagine there is a dead leaf. Let's consider this is a leaf, fallen leaf. Obviously it is an organic matter. Now a slime mold is present here. Let's assume like that. A slime mold is there. So this slime mold, which is having only single nucleus. This is having a single nucleus. So if you just look at the nucleus nature, which is 2n in number, 2n, diploid. It is a diploid in nature. Now it has to spread, it has to eat this leaf. So that's why it multiplies its nucleus. It multiplies its nucleus like this, not by showing meiosis, by showing mitosis. So now two nuclei are formed within a single cell. So you cannot consider it as a two cell. Usually in cell division what happen? If there is one cell, if there is one nucleus, during cell division I am talking. You will study in detail about the cell division and the characters comes under the cell division. It is having got two things. One is karyokinesis. I won't waste my time because all these things you study in detail in cell cycle and cell division chapter, but let us not move by having doubts in our mind. That's why I'm telling in brief about this thing. Karyokinesis means division of nucleus. Then the second phase is cytokinesis, division of cytoplasm. During cell division, the first step is always karyokinesis. It means first the cell enlarge, its multiplied cytoplasm, all the contents, everything, and the nucleus get divided like this. Okay. Now the karyokinesis process over, so that we got two nuclei. And after that, anyhow, the cell content has increased, right? Now what happened? Here the cell starts splitting like anything. I mean splits, so that finally we'll get two cells, and each nuclei will move in each cell. That means the cytokinesis means division of cytoplasm. Cytoplasm also got divided. Okay. So this is what the nature of cell division. It has to occur like this. But here in the slime mold case, only karyokinesis is taking place. The cell is enlarging. One karyokinesis occur, two nuclei form. One more karyokinesis occur, from each nuclei, one more nuclei form. Total 4 nuclei. Again karyokinesis occur, again 8 nuclei form. That means it is showing multinucleate condition. Let us go back. Here, as I show here, let me show here further. Next step I will cover here in this. Imagine the cell has become so bigger, it starts showing multiple nuclei. 1 become 2, 2 become 4, 4 become 8. So finally you have to think that this leaf is eaten by this structure of slime mold. Now it is having a multinucleate condition. So this multinucleate condition of slime mold is called plasmodium. Now it has engulfed all the leaf, all the organic matter. Now it starts feeling unfavorable condition. Unfavorable in the sense, imagine certain toxic is present in the environment, or the food is not available in the environment. Every area has become so dry, it is completely got barren, it's not getting any food. Now it is an unfavorable condition for it. Okay, so what it will do? It will start making a plant. So in this plasmodium itself, it start bearing a fruiting body. Okay, so now imagine this is a plasmodium. Multiple nuclei are there. How these multiple nuclei are formed? Only because of this mitosis. Mitosis means everybody is 2n. Everyone is 2n, 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 same to same. If it is having 10 nuclei, number of chromosomes 10 present in this nuclei, the same chromosome 10 is present here also. You will study about the mitosis, meiosis in the cell division chapter only. But you just remember, in mitosis, the same number of chromosome will be continuous. Whereas in meiosis, the chromosome number will be get reduced. If it is 10, when meiosis occur, it become what? 5. The chromosome number in that. How it takes place, ma'am? That you will study in cell cycle, cell division chapter. Okay. Now, it made a plan, I said. it start bearing the fruiting body on its surface. It said, oh, if we simply lie like this, we'll get destroyed. So let us bear some fruiting body. So like that, everywhere the fruiting body has formed. Within that fruiting body, meiosis occur. Now meiosis occur. Now all these become a haploid spores. Now this fruiting body, let me write it as a fruiting body. When the fruiting body bursts open, these spores that will be released, and the speciality of these spores is that they will be having a wall. They will be having cell wall like this. The cell wall are a true cell wall. Okay, so they have a character of the plants. That means the cellulosic cell wall they have. Or we can say the true wall. These are the spores. They start spreading in the air. Now, whenever they can survive for many years because they are very resistant, even in unfavorable condition also they can survive. Now, when they get a suitable environment, imagine there is one more leaf they got. So, finally, anyhow, we got our food. Again, let us start growing, let us start multiplying, let us start converting into plasmodium. So, like that, they start their journey again. So, this is what the life cycle of slime mold. Okay, hope I'm very clear. So, the true wall, again, it is a NEET question from this part. Fruiting body, it is formed during unfavorable condition. Okay, you can write it here as haploid spores because they are showing meiosis. So, this is about slime mold. Now, let's move on to the next category. This one over, slime mold, this part over. Protozoa. See, protozoa, it has believed that all the animals that you are going to found in kingdom Animalia. If you don't know about kingdom Animalia, don't worry. There is a separate chapter as called Animal Kingdom. There you are going to study. But hope you have studied few things in your lower classes about invertebrates, right? Like Porifera, Coelenthrata, Ascomycetes, Annelida. You have some idea. Basic idea you have. Okay, if you have that much idea, that is more than enough to study about this. So, all the animals, what you are seeing from invertebrata to vertebrata. So, it has believed that all these animals are formed because of this protozoa. That means, the protozoa is the ancestor of all these animals. How it is formed, ma'am? That you are going to study in evolution chapter. That is in class 12th. Okay. Now, this protozoa, it cannot prepare its own food. Correct? Then where it is present? Obviously, it has to depend on other organisms because it is a heterotroph. It says, I have mentioned there no slime mold as a saprophyte. Now, some are free living and some says we depend on others as a parasite. Of course, free living means they are not depending on food and shelter on other organisms. They are not giving torture on other organisms by sitting inside or outside their body. But they ingest other organisms by considering them as a prey. So that's why, of course, they are living freely, but they will eat other organisms. They cannot prepare their food. Okay. And where they are present, they can be found in aquatic environment. They can be present even in the soil. They can be present even in as a parasite, as I said, as a parasite. Okay. Now, within protozoa, we can make 4 categories. Okay, let me write here. Protozoans can be classified into 4 categories. One is amoeboid protozoa. Second one is flagellated protozoa. Third one is ciliated protozoa. Fourth category is sporozoa. So let's study about one by one, only 1 or 2 sentences given in NCERT. Let us cover that much only. Okay. If I give additional information, as I said earlier, again it will be important for NEET. Where it went. The first one is amoeboid protozoa. So here the name itself is telling, yes, we are talking about amoeba. You have seen amoeba, right? Let's taking an example of amoeba itself. Let's talk about amoeboid protozoa. So, if I ask you a simple question, do you consider the shape of amoeba is like this? You can say yes, ma'am. If I ask this question, Is this amoeba is having this shape? You can say yes, ma'am. That means amoeba is present in wide variety of shape. That means it is not having a proper shape, first of all. Why not proper shape? It can change its shape according to the need, right? So I will tell you why. So here inside the cytoplasm is present, correct? Now imagine these are what you are saying. They are actually not leg or not any particular shape. Okay. So let me talk about this. Imagine here is a foot. Here is a foot. Now it is not having a mouth. It is not having a leg. Not even having a hands. But it has to eat this food. How it is going to eat it? It start pushing the cytoplasm which is present inside. Okay. So imagine the cytoplasm is present inside. It is start pushing it. So how it is pushing it? Something like this. Something like this. It start spreading. It start spreading. Finally, it will change its shape like this. It completely captured. Now, look at how the shape become. Earlier, it was not having this shape, but by pushing the cytoplasm present inside, it start moving ahead to capture the food, to engulf the food. So that's why it has changed its shape. So that's why here, what the structure you are seeing, these are the structure, we can consider them as pseudopodia. Pseudopodia means false feet. Pseudo means false. False feet. Podia means feet. That means you are not having a proper leg. But you consider them as a leg. That's why you are spreading it. But you are changing your leg shape immediately wherever you want, whenever you want. Okay. So that's why its name is what? A pseudopodia. So this is one character. It is one locomotory structure. And where we are going to find this? Of course, in an aquatic environment. Aquatic environment. We can take an example. Many in this category. So in our NCERT, mainly they have given two examples. One is amoeba, which is found in aquatic environment. And another one is entamoeba. We will study about this entamoeba histolytica in detail in human health and disease chapter, which comes in class 12th. Okay. So entamoeba histolytica, it acts as a parasite. It lives on human body. It enters into the large intestine. Imagine if a person, if he get infected by entamoeba histolytica, which attack its large intestine and start eating its large intestine and start breaking the mucus line present in the intestine, start eating the red blood cells. So that's why in his fecal matter, the red blood cells, the mucus, everything it start excreting. So that is what such a danger work done by this entamoeba histolytica. That's why we can say that it is a parasite as it is living inside the body. Right? So these are the two examples. And in certain aquatic, especially in marine environment, these organisms, they have a silica shell. If they are present in marine, they have a marine or aquatic. Marine is also aquatic form only. They have a silica shells on their surface. Not all, few. Okay, so this is about the first category of protozoa, amoeboid one. And the second one is flagellated protozoa. So name itself is telling these protozoa, they will be having flagella. For example, look at this organism. If I draw this organism, it will be having flagella like this. Okay, so the flagella is also a locomotory structure. So with the help of this, they can move. So the best example here, the flagellated protozoa, we can take as trypanosoma. So this causes sleeping sickness. Okay. So don't worry, it is actually the African sleeping sickness, not in India. But if the person get infected by this trypanosoma, he has to treat properly, or else he may go into coma stage or he may get died finally. So there is a vector in the sense like a vehicle. Someone should act like a vehicle to carry the person from one place to other. No, I mean the vehicle, how it works. In the same way, there are certain organisms, they act like a vehicle to carry these organisms from one side to another side. Right? So here, so such vector is CC fly. CC fly. So this is one example about flagellated protozoa, which is given in your NCERT. This is second category. This is the first example. So like that, we can take one more example here. That cause STD. STD means sexually transmitted disease. That organism name is Trichomonas, which cause a disease called trichomoniasis. It is a sexually transmitted disease. It will transmit through sex. Okay. So this is about flagellated protozoa. What is there to learn? Just one example, and it is having a locomotory structure called flagella. And the third category is ciliated protozoa. Here name itself is telling the locomotory structure is what? Cilia. So the cilia are the very strong, eminent, prominent locomotory structure compared to these pseudopodia, flagella, everything. So here, let me take an example first itself, that is paramecium. Paramecium. So this paramecium somewhat looks like a slipper shape, something like this. Like this it will be, looks. Okay, so on its surface, it bears lot of hair-like structure, small, thousands of hair-like structure. So these small hairs are called nothing but cilia. And inside its body, it will be having one big nucleus, one small nucleus. So this bigger one is called macronuclei or macronucleus. The smaller one is called micronucleus. Micronucleus, okay? And here it will be having a contractile vacuole, which is responsible for balancing the water. Okay, so how much water should be there and how it has to make the body to get balanced. So like such work will be done by this. Nothing to worry about all these things. Contractile vacuole. And now this part is called gullet. Gullet, okay? Now let's assume here is a foot, here is a foot, here is a foot. Earlier the foot was here. Now with the help of cilia, it is just keep pushing. It keep pushes the food so that it has to come near the gullet and finally it will engulf inside its body. So now the food has entered inside because it has entered near the gullet. Now it will be get digested, right? So this is how it will manage its work. The main locomotory structure is cilia, example is paramecium. That's it. And the last category is the sporozoa. So here the sporozoa means these are spore-like structure. Spore-like structure. So the best example we can take here for this sporozoa as Plasmodium. Have you heard this term? Plasmodium is a protozoa which cause malaria. This is a malarian parasite. Once again, about this Plasmodium, you are going to study in detail in your class 12th, Human Health and Disease. So here the Plasmodium, which show its life cycle both in mosquito as well as in human body. So in mosquito body, it will complete half of its life cycle. And the same mosquito, when it bite the human, this is spore-like structure that will be transmitted into the human body. There it will undergo further multiplication, further development. So there it complete further life cycle. So like that it will take two organism help, two host help to complete the life cycle. But as it is causing in human body as a disease called malaria, that is done by a vector called vector. Vector is nothing but mosquito only. So this mosquito is a special mosquito. It's not an ordinary mosquito. It's a female Anopheles mosquito. That's it. That's it. So if you remember this much, that is more than enough for your class 11. But you are targeting for NEET. So you are going to study in detail about all these things in your class 12. The continuation. Okay. So now we have completed even the kingdom Protista. Now let's talk about few questions. But before that, these are the images given in our NCERT. Can you guess what are these images? This is about what? This is Dinoflagellate. This is Dinoflagellate, which is having two flagella. So one is transverse flagella, which is somewhere here, and another one is longitudinal flagella. This one. Let me take this color. Okay, this is transverse. This is longitudinal flagella. And this one, which is this organism. See, this is the short flagella and this is long flagella. Correct? So, which organism it is? Yes, your guess is right. This is Euglena. This is Euglena. And this one is Paramecium. And what is this? This is nothing but amoeba. This is amoeba. This comes under, these two category comes under protozoa. This is euglenoid. And here this is dinoflagellate. So if such question comes, if the question asks you, by just giving the image, you should be able to identify it. Okay. Now let's move on to the question part. One second. This is not amoeba. This is slime mold. Okay. So this is slime mold. See, here multiple nuclei are given. Cenocytic condition. That means multiple nucleate condition. Right? So slime mold, paramecium is nothing but protozoa. This is euglenoid. This is dinoflagellate. From everything, one example has given as an image. Right? Now let's move on to the applied as well as the concept-based question. Which one of the following is a characteristic feature of chrysophytes? They are parasitic form which cause disease in animal. First of all, chrysophytes are photosynthetic. They do not cause disease. Therefore, this is wrong. They have protein-rich layer called pellicle. So, pellicle is a characteristic of euglenoid, not Chrysophyta. Again, it is wrong. They have indestructible wall layer deposited with silica. This is correct. This is correct. They are commonly called dinoflagellate. Dinoflagellate, wrong. Chrysophyta is different. Dinoflagellate is different. Next question. Though they are photosynthetic in presence of sunlight, when deprived of sunlight, deprived in the sense when the absence of sunlight, they behave like heterotroph. Predating means they capture other smaller organisms. That means the smaller organism will be considered as prey. They are, to whom they are referring? They are talking about mixotroph. That is nothing but euglenoids. So here it is not a slime mold, protozoa, Chrysophyta, it is euglenoids. Okay. Next question. The given statements are describe a group of organisms. So the 4 statements are given. So to which group of Protista these points will be considered? Let us talk. Instead of cell wall, they have a protein-rich pellicle making body flexible. So just by reading the first sentence itself, we got the answer that yes, they are talking about euglena. Okay. But still, let us talk about let us read the rest of the things. They have 2 flagella. Of course, euglena have 2 flagella. One is short, another one is long. They show mixotrophic nutrition. Correct. They are connecting link between the plants and animals. Okay. So once again, this could be a statement-based question. This could be a statement-based question. Within the question, you can frame one other question. Why? Just by giving this line, whether it is right or not, they can ask, this is right. For Protista, it is right. Why? Particularly for euglena, it is right. Can you think? Because, see, euglena, they will be having chlorophyll pigment, correct? They will be having chlorophyll pigment. So, this chlorophyll pigment, this is the nature of plants. The chlorophyll pigment is the nature of plants. So, that's why few euglenoids are the organisms which comes under the euglenoids group. Euglenoids in the sense it is not just euglena. Euglenoid is a group. Within that group, many organisms can come. So, some can behave like a plants. Some can further in the evolution period converted into plant. And some can even act like an animal. Therefore, it is a connecting link we can say between the plants and animals. Okay. Right. Dinoflagellate, not an answer. Slime molds, desmides and atoms. No. The answer is euglenoids. Next question. And here, I didn't explain about the animals. No, yes. Why they can behave like an animal? Because they are going to capture the food. That is a property of animals. Therefore, they behave like both. Right? Next question. Select the wrong statement. The atoms are chief producers of the ocean. Correct? It is not our answer because we need to select the wrong one. The atoms are microscopic and float passively in water. Correct? It's not another answer because it's right one only. Walls of the atoms are easily destructible. This is wrong because walls of the atoms are indestructible due to silica. So this one itself our answer. But still, let us check the last one. Diatomaceous earth is formed by cell wall of the atoms. Correct. So everything is correct except the third one. Therefore, that is the answer. Dinoflagellates are called fire algae. Due to which character? They appear like fire due to pigments. They produce fire due to friction. They occur in burnt places. They show bioluminescence. What is that? They capture light. by sunlight they are going to capture by luciferin protein. They emit that. So when they emit, obviously they look like a light, bright light in the night. So that's why they might be looking like a fire. So that's why the fire algae. So this is the answer. Next question. The most efficient locomotion in protist is through. See, pseudopodia, flagella, cilia, both are all these are locomotory structure in Protista group. But when it comes to the efficiency, the cilia which show more efficient movement. So therefore, let us consider the cilia as a most efficient locomotion movement in Protista. Next question. Select the incorrect about the kingdom Protista. Being eukaryotes, the Protistan cell body contains a well-defined nucleus and other membrane-bound organelles. Correct. They reproduce sexually only. No. They can reproduce both sexually and asexually. So this is wrong. So here itself we got the answer. Still check the rest of the things. Boundaries of this kingdom are not well defined. That means I said no, most of them are unicellular. But some even comes under the multicellular characters. So therefore there is no particular boundary, because we say that, oh, you are autotroph, you are heterotroph, and some other will come, we are mixotroph. So there is no specific boundary. So everything is right. Members are primarily aquatic. Except second one, everything is correct. Next, which statement is wrong about chrysophytes? Microscopic right, photosynthetic right, datums of cell wall form two thin overlapping shells which fit together as in soapbox right. They are found in terrestrial habitats only. This is wrong. Particularly Chrysophyta, I said, how you can remember, they are crying. That's why the entire water body has got filled like an ocean. So therefore this is wrong. Here it is telling only present in terrestrial. Next, which of the following statement is incorrect? Again, Dinoflagellate, two flagella, correct. Euglenoid are freshwater organism, correct. Slime molds are saprophytic protist, correct. Spores of slime molds lack cell wall. This is wrong. This is the important word first of all. Slime mold, spores which are so present, they will be having true cell wall, cellulosic cell wall. But they are telling lack. Lack means they do not have cell wall. How come it is possible? Therefore, this is incorrect. It is our answer even. Oh, that's it. Now let's move on to the last kingdom fungi. Okay. So here, not diagrammatic explanation given in our NCERT. Just everything has given in the form of text format. So that's why if my belief is that if I make you understand every concept through diagram, it can easily, you can remember it easily, so that the pictures speak more than 1,000 words, right? That's why I will draw few diagrammatic schematic representation about the fungus, so that if you make a notes and at the exam time, if you just go through that as a short notes, if you make, easily you can remember everything, you can recall, right? So let us talk about this fungus first. Okay, kingdom fungi. So here in this kingdom, all organisms which also comes in this category are nothing but heterotrophic. Heterotrophic in the sense they cannot prepare their own food. Okay, so they have to depend on others, either in the form of saprophyte or in the form of parasite or whatever be the mode, but they cannot prepare their own food. They are not at all autotrophs. Then these organisms, they are even multicellular or unicellular. See, listen to me carefully. When we talk about the kingdom Monera, they are only unicellular, correct? But they were prokaryotes. When we talked about kingdom Protista, they are also unicellular, but these are eukaryotes. So we are studying all these kingdoms in an advanced level, that is in an evolutionary basis. So one thing you have to keep it in your mind that all the organisms, either it is a plant or animals, what you are seeing at present, all are evolved from a single cell. That means our ancestors are nothing but a single cell prokaryotes. From these prokaryotes only eukaryotic like organisms formed. From these eukaryotes, the next basis of evolution, that is nothing but the next fungus, plants, animals, all these things started evolving. Okay, so that's why after kingdom Protista or after kingdom Monera, all the organisms are showing this eukaryotic nature, right? So here few points I have mentioned regarding kingdom fungi. So it's a general character. Just now I said no, it's a eukaryotic, it is a heterotrophic. Majority are multicellular. Majority, most of them are multicellular, but except and yeast. So yeast is unicellular. See, whenever we talked about a yeast, one thing comes in our mind that that means that it will be used in the preparation of bread, cake, even the beverages. Correct. So yeast is actually a group. It's a group. It's not a single organism. So in this yeast group, there are so many organisms comes. That means so many different fungus comes. But one fungus name is Saccharomyces cerevisiae. Saccharomyces cerevisiae. So this will be used in the preparation of bread and cake. Therefore, it is called what? Baker's yeast. And even the same yeast will be used in the preparation of wine, beverages and all. So therefore, it is also called brewer's yeast. Brewer's yeast. Okay, so this is only unicellular. Rest of them are multicellular. Okay, you should remember this. And where they are going to form? In a diversified habitat, a wide variety of such a group we can able to see. They are present in different habitats. Say for example, especially they prefer moist or warm area, warm or moist area, damp places or rotten things. Okay, dead and decaying organic matter. So in all areas we could able to see these fungus. Even they act as a parasite. You might have seen ringworm and all. So the skin infection, that is a ringworm, no? It is a fungal infection only. So here the skin is nothing but the host. Okay, it's a food for those fungus. They started feeding on the skin. Therefore, it started showing the ring-like appearance. So in the same way here, between the toes, between the fingers, you might have seen some people will be suffering from fungal infection. So that is nothing but it is a food for those fungus. That means those fungus belong to the category of parasites. Suppose if they're depending on dead organic matter in the sense what? Imagine a leaf. If it is present on a tree, it is a living. If it is fall down, then it is a dead. So like that, a tree, when it was alive, it might be infected by a fungal-like organism, then such fungus are parasite. Suppose if the same tree got died, that is nothing but a fallen tree, then that is nothing but a organic matter. It's a dead organic matter. So human being, when he is alive, okay, when he got died, it's a organic matter, and even his fecal matter is organic matter. So on such thing, if those fungus feed, if they start growing on them, we call them as what? Saprophyte. So like that, we could able to see these fungus in a wide variety of habitats, right? Now, here whenever we just think about a fungus, we think that all these fungus are disease causing only. They are villain only. But no, there are certain beneficial fungus also there. Just now I mentioned about yeast, right? Can you see a small bubble-like structure here in this image? So these are nothing but yeast. In this diagram, you can see a budding is taking place. See, this is a yeast. A small outgrowth appeared here. Can you see here? Can you see? This small outgrowth is nothing but a bud-like structure. This process itself is called budding. Now this bud started growing like anything. See, it started growing and that will be get detached from the parent body. Again, it become a separate organism. So it become a two yeast. So this is a mode of reproduction. That is a budding. So this yeast is a beneficial organism. We can use, as I mentioned, in the beverages as well as in the bakery product like bread, cake and all. Can you see here? The dough we have prepared from this yeast. The dough has been raised. Why? Because yeast. Here the fermentation process is taking place. And now here the Puccinia, once again a villain, because it is causing disease in a plant. Okay. And therefore it's a parasite. It's a parasite. Can you see here? A small dotted structures. In a group it is present. And this is Penicillium. When you observe this Penicillium organism under microscope, you can able to see like this structure. Again, it is a beneficial because from this penicillium, we can prepare a chemical called antibiotic. This antibiotic name is penicillin. It has saved the life of millions of people during Second World War. Now also we are using this penicillin as an antibiotic to treat several diseases. So once again it's a beneficial. So every fungus is not harmful. Like that every fungus are not even a beneficial also. So both category of fungus we could able to see in this kingdom. Now when you observe these fungus under a microscope, what type of structure you could able to see? Let me take an example of a fungus which is growing on a bread. Okay, let's imagine this is a bread. On this bread, a thread-like structure is growing like this. It's growing. These are the thread-like structures. They are growing. So this group of thread, what you are seeing now? So this is nothing but mycelium. In this group, a single thread, suppose if I take, this is a single thread. This is called hypha. Singular is hypha, plural is hyphae. Hyphae is a plural. Okay, so such an interconnected hyphae, what you are seeing, this is nothing but a mycelium. So sometimes you are able to see like this also. These are interconnected. When you just look at the bread, if you just keep it outside without keeping it inside the refrigerator, then obviously after 3 or 4 days, these whitish hair-like structures will be growing on the bread. That is nothing but the mycelium. Right. So a single thread in the mycelium is called hyphae. So this is another hyphae. This is one hyphae. This is one hyphae. Right. So in this hyphae, the cytoplasm is filling. Okay. So let's imagine this is the cytoplasm. It has been filled inside this hyphae. Now, it's a tubular structure. Can you see this is a tubular structure? Now inside this tubular structure, within the cytoplasm, many nuclei are present. Many nuclei. But they are looking like a single cell. So what I said, if there is no cell division, only nuclei start multiplying, then that condition is called what? It is a cenocytic. It is a cenocytic condition. Cenocytic means, once again I will write, multinucleate condition. Multinucleated condition. But here there is no line between the nucleus. So such hypha are called aseptate. So such hypha are aseptate. Septa means crossword. But in some cases, in some fungus, what happens? Imagine a substratum or any food particle, whatever. Okay, just an example I am taking. Here the fungus is growing. These are the fungal hyphae. So in this fungal hyphae, lines are formed in between. Okay, so lines are forming. So in these lines, here the nucleus is present. Then obviously they are considered as a separate thing. Right? So now, such a structure are called septate hypha. Septate hypha. Septa means the crossword. Okay, and we all know that when you just take any cell, of course the nucleus is present, and outside what you are seeing as a cell wall, this cell wall is made up of what? Cell wall is made up of a substance called chitin, and even it also consists of polysaccharide. So this is how we can able to see the structure in a microscope, under a microscope. But of course, there are certain additional structure are also formed during the reproduction time. But it is a general structure, a basic structure. Hope I am very clear about this. Now when you just entered into the nutrition mode, once again, as I have mentioned, it's a heterotrophic, cannot prepare its own food, can opt. Why this diagram came here? Okay. So, can show these 3 types of nutrition mode. One is saprophytes, as I have mentioned. They can absorb the soluble organic matter from from the dead substrate. It means what? They absorb the soluble organic matter in the sense, see, ours is a holozoic nutrition. Listen to me carefully. Ours is holozoic nutrition. In the beginning itself, I gave the example. If any bread piece is there, first we are going to ingest it as it is. Then we are going to chew it. Then it will get digested inside our body. After digestion by enzymes are required very much for the digestion process. After the digestion process, then it will be get absorbed inside the blood. Correct? And Anyhow, after absorption, the body which do not consider for absorption process, that will be ejected. That is egestion. So that is a holozoic nutrition. Of course, we are also cannot prepare our own food. But here, these organisms, when they comes under the category of saprophytes, first they are going to release their enzyme outside. They are going to spit their enzyme outside the organic matter. For example, let me consider the same bread. Same bread. Let's assume, let me rub this part. First, what they are going to do is, first they release certain enzymes here. Okay, so all these enzyme release outside. So these enzyme digest this bread outside only. So that's why when you just touch the bread or even any vegetable, it will become watery. Why? It is started getting digested outside. Now after digestion process, then it says, oh, my food is tasty. Now I can absorb it. Then it starts absorbing. So such a type of nutrition is nothing but saprophytic. Okay, I will tell you about this diagram later. So this is one category of nutrition. And the second category is parasite. Parasite means it is depending on living plants and animals. All disease-causing organisms, all disease-causing fungus, or even bacteria are nothing but parasite. As I gave the example of a ringworm, which is growing on a skin of an animal or a human being, that is nothing but a parasite. Okay, and even there are certain fungus which cause disease in plant, it is a parasite. Okay, so they are depending on living things. But symbiont means what? Here, if the two organisms, this is organism 1, this is organism 2, both are mutually getting benefit. If you benefit me, then of course I will also benefit you. Okay, that means both are depending on each other for their needs. So such a relationship we call it as symbiosis, and such organisms under such relationship is known as symbiont. Okay, so here the best example for these symbionts we can take as lichen, and the second example is Mycorrhiza. Mycorrhiza. Though these terms seems to be difficult, but trust me, these are very easy. Very easy. Within 2 minutes, you can understand everything. Kingdom fungus is not at all difficult. Okay, let's learn about lichen. Okay, so here in lichen is one group. Here in this group, fungus of course is there. And along with fungus, algae is also there. You are going to study about this once again in your class 12th also. If you have this concept in your mind, if you have a clear idea, then it will become easy there. Okay. Now, what algae is going to do here? Imagine this is algae. Algae says, I have photosynthetic pigment. I can prepare my own food. But the problem is, for the food preparation, I need ingredients. Correct? If your mom is going to prepare certain dish in the kitchen, obviously she need ingredients, right? Then your dad is going outside, then he'll bring all the ingredients which are so required. Then she could able to prepare the food. But when she prepare the food, is she going to eat alone? No, she is going to serve everybody at home. Correct? In the same way here also, the responsibility of cooking is given to this algae. But for cooking, the ingredients which are so required, no? So that responsibility has been handed over to this fungus, right? So let me make this algae in green color because it is having a chlorophyll pigment. But majority is covered by this fungus only. Therefore, when you just look at the algae, when you just look at the lichen, it look almost whitish in color. Now this fungal hypha, they are spreading like this, around this. So what it is doing? Fungus says, don't worry my friend, I am going to absorb many things. You want water? No, I will absorb water. You want minerals? I will absorb minerals. Minerals like phosphate ions. So you want other materials? So I will absorb everything. Okay. So finally, after absorbing, I will give it to you. Now you can do your magic. So what magic? You can use sunlight. By using sunlight, by using all ingredients like water as well as mineral, even the gas, that is the carbon dioxide, by using those things, you can prepare the glucose. Glucose is nothing but the food. Now, after having glucose, you can give it to me also. Now, the glucose will be given to these fungal hypha. So, both are getting benefit, no? So, once again, it's an example for symbiosis. So here also the fungus is helping and algae is also helping. This is one example. If you just look at this image, you can understand how this, where it went. One second. Okay, the image is appearing on your screen. You can see this is the lichen, which is growing on the tree trunk, right? So now let's move on to another example of symbiont, that is Mycorrhiza. So myco means fungus. Myco means fungus. Rhiza means root. So which plant root? So it's a higher plant roots. Higher plant roots. You all heard about Pinus, tree Pinus, correct? So this is how it looks. Now Pinus is telling, I am not having that much enlarged root. I am having very limited root. But by having such a limited root, I cannot spread my root because I have to absorb the nutrients, I have to absorb the minerals, but that work I cannot done alone. So that's why I need the help of whom? This fungus. Then the fungus will come for itself. It start attaching to the root of this plant. So finally, it will make a thread-like structure. There is a hypha started getting attached to these roots. It started spreading like anything. It started covered to a large area inside the soil. Now easily you can understand what work it can do. It start absorbing the minerals which is present in the soil, like phosphate ions. It start absorbing the water. It start absorbing many nutrients. And finally, that will be given to what? Nutrients, and that will be given to these roots. That will be given to the roots. Now the plants once again start preparing the food. So this is the leaf. So the food has been prepared. So that food that will be transmitted even to this fungus also. So again it is a symbiotic relationship. Here in this image you can understand how it looks. So here you can see the tree. And below, if you observe here, I will show it in green color only. So these are the roots of these plant, Pinus plant. But on either side of these roots, you can able to see this, the fungal hypha, which has been spread to a larger area, which has covered larger area. So like this, they could able to get the nutrition in different mode. Okay, now let's enter into the next part about the reproduction. See, I will give a minimum idea regarding reproduction here itself in this slide, because you will learn about the reproduction as we move into the different classes of fungi. Here only in this class only I will tell everything, but not in this slide, because when you start understanding the different classes of fungus and their reproduction which happens in that particular class, then easily you can understand about the sexual as well as the asexual reproduction. But let me tell you about vegetative reproduction first. Okay, so vegetative reproduction means what? It is not taking help of other organisms in the sense other sex. Okay, here first of all there is no male and female fungus like. So all these are haploid only. I will tell you the meaning of haploid, diploid, everything in plant kingdom in detail. But how much you require to understand this concept, that much I will explain here in this chapter. But in detail, you have to wait till the plant kingdom chapter. Okay. So now, in vegetative reproduction, what happens? The three mode of reproduction can take place. One is fragmentation. Imagine this is a fungal hyphae. Correct. So all of a sudden, if it is get fragmented in the middle, that means fragment in the sense, if it is just broken down in the middle, then what happens? They become separate, right? The two mycelium got separated. Now, this grow as a separate mycelium. It started growing the hypha, all its hypha, so that it become a new fungus. And this one also started growing like this and it become a new fungus. So once again, you could able to see the reproduction in this way also. And the second category is fission. Already we have understood in bacterial reproduction. How a single cell first multiplied its nucleus and finally gets cut from the middle and become a two cells. So this is called fission only. And about budding, just now I have shown the yeast diagram. The yeast is a single cell. Correct. So this yeast, it show a small outgrowth on its surface like a bud, and It multiplied its nucleus and moved into the bud part also, and finally this bud started growing. Sometimes the chain of bud can also appear on the parental yeast body only. So finally, as they started growing, they get detached from the parent body and started getting separate as a new cell. So like that, they started showing the reproduction. They produce new progeny. All their genetic makeup will be same to same. Where? In the vegetative reproduction. Whereas in the asexual reproduction, here no such separate structure will be formed, but instead certain spores formation takes place. Certain spores are going to form. Imagine this is a hypha, correct? On this hypha, a special structure is going to form, and inside the structure, a small spores are going to be present. So when that structure bursts out, these spores are released into the air. Whenever they get a suitable substratum, say for example, one bread piece is there, when they just land on this bread piece, then they start growing into a new fungus. So what are the different types of spores? How it will look, how it will be formed, that we are going to study in the next slide. Okay, so like that, in sexual reproduction, it needs two different fungus. This fungus genetic makeup is different and this fungus genetic makeup is different. So they have to come near for the sexual reproduction. But this process you will understand slowly as we learn the different classes of fungus. Okay. So here mainly 3 steps involved. One is plasmogamy, another one is karyogamy. The third one is meiosis in zygote. I know you are getting confused about the sexual reproduction, the 3 steps, but trust me, at the end of this kingdom fungi, you will understand all the 3 steps. First, let us move on to the classes. What are the various classes of fungi? Okay. So basically, we have 4 groups or 4 classes in the fungus kingdom. What are those? The first one is class Phycomycetes. So this is the primitive fungus. And after that came Ascomycetes. Okay. And it is also called sac fungus. I will tell you why it is called sac fungus. And the third one is Basidiomycetes. It is also called bracket fungus, puffballs, many word. Okay. And the last one is Deuteromycetes. So this is called imperfect fungus. So we'll understand about all these fungus in detail one by one. Okay. So now let's move on to this Phycomycetes. Phycomycetes is primitive. So it said, I'm very, very primitive. I'm very old man. I do not read any fancy septa like hypha. I'm just very cool. I'm just having a single hypha. Single in the sense, I'm just having a hypha without any septa. Septa means, as I have mentioned, no, if this hypha, if it is having a wall-like structure, cross wall, then we call such septum as, such hypha as septate. But here, no such structures we could able to see. Only nucleus is present inside, completely filled by cytoplasm inside this tube. So such hypha are aseptate and cenocytic. So this is the only fungus. This is the only group of fungus which are showing aseptate and cenocytic. But when you comes to the rest of the fungus, that is Ascomycetes, Basidiomycetes and Deuteromycetes, in every case, we are going to see the fungal hypha which are having these lines. There is a cross walls. Okay, inside which present the nucleus. Then what do you call such hypha? Just now I have mentioned. So such hypha are nothing but all these are septate and branched. Okay, so this is the one basic difference you should know about the within the classes of fungus. Okay, now let's enter into the Phycomycetes. While explaining Phycomycetes itself, we will understand about sexual and asexual reproduction, what I have put pending in order to explain in a previous slide. Now, look at here. Even if you can't see this properly, you can open your textbook and you can read along with me. See what Phycomycetes, information given here in NCERT. The members of Phycomycetes are found in aquatic habitat. Aquatic means water. Okay, so where they are going to form the moist environment, there you can see such fungus, one thing. And on decaying wood, that means obviously it is a saprophyte. In moist and damp places, that means in a wet area, as obligate parasite on plants, they can even found as a obligate parasite, means if I won't get this plant, then I won't grow. So that is what the strike made by this fungus. So such condition we call it as obligate. Facultative means if I found, okay. If I do not found, then also okay. So as we have seen in the case of bacteria, correct. Now, the mycelium is aseptate. It's a NEET question. And cenocytic, multinucleated condition. Now, rest of the thing is given about the asexual reproduction as well as the sexual reproduction. Now, let's study about asexual reproduction. This is one example. The image which is actually given in your NCERT, the same image I have put. So, this fungus name is mucor. So, if such image given in your exam, then if they ask to identify this, you should not get panicked. You should know this is a mucor. Okay. Now, let's talk about the asexual reproduction of Phycomycetes. Very simple. See, let me draw a bread piece again. So, this fungus said, I am okay with the bread. I am okay with the bread so that I can grow on this bread. So that I can produce my mycelium, that is hypha. So, it is cenocytic. Many nuclei are there. No septa, that is aseptate. Now it start thinking, why shouldn't I increase my progeny? Why can't I reproduce? Then it got an idea. It start producing a globular structure on its head. A special body will be produced here only. So on this special structure, it start producing a globular head-like structure. Okay. So this structure is called sporangium. Why sporangium? Because inside this structure only the spores are going to form. And one thing you have to remember, this is very, very important thing. You need to learn here itself that all fungus which can be belong to the group of Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes, you have to learn this class's name. Okay, it's very, very important. How ma'am, how can we remember? If you keep reading, you can remember. If you keep reading, definitely you can able to remember. Okay, in the beginning, you may feel difficult. Ma'am, I'm getting confused whether it is a Deuteromycetes or Basidiomycetes. Definitely for everybody, even for me also it happens in the beginning. But when you start reading, when you start doing practice, definitely it will come. Okay. Yes. Let's come back to our previous point. What I was telling. You take any fungus of any class. All fungal body are haploid. Okay. All fungus are haploid. Haploid in the sense they will be having the nucleus N. Diploid means 2N. 2N. Haploid means N. Ma'am, then what is the difference between haploid and diploid? See, if you take a nucleus, inside that nucleus only one set of chromosome is there. Imagine, I will tell you in detail in next chapter as I said, but okay, now no problem. If it is a nucleus, here it is having only single set of chromosome, then that condition is called N, haploid. The number can be various. I will tell you, it depends on the organism. Suppose, along with this, if some organism, they will be having one more copy of the same. That means 2. Each chromosome will be having 2 copies, right? Then we call it as 2N. So 2N condition will be present in every cell in all animals. So 2N, that is a diploid condition, will be present in all plants except few primitive plants. Say for example, if I take algae, primitive algae, it will be having its body cells will be having haploid condition. Okay. So like that, if I take certain bryophytes, so their body cells are also having this haploid condition. Haploid means only a single set of nucleus, single set of chromosome within the nucleus. Okay. Ma'am, I'm not getting anything. I'm getting lot of confusion. Okay, no problem. Just forget it. Don't worry about this, because as I have told, no, we'll learn in detail, but you just learn this much. All fungals body are haploid. Whenever the condition haploid is present, here mitosis never happen. Meiosis never happen. Meiosis takes place only in 2n condition. Okay, so meiosis means when a cell division, it is a kind of cell division. When meiosis happen, then 2n converted into n. Right? When mitosis happen here, the same to same chromosome will be continued. Wherever the n is there, always mitosis is going to happen in that condition. Meiosis never happen. So this is a basic idea. If you know this, then you can understand about sexual reproduction of this fungus. Okay, little bit. You can avoid the confusion if you know these things. Now let's come back here. Now here all these nuclei are in which condition? N only, because they are all haploid. All these nuclei are haploid. So if there is haploid, which thing should happen? Only mitosis should happen, correct? Now mitosis is taking place. All these nuclei are multiplying, so that here also in this structure called sporangium, the nucleus is multiplying, producing lot of nuclei. Now these nuclei, they start storing certain cytoplasm also along with them. So they become a spore-like structure. So spore means nucleus with some amount of cytoplasm, nucleus with certain amount of cytoplasm. So like that, we started getting the spores inside. So as the spores are producing inside the sporangium, so those spores are called endogenous spores. Or we can say the spores are producing endogenously. Endogenous means inside. Now when they get filled with the spores, they will get burst out. So let's imagine this sporangium has got burst up. So when they burst out, these spores start releasing out in the air. They started releasing out. So within the sporangium, we could able to see two different types of spores. One category of spores, imagine these are the category of spores. They are not having any motility, that means motile structure. They are simply just moving along with the air current. So we call them as amotile or non-motile aplanospores. Important again, aplanospores. Certain spores, they will be having flagellar motile structure. So they are called, imagine, they are called zoospores or motile spores. Motile or zoospores. But this is compulsory, you have to remember that zoospores and aplanospores are asexual spores. When we just look at this structure of reproduction, see, in the reproduction, I said asexual reproduction that has happened, performed through spores only. So, which spores here? In the case of Phycomycetes, aplanospores as well as the zoospores. Correct. So, like that, as we move into different classes of fungus, definitely we are going to see different spores names also. Now, they will be finding out for a suitable substratum. If they got found any other bread piece or any other substratum, then they fall down here. So, again they start growing. And again they become a new mycelium. So, this is how the reproduction takes place through asexual method. Where? In the case of Phycomycetes. Suppose if this Phycomycetes plan to go for sexual reproduction, then what it will do? Listen, let us learn. This is also very easy. Here, no need of formation of gamete. Why? It is not forming gamete because already their body is in haploid condition. If the gamete has to form, then it has to undergo meiosis only. But what I said, in haploid condition, meiosis never happens. So therefore, they do not expect the gamete has to form. Okay? But now, they are very smart. Still without forming the gamete, as their body itself is having lot of haploid cells or haploid nuclei, by using such nucleus only, they will carry out sexual reproduction. How it happen? Let's understand. Imagine this is one fungus of one category within the Phycomycetes fungus. So it will be having lot of nuclei inside. Correct. Now it is planning to go for sexual reproduction. So like that, there is one more fungus which is coming here. So this is another fungus, but belong to the same class, Phycomycetes. Now both are ready to go for sexual reproduction. So during this process, what happened? They start producing a small outgrowth like this. A small outgrowth will be formed. Okay, so this outgrowth, they start enlarging. During the sexual reproduction, they come close together with these enlarged structure. Okay, now if we just look at the same structure, somehow it looks like this. So now this structure also moving out. This is also moving out. Now they start pushing their nucleus inside this structure. Imagine their nuclei comes here, and its nuclei also came here. Now both started fusing. A small wall which is so present between them start getting melt. They start getting melt and they start forming a structure within that. So as a result, if you just look at the same thing here, it start forming a covering around it and a new structure has been formed inside. So this structure is called zygosporangium. So what is the name of this structure? Zygosporangium. Now when you just look at the zygosporangium, something like this it has formed, right? Here both the fungus cytoplasm has been fused by melting their wall. So as a result, inside I could able to see only the fused cytoplasm. So here cytoplasm has got fused, no? So this is the first step here in sexual reproduction. Okay, but here the nucleus will be as it is. So this first step is called plasmogamy. Plasmogamy, that means fusion of cytoplasm. Now the nuclei is as it is. These nuclei and this fungal nuclei is as it is. Correct? This is the zygosporangium, the enlarged structure I have written. Now the zygosporangium, it is just waiting for the fusion of these nuclei also. See, in some cases, if you take the example of Phycomycetes, that is here only, immediately soon after the fusion of cytoplasm, the nucleus also started fused. Okay. Whereas, if you take the classes like Ascomycetes or Basidiomycetes, first the cytoplasm fused. Okay, that is always do occur. But for the fusion of nucleus, it takes several years, 10 years, 20 years, hundreds of years. It depends on the kind of the fungus where the sexual reproduction is happening, right? Now, the next step, they are taking place, the fusion of nucleus also occur. Imagine, this is the zygosporangium and here the nucleus of both the fungus have fused together. Now they become what? So these are N. So these are N. Here also N, haploid. Now they are in NN condition only. When both fuse together, it become what? It become a structure called 2N. We can also consider it as a zygote. Okay. Now here within this zygosporangium, an outgrowth formed. So now in this outgrowth, it started showing the meiosis. So here the fusion of cytoplasm occur. This is in the first step. And in the second step, the nucleus fused together. It is called karyogamy. Fusion of nucleus. And after fusion of nucleus, the zygote which are so formed. So this zygote undergo the third step called meiosis. Now, whenever the meiosis occurred, from one cell of 2N, it start forming the 4 cells. But all 4 cells are haploid only, N only. So that is the beauty of meiosis. Now, here inside the cells are forming, all are N only. So these are nothing but the spores. They are in the form of a spores. So all these spores are N. Now once again, this structure is called zygosporangium. This is zygosporangium only. These spores are called zygospores. So remember, zygospores are sexual spores. Now when this zygosporangium burst out, again they fall on the suitable substratum. Again they start forming a new structure like this. There is a haploid plant body. So this is what the life cycle of sexual reproduction of Phycomycetes. Hope 3 steps are very clear. Plasmogamy, karyogamy and meiosis. So that is what I said, which happens in every classes of fungus. So now let me take those steps where I have stopped. See, reproduction part I said, asexual reproduction, where in the case of Phycomycetes is aplanospores and zoospores. When it comes to sexual reproduction, particularly about Phycomycetes, of course it shows these 3 steps. Plasmogamy, that is a fusion of cytoplasm. Correct? Karyogamy, fusion of nucleus. And obviously the meiosis in zygote. Final step. So that results in the formation of haploid fungal body. So the same information is given in your NCERT before the Phycomycetes class. But now you can understand better in the classes. That means when you start studying the different classes of fungus, easily you can understand the steps of the sexual reproduction. Okay, so that's why I did explain that. Now let's move on to the next class. But before going into the next class, let's read the same paragraph where we have stopped. So we have understood where they are growing and how they are showing their body structure. Now let's continue from these asexual reproduction. Asexual reproduction takes place by zoospores. These are motile. Again, it's important. Or by aplanospores are non-motile. Okay. These spores are endogenously produced. Endogenously means within the sporangium. In the sporangium. A zygospore means what? It is a sexual spore. Is formed by the fusion of 2 gamete. Here I said no need of forming gamete. But here NCERT is telling by fusion of 2 gamete. See here, their body cells itself considered as gamete. So therefore, again special new gametes are not going to form, just like how it happened in the case of higher animals and plants. Right? But here the entire plant nuclei itself are haploid, therefore they will be considered them as a gametes only. They started fusing, and these gametes are similar in the morphology, that is isogamous, or dissimilar, that is anisogamous or oogamous. So anisogamous, oogamous means their shape. Sometimes if it is having very smaller cell and another one is larger cell, it is anisogamous. And you will understand about these anisogamous, oogamous in plant kingdom chapter very well. Some common example. So this much information is given. But when you understand the process through diagram, easily you can understand how beautifully the process is happening. That's why I said in the beginning, I'll prefer making you understand the concept through diagram, because easily it can be printed in your mind. So the main thing, once again, some common examples are mucor. So here it is given in NCERT. Rhizopus is called bread mold, and albugo, the parasitic fungus and mustard. Look at the image here. This is bread mold. Can you see the bread piece? Here the greenish color, can you see? So the enlarged part here they have shown. So these are sporangiophores. Sporangiospores means a special structure I said on which the spherical bulb-like structure is formed. No, this bulb-like structure is called sporangium. And inside the spores are going to form. And the special structure which is bearing this sporangium is called sporangiophore. Phore means bearing, and rest of the things, stolon, rhizoid, these are nothing but their hypha, how it is growing. So this is one example. It is commonly called bread mold because they prefer to grow on bread, that's why. Okay, and another one is albugo, which usually prefer to grow on mustard plant. So as I said, no, all disease causing fungus are nothing but comes under the category of parasites. So this is a parasitic fungus. This is a mustard plant. Here the whitish structure what you are seeing, this is nothing but albugo only. Okay. So very simple. What are all the names we need to remember here? For Phycomycetes, main examples, Ram. You just remember a student name called Ram. So Ram, full form here, Rhizopus, albugo and mucor. That's it, over. And here spores. If it is asexual, what is the asexual spores name? Tell me. One is zoospores if it is motile, or aplanospores if it is non-motile. Even though you cannot remember the entire life cycle, how it is happening, if you just remember the spores name, then also you can survive in your exam. Then sexual spores. Sexual spores are not zoospores, zygospores. Correct? That's it. And the spores are producing endogenously. That means within the sporangium. That's it. Now let's move on to the next class of fungus called Ascomycetes. Okay. So Asco refers to sac. I will tell you. Okay. Ascomycetes. So once again, hope you have textbook with you. Just open this page. That is Ascomycetes heading and just read. It is commonly known as sac fungus. Sac fungus means, sac means what? Sac means nothing but a bag. Bag-like structure. So this fungus might be looking or it might be having a bag-like structure. So that's why it has got the title called sac fungus. Okay, so once again it is very important. The Ascomycetes, in fact I will tell you here only, ascus. Ascus means bag-like or sac-like. Hence it is called Ascomycetes or sac-like fungus. Now, Ascomycetes are mostly multicellular. Example Penicillium, rarely unicellular. Example yeast, that is Saccharomyces. So Saccharomyces cerevisiae, what I said in the beginning of this kingdom, it will be used in the bakery product making as well as the beverages. So that's why it is called what? Baker's yeast and brewer's yeast. So rarely unicellular. It means most of the fungus which comes under this class are nothing but multicellular only. But very rare, it is unicellular like yeast. Once again, it could be a NEET question. They are saprophytic. That means we all know that they depends on dead and decaying organic matter. Decomposers, parasitic or caprophilous. Caprophilous means which is growing on down. Again, it could be a NEET question. Caprophilous, it can be asked in the form of mastoporoid. Right? So Mycelium is branched and septate. So that is what I said in the beginning. Except Phycomycetes, the rest of the fungus or rest of the class fungus, what you are seeing? Ascomycetes, Basidiomycetes, Deuteromycetes, all are septate. Septate means, so their hypha will be having a cross wall like this. Okay, this is nothing but septate and branched. The asexual spores. Now, from here, let us talk in detail. Just through diagram, okay, just like how we have learned in Phycomycetes. But once again, to remember the examples of this class, let me tell you a small trick, okay, so that you can easily remember it. Yes, Ascomycetes. Here, purely we are focusing on the reproduction part. Let me take an example of a Penicillium, okay, or Neurospora. Neurospora also comes under this category of Ascomycetes. Right, imagine, this is the mycelium, which are branched and septate like this, which will be having nucleus in each septa. Okay. And here, there is a special branch. There is a special branch. On this special branch, this one also once again, branched and septate. A small spores are forming. Right? So, let me write the spores here only in this structure. Or let me take some other structure. Some other color. So, these are the spores which are present outside. They are forming outside. Whereas in Phycomycetes, which are forming inside. But here, they are visible. See, they are outside forming. Right? So, such spores, what you are seeing, they are called conidiospores. Conidiospores are conidia. Conidia. In simple language, you can call it as conidia. So, how they are forming? They are forming exogenously. Now you got the meaning. You got the difference between endogenous and exogenous. Exo means outside. Now, along with the air current, when they get fallen out, that means fallen off from these conidia. That means it is a special branch called conidiophore. Conidiophore is a special branch. On this special branch, the conidia are forming. So these are the conidia. So that will be fallen like this. And wherever they get a new area, or any special substratum, like it might be a cow dung or it might be any wooden piece or anything, okay, dead organic matter. So obviously it start growing above this also. It start growing and again it forming a new fungus. So this is what asexual reproduction, okay. So hope I'm very clear about asexual reproduction. Now let me show sexual reproduction of this fungus group here itself so that it would be easy for you to understand. Now let's imagine there is one more class. sorry, not one more class. There is one more genetic makeup of the same Ascomycetes, but totally its genetic makeup is different from the other fungal genetic makeup which comes under this category only. Let me take the example of Neurospora. So here, this is one Neurospora. This is another Neurospora where its genetic makeup is different. Okay. So now it is also forming a conidia here on its surface. Are you getting my point? So it is also forming conidia. Now, apart from falling on this substratum or any wooden piece or whichever be the substance, now this conidia, if it stand over here or if it sit on this conidia of another Neurospora or another fungus, then what happens? Here they start forming the sexual reproduction because both of these fungal genetic makeup is different. Now what I said, all the nucleus which are present in the fungal body are haploid only, right? So haploid means they are N. So here also this fungal genetic makeup is also N and this fungal genetic makeup is also N. Now both are ready to fuse their cytoplasm. Okay, so there is a small wall-like structure will be there. There is nothing but the chitin. So they start gradually melt each other so that the barrier which is so present between these spores of conidia, they started melting and cytoplasm of these two spores gradually fused together. Okay, now in the next step, if you just observe, it somehow looks like this. Here itself, let me take one more color to represent. So now you can see the structure like this, where both of these conidia cytoplasm had got fused. But listen to me carefully, their nucleus is still as it is apart. So what is the first step here then? Of course, it is a plasmogamy. This is the first step. Plasmogamy. Here, fusion of protoplasm occurred. Fusion of protoplasm took place. Now, except Phycomycetes, in Ascomycetes as well as in Basidiomycetes, these nuclei do not fuse together immediately after protoplasm fusion. It takes time. They are very lazy. They are telling there is time. So we will be separate as it is. We won't fuse together. We won't come together. So like that, they make a strike. They will be sit as it is for several years. It might be 10 years, 20 years, hundreds of years. Now, when you just look at this structure, the nucleus is as it is apart, no? Then such condition we call it as what? N plus N condition. Why N plus N condition? Tell me. Because this nucleus is also N. This is also N. This is also N. Therefore, both are N plus N condition. So, such condition is known as what? Dikaryon. Dikaryon. Now, you can understand the meaning of the paragraph which is given above the Phycomycetes. Okay, so that's what I said. While explaining the different classes of fungus, I will explain the reproduction mode which is given actually in the beginning itself. Okay, now this is the dikaryon stage where N plus N is separate only. Now the structure what you are seeing now, it is somewhat like a bag. Correct. So this bag-like structure itself we call it as, this one is nothing but ascus. This is called ascus. Now let's assume it might be took 100 years. Now after 100 years, they start analyzing, they start realizing that, oh, this is our time. Let us fuse together. Now both the nuclei start coming together and they fuse together. That condition is what? That is the second step in sexual reproduction. That is karyogamy. Now karyogamy also took place. Let me write only the ascus. Let me take only the ascus here. So now this N as well as this N fuse together so that it forms a single nucleus. This condition is what? This is nothing but 2N now. They are diploid. So this is the second step called karyogamy. So hope you are understanding. So karyogamy occurs within the ascus. Now in this ascus, they start thinking, now it has become a diploid. So now what is the third step after diploid condition? It has to undergo meiosis. Meiosis means one cell has to divide into 4 cells. So as a result, haploid condition again formed. Correct. So now here, the third step is taking place. The third step is meiosis. Remember, these 3 steps is common in every sexual reproduction. Whether it is a Phycomycetes, Asco, Basidio or anything. Okay. But these 3 steps, one after the other, plasmogamy, karyogamy, this is the second one, and meiosis is must and should. Now let me draw again this ascus, which is undergoing meiosis and forming 1, 2, 3, 4 nuclei. So all these nuclei are N only now. Earlier they were 2N, now they are N. But now they are telling, instead of having only 4 nuclei, let us make 8. So if they want to become 8, but their number should be N only, then which step they have to go? Which cell division they have to follow? Yes, you are right. It is mitosis, not meiosis. Because if the chromosome number is N, the meiosis do not take place. It should be mitosis only. Okay, now they start thinking that let us make one more mitosis. Then they start doing mitosis here. But such thing we didn't see in the case of Phycomycetes. Here it is quite different. 1, 2, 3, 4. Because of mitosis, one cell, one nuclei start giving 2. Therefore, 1, 2, 3, 4. 8 nuclei we got. Understood, no? 8 nuclei, all are haploid nuclei we got. But actually, in this conidia, or in this fungal, on the surface, this ascus which is so forming, it is not a single ascus. The group of ascus or group of such a sac-like structure will be formed. Suppose all these structures are forming here only, no, on its surface. This is the hyphal structure. On this, above only this ascus has formed. Now imagine it is having 1, 2, 3, 4, 5, 6, 7, 8. Now, remember, it is not a single structure. It will be having more structure like this, more structure like this, more ascus, more ascus, more ascus. Okay? In all this, now in a plural, we won't call ascus. We call it as what? Asci. These are asci. So in each ascus, we are seeing 8 nuclei. 8 nuclei. But now they are telling we need a bag-like structure so that we will be get dispersed. If we are together, then we could make a balance, right? So that's why a bag-like structure is going to form around them. Something like this. Something like this. A bag-like structure. Right? So this bag-like structure is nothing but a fruiting body. It is called fruiting body. Now this entire structure, what you are saying, this fruiting body inside the asci are present. It is nothing but ascocarp. So what is the fruiting body of Ascomycetes? Ascocarp. Now let us go back. Here we can see many asci, correct? Many asci like this, which are enclosed within this bag-like structure called ascocarp. Correct? Now this is the ascocarp structure. Ascocarp. Now here, these nuclei start getting matured. They start releasing the spores. That means, along with the nuclei as well as the cytoplasm, they start releasing out because they start getting burst out. Now each nuclei along with some quantity of cytoplasm, they start releasing outside like this. They start coming outside. So again, when they fall on any substratum, again they start growing into a new fungus. So these structures, what you are saying, they are nothing but, where shall I write? I will write here only. They are ascospores. So ascospores are sexual spores. Understood now? So suppose if I want to show the same structure here, soon after their maturation, they start getting burst out and forming these ascospores. Ascospores are sexual spores. So when they fall on any suitable substratum, again they start growing into this fungus. So again the cycle starts going again. If the conidia fall again on the new substratum, it is nothing but asexual. Or if they undergo such type of cycle, this is a sexual. So hope I am very clear. So this much what we have seen, up to this much, this is the asexual reproduction. Whereas this part is sexual reproduction. So all this schematic representation is not given in your NCERT. If you make a note of all these things, just like what I have drawn here on the board, if you also make an image, diagram, trust me, it helps you a lot during exam. Okay, need not to study NCERT textbook at the time of exam. It's like a quick revision. Understood? Now let's go back to the text part. Now we can understand. The asexual spores are conidia. produced exogenously, that means outside, on the special mycelium called conidiophore. I said no, there is a special branch. The conidia on germination produce mycelium, that means whenever they get a suitable substratum or a matter, they start germinating and produce a new hypha. Now, from here, the sexual reproduction starts. The sexual spores are called ascospores, again it is important, which are produced endogenously in the sac-like asci. See, it is endogenously, which are forming inside only. Because of the bursting action only, they are releasing out, correct? But whereas these things are produced outside, therefore it is exogenous. This is endogenous. Where they are producing sac-like asci. Asci means singular is ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Okay, some examples. Now, to remember the example, I have made a trick. So here, which examples they have given? Mainly, they have given the examples like Aspergillus, Claviceps, Neurospora. Neurospora is used extensively in biochemical and genetic work. Again, it is important. Which fungus will be used in genetic work? Okay, that is Neurospora. Many members like morels, truffles are edible and are considered delicacies. So here, only few fungus name they have given. Let me show here. First, let me write all the fungus. First they said, here Penicillium as well as yeast example they gave. Penicillium and yeast. Right? And after, they gave the example of Neurospora, Aspergillus and Claviceps. Okay. So 3, 4, 5, 6 and 7. And rest 2 are morels and truffles. Remember, these morels and truffles are edible fungus. Again, it could be a NEET question. Which of the following is edible fungus? So for all this sum, I can't frame the question. So while doing lesson itself, whichever the point that I will tell, so this could be NEET question, this could be this one. So just make a note in that. Okay. And here, Neurospora, it is used in genetic work. Again, NEET question. So how to remember all these fungus so that it should be comes under the group of Ascomycetes? So it is a small trick. Usually I used to remember in this way. Suppose if I tell this trick, it could be helpful for you. So why usually I remember that ascus is nothing but a bag. Ascus means bag or a sack, I said, right? So here is a boy. So let's imagine what is there to imagine, right? So a boy. So now this boy, he is going for a genetic research work. So my way of remembering, if you have any other tricks, you can put in the comment box so that it could be helpful for other student. So he is carrying a bag. So this bag is nothing but a sack. So that's why it is Ascomycetes. Now, as he is moving for research work, no, first he had put some Neurospora inside his bag. So how we can remember Neurospora here? Neurospora, it is a genetic work, correct? And now he is planning, on the way, I may get bored, right? So that's why, just to make a time pass, or I may feel hungry, that's why let me take some edible fungus. So he had put some edible fungus, morels and truffles. Morels and truffles, so that he can eat along on his journey, okay? Now, once again, you know that on his way, he may get few friends. So that's why he is planned to make a party with them. So for making party, he is planned to carry some beverages. So who is the best fungus for making beverages? Of course, it is yeast only. So for that, he is carrying yeast also in his bag. It is a, what is this? Baker's yeast or brewer's yeast, right? And again, he is so cautious. Suppose on its way or on his journey, if he gets stomach upset or anything happened, that is to his health. So there should be some antibiotic, no? So which fungus is responsible for preparing antibiotic? Of course, it is Penicillium. From this, we can prepare antibiotic called Penicillium. So that's why he thought, why can't I take a Penicillium along with me? So he's carrying Penicillium also, right? Now, he know that the place where he's going, it is having a lot of, that means it is not so cool environment. It will be having lot of issues, so that if there is a AC, so that I can feel so comfortable. Okay, so there might be mosquitoes, there might be very hot. So that's why he's carrying AC along with him. AC means Aspergillus and Claviceps. So now if you count, all our 7 fungus came here. Aspergillus 1, Claviceps 2, Penicillium 3, Yeast 4, Truffles, Morels 5 and 6, Neurospora 7. Over. Okay. So here the main point you have to remember is these 2 are edible fungus. This will be taken for genetic engineering. From this, antibiotic will be prepared, and from this, the bread, what is that? Beverages, everything we can prepare. So this is a small trick how I used to remember what I said just now. If you have other trick, you can put in the comment box. Now look at this image. This one over. This one over. Yes. These are morels and truffles, edible fungus. And this one, Claviceps. This is actually a parasite which causes disease in the wheat plant. Can you see a black color structure here? So these are nothing but the Claviceps. Right? And rest of the things are very microscopic. That's why I didn't put the image of those. Now the third category, or the third class of the fungus is nothing but Basidiomycetes. Come, let's read. So once you understand the sexual mode of reproduction of any one fungus, the rest of the things are easy. The only thing is you need to understand the differences. Small differences like asexual spores, sexual spores. Okay. Here, they are commonly called Basidiomycetes are mushrooms. The commonly known form of Basidiomycetes is mushroom. Remember, mushroom. Bracket fungi or puffballs is also a common name we have given to this. So look at this image. This is a bracket fungus which is so growing on the tree trunk. It looks like a bracket. How we are going to put a bracket? In the same way it is looking. That's why it's a bracket fungus. And it is also called puffballs. Look at here. It's just like a puff. It looks. And obviously we all know about this mushroom. Correct? So all this comes under the Basidiomycetes only. Where they are growing? Obviously, they grow on the soil, on the logs and tree stumps, and in living plant bodies as a parasite. That means, always they are not good. They are even very criminal. That means, they cause a disease in certain plants. For example, the rust fungus, the smut fungus comes under this category only. The mycelium is branched and septate, as I said, except Phycomycetes, rest of all branched and septate. Now, listen here. Asexual spores are generally not found. That means, here, thank God, we got escaped from studying asexual reproduction. So, no asexual reproduction here. So, this is one important criteria we need to remember in this fungus. Right? Next thing. But vegetative reproduction by fragmentation is common. I said what is fragmentation? If any fungal pieces get cut down, from that cut part only the new fungus start growing. Okay, so vegetative reproduction. The sex organs are absent, but plasmogamy is brought about by the fusion of 2 vegetative. From here, let's understand through diagram. Okay, that is our schematic representation. Yes, let me show here. They are very smart here. These fungus are. Let's imagine this is one fungus. Which belongs to Basidiomycetes only. This is septate and branched. And another fungus is growing near to this. So both are coming near because they are having a plan of doing what? Sexual reproduction. Sexual reproduction. So now, what they are doing? They do not have any special sex organs. Because I said they are haploid only. If they are haploid, their nucleus is young. Why they have to produce gamete? They themselves act as a gamete. Correct? Now, during the process of sexual reproduction, they form a branch, a side branch. Okay. So here also a small outgrowth is going to form. Now, it is also growing. Let me make in some other color. Okay. Let me do like this. So this is also growing. And this one also fuse with this, and this is also growing. Now it is forming a special branch. They are growing continuously. Let me make it in some other color so that you can understand it clear. So this is a special branch formed. by the fusion of these two fungal hypha. Okay, but remember, here their nucleus is separate only. Their nucleus is not yet fused. Though their protoplasm fused, but nucleus not yet fused. That means which step has occurred here? In sexual reproduction, the first step is plasmogamy. Plasmogamy took place. Now, this branch grows continuously. Grows rapidly and continuously. Rapidly and continuously. They itself turned into a bulb-like structure. Okay. Now, if you just look at the structure, how it formed, it somehow looks like this. Okay. So, now, here, below this, it will be having many small structure which are present beside it. That means, here I took the example of this mushroom. Can you see here a small lines? Are you seeing below this? These are nothing but the gills. So here you can see the gills. So overall this part, what you are saying, this is nothing but a fruiting body. So this fruiting body, they grows continuously and form a fruiting body. This fruiting body itself we call it as what? Basidiocarp. Basidiocarp. Why the name Basidiocarp? Because within the gills there are small structures present. These are nothing but the basidia. They are lined by basidia. If I just enlarge these basidia, some things looks like this. Now, within this basidium, the nuclei is apart itself, just like as we have seen in the earlier structure, they are apart only. Correct? So, what they are doing? They are following N plus N condition only. N plus N, that means dikaryon. Dikaryon for several years. But on one fine day, the nucleus also start getting fused. So, once the nucleus fused, it forms a structure called what? Structure is same. This is basidia. Let me write the basidia. Singular is basidium. Plural is basidia. Many basidia it is having. Now, within this basidium, when two nuclei fuse, then the nuclear number become what? 2n. So, now let me draw this structure. Inside this basidium, this nucleus and this one nucleus fuse together. Fuse together. Now, this is 2n. It is nothing but the zygote. So, once the zygote form, obviously it has to undergo the next step. So, this is the second step called karyogamy. This is plasmogamy. Right? Now, it is going to the next step called meiosis. So, now, after meiosis, 1, 2, 3, 4. 4 nuclei are formed. So, all these 4 nuclei are nothing but haploid only. Haploid only. Now, they are thinking. These nuclei, instead of sitting inside, they come forward, they come outside along with certain cytoplasm and they start forming the spores. Now, if you just look at the same structure, let me draw here something like this. This structure, they form like this outside. So, these nuclei move outside. Okay, now inside, I will rub this part. I will rub this part. Okay, so all these spores are comes out. So, we can say the spores are forming exogenously. Exogenously. So, these spores are nothing but basidiospores. So, when basidiospores get removed from this basidium, they become free. When they land on the suitable substratum, again they start growing into a new fungus hypha. Okay. So, this is how the process of sexual reproduction occurs. Here, remember, they do not have asexual reproduction. So, that's why if you just remember the sexual spore, that is basidiospore, that is enough. Now, let's go back here. Let's read NCERT textbook. So, here I have stopped now. Yes, the plasmogamy is brought about by the fusion of vegetative or somatic cells of different strains or genotype. That means somatic strain in the sense this fungal hypha is different genetic makeup and another fungal hypha is having its own genetic makeup. Both are coming together. Okay. The resultant structure is dikaryotic. Dikaryotic means the nucleus will be separate, which ultimately give rise to basidium. That means they form a fruiting body. Inside the basidium will be present. Karyogamy and meiosis takes place in the basidium. Of course, that we have seen. The 4 basidiospores are formed, correct? There 8 we have seen in Ascomycetes. Here 4 basidiospores are forming. The basidiospores are exogenously produced. Remember, it is not endogenous. In Ascomycetes, it was producing inside. Now they are producing outside on the basidium. Okay, plural is basidia. The basidia are arranged in a fruiting body called basidiocarp. So that bulb-like structure, button-like structure is nothing but basidiocarp. Some common members are agaricus. Mushroom is nothing but agaricus only. And ustilago, it is called smut fungus. They are parasite. Remember, it is very, very important. Very, very important. Puccinia, rust fungus that we have seen. Rust fungus in the beginning of the session itself. Beginning of the session in the missions. The kingdom fungus, I said. The small rust fungus, Puccinia, is a parasite. So these comes under the Basidiomycetes only. So the main example here we need to remember is APU. APU. I don't know. I have not remembered any tricks here. A means agaricus. P means Puccinia. U means ustilago. This one. Ustilago. So if you just remember, finish. Right? So Basidiomycetes part is over. Now we are in the last part called Deuteromycetes. Deuteromycetes is very, very easy. Very, very easy. It's a poor fellow. You know one thing? What the scientists are going to do? First of all, one main important function, important character you have to remember in the Deuteromycetes is it do not show sexual reproduction. No sexual reproduction. Whereas in the case of Basidiomycetes, no asexual reproduction, but here no sexual reproduction at all. So, if the fungus is not showing sexual reproduction, then we call it as what? It is an imperfect. Man, you are imperfect. So, it is an imperfect fungus. Imperfect fungus. Okay? So, the funny thing is, the scientists, whenever they made research a new fungus, first they start researching about their sexual and asexual reproduction. Suppose if they do not understand the sexual reproduction mode, or if they do not notice the sexual reproduction in the newly discovered fungus, they just put into this category of Deuteromycetes. When another scientist, when they start researching or when they came to know that yes, it is showing some sexual mode of reproduction, so that it is producing some basidia or some spores which are producing outside or inside, then they start thinking that, oh, it might be belong to Basidiomycetes, or it might be belonging to Ascomycetes, but they cannot be belonging to Phycomycetes because this one also is having what? Septa. So whenever you have a septa and branch hypha, of course, it is not comes under the category of Phycomycetes. So this is what the funny part. So whenever you don't understand the sexual reproduction, just go and dump into this Deuteromycetes. So this is what the thing is happening. But when they come to know, again they will be placed. Okay. So that's why it is called what? Imperfect fungus. So here the main example you have to remember is cat. Coletotrichum. Coletotrichum. Alternaria and Trichoderma. So this Trichoderma will follow you even in your class 12th. That is in Microbes in Human Welfare chapter. It is very beautiful fungus which protect the plant roots from attacking pathogen. Okay, it's just surround the fungus. It acts like a barrier. Oh, if any pathogen comes, I will attack you. So like that, unknowingly it will do its work and protect the plant roots. But these are the main example. Let's read this. But it is having asexual reproduction. Asexual reproduction is done by conidia formation. That's it. See, commonly called imperfect fungi, as I said, because only the asexual or vegetative phase of fungi are known. That means only asexual reproduction you have understood, and vegetative, of course, either by fission or fragmentation. That's it. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. That means when they came to know the sexual reproduction, then they start moving into the right classes, either into Phycomycetes or into Basidiomycetes. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Sometimes it do happens. When they come to know the asexual reproduction, that will be placed in one fungus. There is sexual reproduction, asexual is in one category, sexual reproduction is another category. Like that, sometimes it happens. Later, when the linkages were established, the fungi were correctly identified and moved out into Deuteromycetes. Understood? First, the same fungus. Oh, you are showing asexual reproduction. Come on, sit here. When they came to know that your sexual spores are like this, your sexual reproduction is like this, oh, we did a wrong thing. Okay, just go to your class. Understood? Like that, the scientists were doing. Once the perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes or Basidiomycetes. The same thing. The NCERT lines are repeating. That's it. The Deuteromycetes reproduce only by sexual spores. Important. Known as conidia. So, conidia is the name we even learned in Ascomycetes. Correct. Here also the same spores, conidia. The mycelium is septate and branched. Some members of saprophytes, that means grows on dead organic matter, and some are parasites, while large number of them are decomposers of litter and help in mineral recycling. Some examples are Alternaria, Coletotrichum and Trichoderma. Coletotrichum, look at the spelling, Coletotrichum. Yes, I did right. Okay, so this is about Deuteromycetes. So hope I am very clear about all these classes of fungus. So before entering into check our knowledge by solving few MCQ, let me make a table so that you have to tell the answer. Of course, I can't see whether you are telling the right answer or wrong answer, but if I make a table like this, it would be helpful for you. Okay, let me write here fungus name first. Yeah, here I will write. Just make this column along with me. So let us cover the first point. It is commonly called, or common name. What is the common name to this? This is primitive fungus. So Ascomycetes is commonly called a sac fungus. And Basidiomycetes is commonly called puffballs. Correct? Or even bracket fungus. So these are few things we will learn here. And Deuteromycetes is imperfect fungus. Okay? Only the main characters I am writing here. Now the next point. Next point. About its hypha. So hypha here is aseptate and xenocytic. So this is the only fungus showing like this. Rest of all branched and septate. Usually where we are going to stuck while studying the fungus, of course in sexual and asexual reproduction. Correct? Now let me make the asexual reproduction here. So here in Phycomycetes, it produces aplanospores. So aplanospores are non-motile, whereas zoospores are motile. Correct? While Ascomycetes, it is conidia or conidiospores, whereas Basidiomycetes, no, it is, first of all, it is absent. In Deuteromycetes, the same conidia. Now you are feeling it easy, correct? So asexual reproduction part is over. When it comes to sexual reproduction, right? Here the sexual spores are zygospores. Here Ascomycetes, ascospores. Here Basidiomycetes, basidiospores. Here it is what? No, this is absent, right? So many things we have learned. So the last part is example. I don't have any space here, but let me try to make here only examples. So here I said, just remember the name RAM of a student. RAM, Rhizopus, albigo and mucor. And here Ascomycetes, that is back. So here the boy is going for a research work. He carrying neurospora. And on his way, he want to enjoy. Therefore, to satisfy his hunger, he is carrying morels and truffle. To make party, he is carrying the yeast, that is beverages. And once again, what is the other set? Yeah. He is carrying antibiotic called penicillin. That means he is having so much cautious about his health. Then he is also carrying AC, so that if there is any hot, so that he can cool his body. So these are the examples we could able to remember here. And in Basidiomycetes, I said APU. APU means Agaricus, Puccinia, Lustilago. And here in Deuteromycetes, I said you can remember through cat. Coledotrichum, Alternaria and Trichoderma. That's it. So if you learn this much, not this much, you have to learn each and every line. That's why I put NCERT text only. Sometimes we don't know how the questions can be framed in sentence formation or in a statement based. So that's why it is very important to read NCERT. But the basic point, must and should, you have to be very clear. That is this much. Understood? Now let's move on to the questions part. So hope you are making a table along with me. You made this table. You made this table. Right? Yes. The sac fungi belongs to where? Ascomycetes, Basidio, Phyco, Deutero. Of course, it is Ascomycetes because ascus. So now easily you can answer. Within fraction of second, I know you can answer because the concept is crystal clear, I think. The Basidiomycetes include rust, smut, mushroom, all the above. Rust fungus, Puccinia, smut fungus, Puccinia, rust fungus, Ustilago, okay, mushroom, all belongs to what? All belongs to Basidiomycetes only. All of the above. All of the above. Correct? APU, I said. Hey, this is APU only. Agaricus. This is Puccinia. This is Ustilago, APU. So the questions will be twisted and asked. Sexual cycle is absent in. Where sexual cycle is absent? Not in Phycomycetes. Of course, in Deuteromycetes. That's why it is an imperfect fungus. Here it is there, it forms ascus. Here it is there, it forms Basidium. Next question. Penicillium roqueforti. See how the question is twisting. Penicillium is the genus. They said Penicillium comes under what? In Ascomycetes. But just to twist the question, just to make you get confused, they are giving like this. Penicillium roqueforti. It is a separate name. It's a special species. Like that, Penicillium camemberti are used in the preparation of cheese, of course. That you will learn in your class 12. These fungi belong to the class. Whenever you just heard the term penicillium, just go and put into where? Where you have to put. That is in a bag. Ascomycetes. So this is the answer. Not in these classes. Next. Cenocytic mycelium is found. Cenocytic and aseptate is the characteristic feature of Phycomycetes. Correct. So Phycomycetes, directly they have not given about Phycomycetes, but instead they are giving the example. So what I said, Phycomycetes is primitive. So the Ram is very old student of our NAC biology. So therefore, Ram, Rhizopus, Mucor and albugo. But here albugo name is not given. Rhizopus is given, Mucor is given, but Penicillium belongs to Ascomycetes. Therefore, let us go with this option, both A and B. Understood now how the questions will be asked in your exam? Next, the chief characteristic of class Ascomycetes is. So, Ascomycetes, formation of spores. In every classes of fungus, spore formation is common. It is not a chief character. Hyphae, of course, in every class, it is also formed. Formation of ascospores. Ascospore production is the characteristic feature of Ascomycetes only, not in other class. Therefore, this is the right answer. Not zoospore. Zoospore is the characteristic feature of what? Phycomycetes. Normally, how many ascospores are formed in an ascus? So, in an ascus, 1, 2, 3, 4, 5, 6, 7, 8 nuclei. All these 8 nuclei converted into spores. So, totally how many spores will get? 8 ascospores. So, this is the answer. But this word is not given in NCERT. So, sometimes we need to use our common sense. Okay? It's not A, C, D. Next question. Occurrence of dikaryotic mycelium. Dikaryotic mycelium means what? In this hypha, the nucleus will get separate like this. That means N plus N condition. So, that is nothing but dikaryotic stage. Mainly is the characteristic of. Apart from Phycomycetes, both in Ascomycetes and Basidiomycetes, we are going to see in this condition. But the long time duration, N plus N condition remain as it is in Basidio compared to rest of the thing. Myxomycetes, we don't know. Phycomycetes, not. Deuteromycetes, first of all, no sexual reproduction at all. Okay. So, N plus N condition is a step, or it is a step comes under sexual reproduction only. So, therefore, of course, it's the answer of Basidiomycetes. Next question. Deuteromycetes are called imperfect fungi. Why are imperfect fungi? Because you don't show sexual reproduction. No sexual reproduction. That's why you have cell wall, you have mycelium, you have asexual reproduction, but not sexual reproduction. That's why it's a imperfect fungus. Hope that's it. So hope the concept is very clear for you. Now let's move on to the kingdom Plantae. Okay. So here I will tell you now itself. There is a separate chapter called Plant Kingdom. There is a separate chapter called Animal Kingdom in first unit itself. Okay. So we'll study in detail about the plants, their classes, their characteristic features, everything in Plant Kingdom. Right? But here only few things are given. There is a basic points that I will tell. Rest of the things I won't tell anything that we will learn in Plant Kingdom. Okay? Yes. Okay. Kingdom Plantae. So let me tell you few things about this Plantae kingdom. So first of all, we all know that this is the fourth kingdom in the classification of Whittakers. So one common sense we have to use here that all plants that we are going to see on the earth all kept under the kingdom called Plantae. So here basically all the plants are put under 5 categories. Okay, so just like how we have kept all the organisms under 5 kingdom, in the same way, all the plants which are having green in color, it is nothing but it is understood that they will be having chlorophyll in them. So if they have chlorophyll, obviously they have which property? To absorb the sunlight and to prepare the food, right? So there is a main property that we are going to notice in plant kingdom. That means organisms present in plant kingdom, right? So let me write here these points. They have chlorophyll. And obviously, they are autotroph. You know the meaning of autotroph. That is, they can prepare their own food. Correct. Now, if I just make a 5 groups here under this plant kingdom only, let me write here. Yes, we are going to get algae. So these are primitive plants. When the evolution started taking place, when the plants started forming on the earth, then the first formed plants on the earth are nothing but the primitive plants called algae. We are going to talk about all these things in detail in the plant kingdom chapter. There I have done everything in detail I have done. But here you should not get panicked that ma'am has not said anything in this chapter because here NCERT has given about plant kingdom. So that's why to solve that doubt in your mind, I'm just explaining here in a brief. Right? And now, after algae, next evolved plants are bryophytes. And after that came pteridophytes. Then gymnosperms. And the last one is angiosperms. That is nothing but the flowering plants. Okay. So, all these plants are kept in a order of their evolution itself. Right. So here these algae and bryophytes, they are primitive. Compared to bryophytes, algae are primitive. So for their survival, water is must and should. Because one thing it is very clear that when earth has formed, the first life also has formed in the water itself. So that's why when you take first animal that has found in the water, when you take first invertebrate that started surviving in water. So like that, when you take first plant, it started its journey in water itself. So that's why here the algae prefer aquatic environment. Even bryophytes, they become quite advanced compared to algae, but still they require water for their sexual reproduction. Like that, when we come here, the pteridophytes, they are quite advanced compared to bryophytes, but here also the advancement started seeing in their vascular tissue formation, that is xylem and phloem formation. And purely terrestrial, we are considered these two, gymnosperms and angiosperms. So angiosperms are flowering plants. Majority of the plants, what you are going to see in our surrounding are nothing but this angiosperms only, right? So when you just look at all these plants, obviously they are all which color? They are all green in color. Why they are green in color? Because of this chlorophyll pigment. So therefore, they comes under the category of autotrophic nutrition. But there are few exceptions. For example, have you heard the name of insectivorous? Insectivorous plants. So in your lower classes itself, you have understood, you have studied about insectivorous plant. That means they kill insect in order to balance their nitrogen qualities. That means nitrogenous food. So they cannot prepare their own food. They do not have chlorophyll. So that's why they will feed on the insects. So in such plants, we won't see these features. Okay, it is quite exception. So like that, even there are certain exceptions we can see in parasitic plant. So the best parasitic plant. So first of all, see, it is a strange thing to look at a plant as a parasite, because we came to know that many parasite, it may be a bacteria, it may be a fungus. Okay, so they depend on plant because the plant acts as a host for them. So by considering plant as a host, these these organism, either it is a bacteria or a fungus, they are depending on the plants. So these are the parasite. But now here it is a strange thing. So these plant itself, they are behaving like a parasite. So the best example here we can notice is Cuscuta. So it could be a NEET question. So the Cuscuta, so again you are going to study this example in your class 12. Imagine this is a plant. Okay, so in this plant, this Cuscuta, it is somehow surrounds like this. It is going to pierce their root inside this plant only. So they do not have any chlorophyll pigment. Cuscuta, they do not have chlorophyll pigment, no chlorophyll. But it is getting all the nourishment, all the food which has been prepared by the host plant. Imagine this is the host plant. So for the food, it is depending on the host plant. For their survival also, for their support also, they are depending on this host plant. So therefore, this Cuscuta, it is a parasite. That's it. In general nature, general thing, already we know about the cell wall. So whenever you take any plant cell, if you consider this cell as a plant cell, then obviously around this cell, there is a cell wall. So the cell wall is made up of what? Cellulose. So this is a basic thing already we have understood in the beginning, right? So this is some general thing. That is what given in the paragraph. See, let's read this. Kingdom Plantae includes all eukaryotic. See, already as we move forward from Protista group, all the organisms are eukaryotic only. So it is a chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plants or parasite, bladderwort and Venus flytrap. So these are the examples of insectivorous plant. And Cuscuta is a parasite. Again, it could be a NEET question. The plant cell have an eukaryotic structure with a prominent chloroplast. See, chloroplast is an organelle where chlorophyll pigment is present. So you are going to study about the plant cell in detail in the cell chapter. In this class only, class 11th only, there is a separate chapter called Cell, the Unit of Life. There we are going to study about the structure of plant cell as well as the detailed structure of animal cell. But this is the basic thing. If it is a plant cell, there is a structure called chloroplast present. Okay, so it's a chloroplast. And a cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Okay, Plantae includes algae, that's what I said, bryophytes, pteridophytes, gymnosperms and angiosperms. Right? Now, so this is the basic information I gave just now. And in the next paragraph, they are talking about alternation of generation. So alternation of generation means when you just look at a plant or a plant body. Imagine this is a plant. So it can be either algae or a bryophyte or a pteridophyte or angiosperms or gymnosperm, anything. So depending on the nature of the plant, here the ploidy in the sense whether it is a diploid, that means which is having 2n in number in each cell, the chromosome number, or it can be a haploid, that is each cell will be having n, half set of chromosome number, that we are going to decide based on the plant body, based on the plant's nature. So they are going to change there. They are going to shift from diploid nature to haploid to diploid. So in their life cycle, that we call it as alternation of generation. See what it is given. The life cycle of plant has two distinct phases: diploid sporophytic and haploid gametophytic. So this diploid nature is called sporophytic and haploid nature is called gametophytic. I know you are getting lot of confusion. I know it very well. But don't worry. This part, alternation of generation part, has got deleted from our NCERT. Because in plant kingdom, in earlier chapters, that means before the revised syllabus, earlier when you just look at the old textbook, there was a detailed explanation regarding alternation of generation. But this year in the sense after revised syllabus, this part has been deleted. But still, if you don't have a basic knowledge about alternation of generation, literally you are going to get a lot of confusion while reading the plant kingdom chapter. So that's why by having this thing in my mind while doing class, I have covered alternation of generation. I have explained everything in detail about alternation of generation. And after that, I have covered the rest of the syllabus which is given in NCERT. Okay. So if you just watch that video, trust me, 100% you won't get any doubt. So seriously, you can easily understand. Right? So that's why here I won't explain anything about alternation of generation because it is purely related to plant kingdom chapter. Okay? So that's why here name itself is telling kingdom Plantae. Because there is a separate chapter for plant kingdom, you should go and study there. That is what the indication. Here they have just created curiosity. What are the things that you are going to study there? Okay? So still, let's study the rest lines that alternate with each other. That means sporophyte and gametophyte are going to alternate. First, it will convert it into haploid and diploid can be converted into haploid like that. They are going to shift. The length of the haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups of plants. Different groups in the sense bryophytes, algae, pteridophytes, like that it will be vary. This phenomena is called alternation generation. Look at here, NCERT is very smart. It is giving this line. You will study further details of this kingdom in chapter 3. But unfortunately, they have deleted. But still they are telling this line. You will study in chapter 3. So, if we are smart, what we need to do? We need to learn about alternation generation. Suppose if any question comes from this paragraph, we should not leave as it is that it is a deleted syllabus. Understood? Because they are given it, no? You will study in chapter 3. Okay? So, this much is enough regarding kingdom Plantae in this chapter. Okay. Now, the next kingdom is kingdom Animalia. So here once again, I will tell, I have told already everything I have told in Animal Kingdom chapter, but what is given in this paragraph? Let's read it. The kingdom is characterized by heterotrophic eukaryotic organism because animals are heterotrophic only and eukaryotic. So their cells lack cell wall because they do not have cell wall. They directly or indirectly depending on plants for food. If they are herbivores, directly depending. For example, a cow, sheep, directly depending. If you are carnivorous, indirectly depending. By eating cow, it is understood that the cow has eaten the grass. Correct? So like that, indirectly we are depending. They digest their food in an internal cavity and store reserved food as a glycogen and fat. So in our body, the food, excess of food, what you are going to eat, where it is going to be stored? Of course, in liver. In liver, in the form of a glycogen. But still, if you keep eating excess food, where it is going to store? Of course, it is going to be stored under the skin as a fat. So that's why the skin is going to be get plumpy at the same time. It will be get stored by a lot of fatty tissues. The mode of nutrition is holozoic. Already have told the meaning of holozoic by ingestion of food. They follow definite growth pattern and grow into adults and have definite shape and size. Higher forms show elaborated sensory and neuromotor mechanism. So as we move up in the animal kingdom, that means here also in animal kingdom, first invertebrates like Porifera, Coelenterata, Annelida, Arthropoda, like that, their group has been placed according to evolutionary basis. So as we move ahead in their categorization, that means in the characteristic, if we keep studying all these characteristics according to their evolutionary relationship, so in higher organisms or in advanced organism, we are going to see the advanced features like development of neuromuscular system, that is the brain, everything has started developing. That is what the meaning here. The most of them are capable of locomotion. See, when you just look at the lower animals like Porifera, sponge, they are sessile. Sea anemone, sessile. Sessile means they do not move. They will be just stick on to one particular place. But as you move up in the evolutionary pattern, obviously the animals start showing the locomotion. Okay. The sexual reproduction is copulation of male and female, followed by embryological development. That means the zygote has formed. The zygote will be developed into embryo. The salient features of various phyla are described in chapter 4. So that is what I said. No, the same thing NCERT is telling. We are going to study about all these things in chapter 4. That is nothing but animal kingdom. Okay. Now, don't get relaxed. So that, ma'am, we have finished all the 5 kingdom. But still, one more is following us. Who is that? Virus, Viroids, Prions, and Lichens. Correct. So Whittaker, what he did? He grouped 5 kingdoms. Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. So when you just recall what we have learned just now, we will get the answer here itself. Kingdom Monera includes all unicellular prokaryotes. That is nothing but all your bacteria comes under this kingdom. Correct? Whereas in Kingdom Protista, he has placed all unicellular eukaryotes. Okay? So many organisms like diatoms, dinoflagellate, euglenoids, everything came here. So in Kingdom Fungi, all fungus. Either it is a unicellular or multicellular, everybody has kept under this kingdom. Whereas in Kingdom Plantae, all plants. Whereas in Kingdom Animalia, all animals. So everything has been kept properly according to their characteristic feature. But Whittaker didn't get any idea to keep these viruses, viroids, prions, and lichens in these groups where to kept. Because in his 5 kingdom of classification, that is what? Monera, Protista, Fungi, Plantae, Animalia, in this kingdom classification, he has not given any place for these organisms. Viruses, viroids, prions, and lichens. So already we have learned lichens, but still we read NCERT and don't worry. If anything, if I have left, I will tell there only while reading. So he has not given any place for lichens. He has not given any place for viruses. And viroids, prions, first of all, we didn't hear the name only. But still, if you just consider viruses itself, he has not given the place. Why he has not given the place? Because many scientists, even till today, we are considering viruses are nothing but they do not have cell only. If they do not have cell, where you are going to give a place for these organisms? Because first of all, you are not considered virus as an organism only. If it should be a living, then it must possess a cell. Here it is not having a cell, means how do you consider it as a living? Then one more question comes in our mind. If it is not a living, then how it is going to cause disease, ma'am? Because you see, many epidemic diseases which are so making us to get scary, all are nothing but because of viruses only. Just take the recent attack, that is nothing but a COVID by a coronavirus, that is also virus. And if you just go back few years, SARS, dengue, chikungunya, everything, smallpox, chickenpox. So all these diseases which make entire human being to get shake just by because of their symptoms, because of their side effects, all these because of whom? Nothing but viruses only. Then how we are going to consider this as a non-living? So for this question, we need to come here about the viruses component. Okay. When we just look at the virus component, then we can understand a little bit whether it is a living or non-living. Okay. Listen to me carefully. If you consider an organism is a living, it must possess a cell. Correct? Listen to me carefully, you will understand. It must possess a cell. Cell means what? It should have a nucleus. Even though it is not having nucleus, like bacteria. Of course, bacteria is also not having nucleus, but it is having cell wall. If you get any bacterial infection, the antibiotics that you are going to take, when doctor prescribed, so what that antibiotic is going to do in your body? Of course, it is going to fight against that cell wall. So when the cell wall get broken up, immediately the bacteria also get died. So we will get free from the disease. But if it is not having a cell wall, I am talking about virus right now. If the virus is not having cell wall, if the virus is not having any cell parts, for example, chloroplast, mitochondria, other organelles, if it is not there, then how come you going to fight against such an organism? Okay. Then the scientists started making research on the organisms after their discovery that we'll study later. Then they came to know what are all the components present in this virus body. Then they found it is having only two components. Okay, so what are those two components? It is nothing but it is just a nucleoprotein. Virus is just a nucleoprotein. So here in nucleoprotein, we are going to see two words. One is nucleo. Nucleo means nucleic acid. So nucleic acid means what? Either it is RNA. RNA is also nucleic acid, or DNA, deoxyribonucleic acid. But a single virus do not have both RNA as well as DNA. Either RNA or DNA it will be having. Okay. So like that, when you just look at its cell structure, it will be having proteinaceous component also. Proteinaceous component. So now if I just draw a simple virus, then how it could look? This is what its genetic material. Either it is a single-stranded RNA or sometimes it may consist of DNA like this. Okay, let's assume this is the genetic material. Around this, to give protection to this genetic material, this protein is present. So it present like a coat. So this protein coat is called what? This protein coat is called capsid, and this is genetic material. Only two structure, very, very simple structure. And here in this protein coat, this one. This one is nothing but capsomere. Capsomere. So like that, these small units, what you are seeing, no? So these are all nothing but capsomere. So they are surrounding this genetic material. Very simple structure. But all viruses are not showing the same structure. Their capsid nature, their capsid shape could be different. Listen to me carefully. Capsid shape could be different, but the two components are the protein component, that is nothing but a capsid, or it is a nucleic acid, that is nothing but genetic material. Okay, is this clear? Very simple. Now, look at this image. Look at this image. Here you will see how they are showing differences in the shape of the capsid. This is the TMV. TMV means tobacco mosaic virus. So it is going to cause disease in tobacco plant. So viruses are very, very specific. They are not going to infect all the plants, all the animals. No. So they are very specific. That means if the work is allotted to this TMV, that means tobacco mosaic virus, it won't attack tobacco plant only. It won't attack coconut plant or paddy plant or nothing. Okay. So all the viruses which are attacking plants are always single-stranded RNA. They will be having genetic material as single-stranded RNA. Remember, the plant viruses are always RNA. It's a neat question. Now, if you just look at the structure, can you see the green structure here? The greeny structures, what you are seeing now, these are nothing but the capsid. This capsid are in a helical form. Helical means something like this. See, this capsid is surrounds like this. So again, it's going to surround. Again, it's going to surround. So like this, it is present. This is a tobacco. It's a helical in nature. That means I'm talking about the capsid nature is helical. Okay. And inside, it is having a single-stranded RNA. So here, RNA is the genetic material. Right? So the same diagram is given in pink color. They have shown it. In pink color, they have shown. Can you see? Can you see? This is RNA. Now, this is another virus. This virus is going to infect bacteria only. It says, I'm not going to infect plant or animal. So bacteria is very small, no? Look at here. If it is your animal cell, imagine this is animal cell. So animal cell, first of all, cells do not be seen through naked eyes. They are so small. Compared to animal cell, the bacteria is still very small. This is bacterial cell. Now viruses, they are going one step ahead. That means they are telling, we are still very small. If you just take a needle and if you just touch a virus, in the tip of the needle, you could find trillions of virus, billions of virus. Can you imagine? That means such a small sized organism they are. Okay. So obviously they are going to attack even on bacteria also. Okay. So that's why That's why such viruses are given a special name called bacteriophage. Okay, so they attack bacteria, and these bacteria and these viruses, in their body, the genetic material is double-stranded DNA. Okay, listen to me what I am telling. The plant viruses are always a single-stranded RNA. And bacteria attacking viruses are always a double-stranded DNA. Okay, so when you just look at its shape, it may come in your NEET exam. That's why I'm telling in detail. If you just look at the structure, you can see head, this is the collar, this is the sheath, and this is the tail fiber. Okay, very simple. This is the head part. This is the head part. Okay, and below the head, just like our neck, here is a collar. This is the collar. Let me mark it as head. This is collar. And below the collar, there is a sheath. Right? And after that, it says, I will be having many small needle-like structure like this. So these are called tail fibers. Tail fibers or tail sheath, anything. But it is given here, tail fibers only. Okay. So this is head part, this is collar, this is the sheath, and this is tail fiber. Very simple. So this entire thing is protein only. This protein only. It is shown in different shape. And inside present this double-stranded DNA. Got it? Now, another virus which is showing its capsid in a different polygonal shape. Hence, it is called polyhedrovirus. Polyhedrovirus. Okay, so these are the capsomere. Inside the DNA is present. Simple. Now, we have understood the structure of virus. Now, we need to get the answer for this question. Now, tell me, by showing all this structure, what are all these structures? Only two structures. Either a genetic material and protein part. That's it. Now, will you consider this as a living organism? Now we start scratching our head because the viruses are also very smart. They are telling why they are making us confused because of this region. Let me write living characteristic here as well as non-living here. Suppose if I have to consider any organism as living, then it must have a cell, right? But here it is no cell. First of all, it is not considered as a cell only. Okay, and now it's just remain as a dead body outside the body of a host. Imagine a plant. It is getting attacked by a virus, right? Then inside the plant body, it starts multiplying. But when you just remove the virus from plant body and just keep it outside, then it acts like a dead body. But bacteria do not behave like that. Other cells, fungal cells do not behave like that. Whether they cause disease or not, outside they remain alive only till their lifespan get complete. But here, these viruses are not like that. They always do remain dead outside. They not even multiply. They not even show any metabolism. Okay. In the first chapter itself, we have understood metabolism is a characteristic feature of a cell, so that if you have to consider it as a living. But here, no metabolism outside the host. So like that, no multiplication or no replication or no reproduction. No reproduction outside the host. So, we thought, oh, okay, it is not a living, then it is a non-living, right? Yes. But here, when it comes, I made a mistake. One second. It should be non-living. It should be non-living. No metabolism outside the host, no reproduction outside the host. But now, how can you consider it as a living? So let me make it as a living. So if you are considering a non-living, then I can prove that they are not showing metabolism, they are not showing reproduction, they are not at all a cell. Now, once again, it is making us to get confused and they are telling that we are living. Why? Because they are showing metabolism inside the cell, inside the host, not cell, inside the host. And they are showing reproduction inside the host. Now, again, it started creating confusion. You are multiplying, you are reproducing, you are showing metabolism inside, but in outside, you are just like a dead body, you are behaving like non-living. Then who we are? whether you are living or non-living. Tell me. Then it says, I am on a boundary. So I am standing on a boundary between your living and non-living. I can act as a living, I can act as a non-living. Okay, so that's why what the conclusion we got from this point, it acts as a boundary. Viruses are on the boundary between living and non-living. This could be a NEET question. It could be a statement-based question. Okay. Now, by having all this idea, let's move forward. Let's learn something regarding virus. Okay. Yes. Discovery. Who discovered virus? Actually, the term virus for the first time coined by a person called Pasteur. So, the Pasteur name was given in old textbook, not in new textbook. Okay. So, don't say that, ma'am, this person name is not given. I will tell you while doing at the end what the confusion you may get while reading this virus part regarding his discovery. I will clear all your confusion. Don't worry. First, listen this story. So, the discovery of the virus credit is not goes to any one scientist, because plenty of scientists worked hard. Because you know, already the viruses are very, very small. I have just shown how small they are. Even they are very, very small compared to the bacteria. Now, such organism or such component, when enter into a body of an organism, when they start acting like anything, when they start multiplying like anything, then how it is possible us to discover it is. Okay, it is not such an easier task, but still scientists didn't kept as it is. They started working hard, they started doing lot of experiment, a series of experiment, and finally they discovered that yes, this is the agent. This is responsible for causing such disease. Okay. So, in that scene, the first scientist came forward was Dmitry Ivanovsky. He was a Russian scientist. He did experiment in the year 1892. What he did, he was observing a plant, tobacco plant. So, this is tobacco leaf. So, do you say that it is a healthy leaf? No, it is an unhealthy leaf because it has been infected by tobacco mosaic virus. TMV virus. But by that time, in the year 1892, he didn't aware about that tobacco mosaic virus. First of all, he was not aware about viruses only. So that's why in order to discover it, he started doing experiment. So what he did, he took leaf of tobacco plant. It has got infected, no? So it is an infected leaf. Okay. So now what he did, he started crushed it. With the help of mortar and pestle, he made it a paste form. Okay, he extracted the sap from it. Sap in the sense juice from this plant leaf. Now he started filtering it. So imagine he shifted this juice into this jar. So yes, in this jar we have kept it, the filter. So now all the sap, he extracted the sap, that is nothing but the juice he has just added here. Now the sap started getting collected by filtering through this filter. So this filter was nothing but a bacterial proof filter. Bacterial proof filter. It means it is going to filter the bacteria, because he thought that if this disease, if it is caused by bacteria, then obviously all the bacteria which are responsible for causing this disease might get hold here. How we are going to strain by, that is nothing but a tea strainer or a coffee strainer, how all those material debris are going to collect on the strainer, only the liquid part is going to collect at the bottom. In the same way, if bacteria is present, it is going to get hold here only. It will be get held up here in the filter. So it is free from free from bacteria. Now he took this container, the liquid part after straining. Now this one he transferred or he sprayed onto the new leaf. This is a new healthy leaf. Are you getting? On a healthy leaf, he just sprayed this filtrate. Okay. If it is a bacterial disease, suppose, then what should happen? This leaf should remain healthy only, because if it is a bacterial disease, bacteria has been get trapped here only. But the scenario was different. What happened here? The leaf again started getting converted into diseased one. Okay, let me show. Okay. Let me draw another leaf. Now it become diseased. It become infected again. Then two things came in his mind. What are two things? One thing, it could be some particle which could be very smaller than that of the bacteria. So that's why it could able to manage itself to pass through this bacterial filter. If it is very small, then only it could able to pass through the filter, no? So that could be one possibility. And another possibility is that this fluid, what he is going to get after filtering this, no? So this fluid itself could be the poisonous fluid. So these are the two possibilities that he has concluded. What are the two things came in his mind? One is it could be a small particle, but still he didn't say that it is a virus. He said it could be a small particle, that's it. And another thing he thought, it is just, he's suspecting. Another thing, it should be a toxin. It should be a toxic. So now, but he didn't draw any conclusion from this, just his prediction like that. Then one more scientist came. Who is that one more scientist? M.W. Bejernik. So M.W. Bejernik, he did the same experiment. How this Ivanovsky did. Okay, he also took the same leaf in the sense, tobacco leaf he took. So this is the infected tobacco leaf. So he passed through the bacterial filter. So the sap got collected. It's a pure filtrate. All the bacteria got trapped here only in the filtrate. Now, he just sprayed this on a new leaf, healthy leaf. He observed, just like Ivanovsky, again it got diseased. Healthy leaf also got diseased one. Now, again he did the same experiment. Again he took the sap, that means he crushed the leaf. He collected the sap. Again, he is going to did the experiment on another leaf, new leaf. Oh, leaf become yellow. Leaf should be green, no? So again, when he sprayed on the new leaf, again it got disease. Once again, he crushed this leaf. Again, he collected the sap from this leaf. Again, what he is going to do? He experimented on another leaf, another healthy leaf. So like that, he did for several leaves. He did the experiment on several leaves just by crushing the leaf, collecting the juice and spraying on healthy leaf. Healthy leaf become diseased. Now, from the diseased leaf, again he crushed it. He took the sap, that is a filtrate. Again he is going to put on the healthy leaf. Again that become diseased. Again he crushed it. Again he did the same thing. But every time, whenever he do the experiment, that means from the diseased leaf, every time the leaf was going to get infected. It was going to get diseased. Then he thought that it is not a toxic. If it is a toxic, then its nature could be get lowered. Lowered in the sense, its power should be get lowered. Because toxicity level should be a limited way. Okay. It should not go continuous or it should not show its effect continuously for several leaves. So, it might be some organism or it might be some particle or it might be some component, but it is not at all a toxic material. That conclusion he gave. But he thought that this liquid it is there, no, it must be contaminated by something. Okay, so that's why he gave this term. What is the term? It is a contagium vivum fluidum. That means the sap what he was collecting, that is nothing but infectious living fluid. That means something living is there. It is not a toxic. So that fluid has been got infectious. So that's why that infection is keep spreading whenever it comes in contact with the healthy leaf. Okay. So finally, W.M. Stanley, he made crystallization experiment. I mean, he crystallized it and virus got crystallized. That means the protein part in the virus got crystallized. You take any cell, it won't get crystallized. You take any bacteria, it won't get crystallized. But this virus has got crystallized. Okay. Then finally they came to know that yes, it is nothing but it is an organism. But the term virus was given by whom? Pasteur. Okay. So whose scientist name? Who are the scientist name you have to remember? M.W. That means first is Ivanovsky, then Bejernik, then this fellow Stanley. So based on these three scientists, you may get question. But Pasteur, the scientist name has got deleted from NCERT. The chances are there, but 90% chances are very less as it is not given in NCERT. But still, if you know the term, it is very good. Okay. Now let me read the NCERT text part. What are all the doubts that you may get? I will tell you here. Listen to me. Viruses means venom. Venom is nothing but poisonous fluid. Okay. Dmitry Ivanovsky in the year 1892 recognized certain microbes as casual organism of the mosaic disease of tobacco. See here, Dmitry Ivanovsky, first of all, he didn't notice these are the organisms. He just did the experiment. But here this line is given that certain microbes has given, but still, okay, fine, no problem. These were found to be smaller than bacteria because they are passed through bacterial proof filter. It is just passing through bacterial proof filter. If you are not able to visible the text, you can open your textbook and you can read there along with me, or you can just watch the lines what I am reading here. Okay. But M.W. Bijernik in the year 1898, please note down the year, it may come in your exam, demonstrated that the extracted of infected plants of tobacco could cause infection in healthy plants. That means he did the same experiment and named the new pathogen virus. See, this is the sentence where many students get confused and they tell, ma'am, you said the Pasteur has coined the term virus. But here it is given that he named new pathogen virus. But I'm not telling it is wrong. Here, by the time of doing experiment by Bijernik, first of all, he was not aware about the term virus. After this, the Pasteur came into scene, he named the term virus, but he has just said some infectious living organism as there. Okay. But NCERT is making us to get confused. We know that it is virus. So that's why they are giving it the name the new pathogen virus. But you are going to get the question from this line. He called the fluid as Contagium vivum fluidum, that is infectious living fluid. Okay, clear? W.M. Stanley in the year 1935. See, just look at the time gap here, 1898, here 1935. So they took a lot of years for their conclusion, for their experiment result. So by the time the Pasteur also came in between, he coined the term, right? Showed that the viruses could be crystallized and crystal consist largely of proteins. Again, NEET question. They are inert outside. Inert means they are numb. They just like a dead body outside. Their specific host cell. Viruses are obligate parasite. NEET question. Viruses are obligate parasite. Why obligate parasite? Because outside they do not do anything. They simply looks like as they are very innocent or dead bodies. But as soon as they attack the living organism, they started showing their action. Correct? So obligate means compulsory. Compulsory. There is no other option. They must cause disease. That's why we call it as an obligate. Okay? And now, in the next paragraph. In addition, already we have done so many things here. We have covered, but still let us read. In addition to proteins, viruses also contains genetic material that could be either RNA or DNA, not both. Either RNA or DNA. No virus contain both DNA and RNA. NEET question. It could be statement-based question. No virus contains both RNA or DNA, or virus contains both RNA or DNA. So be careful. No virus. Single virus I am talking. In a group of virus, you may find RNA in some virus, DNA in some virus. But in a single virus, you do not find both. That is what the meaning. A virus is a nuclear protein, I said, and genetic material is infectious. See, NEET question. Genetic material is infectious here. Okay. In general, viruses that infect plants have single-stranded RNA. Yes, for this I will explain here about the genetic material. See, how you have to analyze or how you should be careful while analyzing the genetic material of viruses. We all know that the viruses are going to infect everybody. It won't leave even bacteria also. Bacteria saying, hello man, I am very small. I am going to infect others, but you are attacking me only. Virus says, yes, I am the most powerful. Everybody has neglected us now. Yes, I am going to attack on you only. That means the virus is not going to leave anybody, even it could be a bacteria. Okay. So that's why let us make three groups here in order to identify the genetic material of these viruses. Let's make plant as one group and bacteria on the dead end as other group. Now we are supreme animals. So let us keep in the middle. So in one end, that means in the plants, how we are going to detect the viruses are having this genetic material only? The plants are always do have single stranded RNA. Okay. So on one end, just maintain single stranded. On other end, write down double stranded. Double stranded. So one side is RNA and other side you just write DNA. That means always bacteria are infected by double stranded DNA. So or else I can write dsDNA. Here ssRNA. Okay, we are genius, no? Animals. We are saying we are allowing everybody to infect. Either it is a single stranded RNA, they can come and attack on animal. So I am telling, not plants are having single stranded RNA. The viruses which infect plants, they will be having single stranded RNA. The viruses which infect bacteria are having double stranded RNA. Okay, so like that, if the virus want to infect animal, it could be single stranded RNA. The single stranded RNA can go and attack on animal. Double stranded DNA, they can also go and attack on animal. Or even sometimes RNA could be double stranded. Double stranded RNA can also come and attack on animal. So in animals, we found all three categories of viruses. Whereas in plants and bacteria, it is very specific. Okay, can you remember? So this is what the sentence given further. The plants having single stranded RNA and the viruses that infect animals are either single or double stranded RNA or double stranded DNA. That is what I wrote. Bacterial viruses are bacteriophage. That means virus which infect bacteria are usually double stranded DNA viruses. The protein coat called capsid, made up of small subunit called capsomeres, already I did, protects the nucleic acid. These capsomeres are arranged in helical, that means in TMV, polyhedral, geometric forms, that means polyhedrovirus. Viruses cause diseases like, yes, once again, you may get a question in this paragraph, which among the following is a viral disease? Which among the following is a bacterial disease? Like that. So viral disease, mumps, smallpox, herpes, influenza, AIDS in humans are caused by virus. Okay. So these are certain infections we found in animals. Okay. If you come in the case of plants, tobacco mosaic virus, all those things, general symptoms here as given. In plants, the symptoms can be mosaic formation. Okay. Just means a pattern here. Leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Okay. These are certain symptoms. If you just look at a plant, you can able to see that it is not a healthy plant because it is virally infected. If such symptoms given and if they ask which is the causative agent, just tick on virus. That's it. Okay. Now, we are saying that viruses are going to do like this. Viruses are going to multiply like this. How exactly the viruses are going to multiply in an organism? Let us talk by taking example of a bacteria itself. Okay. So, if the virus attack an organism, either they follow lytic cycle or lysogenic cycle. Okay. So, this information not given in NCERT, but you may get a question. You may get a questions. Okay. In applied level, in level 3 type of question. So, if you have a bit knowledge, there is no loss, right? Let's learn. In lytic cycle, what happened? Imagine this is a bacterial cell. Okay. To infect a bacterial cell, who will come forward? Bacteriophage. So now, let's imagine this is the bacteriophage. So this is a capsid, this is a sheath. So these are the tail fibers. So with the help of this tail fiber, they first make a small hole on the cell. Okay. Now, what it will do? Imagine it is its double-stranded DNA. Bacteriophage is having double-stranded DNA, no? So now it will insert its DNA. And inside the bacteria, it is having its own DNA. So as soon as it entered, the first work of the viral DNA is to make a piece. This viral DNA is going to make fragmentation of the bacterial DNA. Now, the bacterial DNA is going to be broken into small pieces like this, like this, like this. Now they become useless. Now these viral DNA are very smart. By using the enzymes which are present inside the bacterial cell, they started multiplying and they started constructing the new viruses. Okay. The new viruses started forming inside inside bacterial body because they use bacteria as a factory. Now, they made several copy of their own, and they started inserting inside a new virus. So finally, it is going to burst out. The cell is going to burst out and they start released outside. These viruses released outside. And now they are going to wait to whom is our next target. So again, they found the next target cell, next bacteria. Again, they just go and attack on them. So like that, this cycle keep repeating. They just keep targeting every bacterial cell. Understood? So this is what we call it as a lytic cycle. Okay. But every time, it is not going to destroy the bacterial DNA. If it is the scenario, then what happens? Every bacteria which are present in the environment, they will die because of this virus. But bacteria are also very smart. So what they are going to do? They having some weapons. That weapons are nothing but restriction endonuclease that you are going to study in class 12 biotechnology chapter. Those students, if you are class 12 student, if you are watching this video, already you are aware about this term, restriction endonuclease. So restriction endonuclease, they are in turn target virus only. They will cut this virus into small pieces, viral DNA into small pieces. So that's why the equal fight will be given by both virus and bacteria. So finally, who is going to win in the battle? That will be decided by their strength. So that's why this cycle, if it is weak, if the bacteria is weak, then it is going to attack on the bacteria like this. But sometimes bacteria as well as the virus, they started behaving friendly. So that happens in lysogenic cycle. So here what happens? Imagine this is a bacterial cell. Here the virus came. Now imagine this is the bacterial DNA. Correct? So when the virus insert its genetic material, instead of killing this bacterial DNA, it incorporate within the bacterial DNA. It started adjusting within the bacterial DNA so that it is also, I am also your family member. Okay. So the bacteria also started treating this viral DNA as its own. So what is the advantage of this? Very simple. Whenever this bacteria multiplies, obviously the viral DNA which is so present, it also start multiplying. Correct? It multiplies here every time. The viral genome also started multiplying. So that is what the advantage. But how long? We don't know when it is going to get triggered and when it is going to turn itself into lytic cycle. Lysogenic cycle, when it is going to turn into lytic cycle, we don't know. It depends once again on the situation. Suppose if any antibiotic came here to attack this bacteria, then this virus started thinking, oh, anyhow this bacteria is going to die. If I simply stay as it is inside his body, obviously I'm also going to get killed by this antibiotic. Then it started, anyhow I am dying. Anyhow this cell is dying. Why can't I kill it? Why can't I go back to a lytic cycle form? So again it started killing this bacterial cell. And sometimes if the bacterial cell is not getting enough food, then also when the situation is about, that means when the situation comes so that the bacteria is about to die, by that time also this virus again go back to lytic cycle, start destroying bacteria. So like that, it depends. But lysogenic cycle is also one of the category of multiplying its form. Understood? So like this, the virus multiply in host cell. Okay. Now we are in the last part, viroids and prions. So already we came to know that viruses are very, very small. Now, one more step ahead. These viroids and prions are challenging that we are still small. We are having only one component from the virus itself, and we are showing our notorious activity. What is that? Nothing but viroids and prions. Okay. So let me tell you what are these things. Virus means genetic material, that is nothing but nucleic acid, as well as the protein. Correct. Now you remove protein. Imagine there is only genetic material. Say for example, single-stranded RNA is there. Here, it has no protein at all outside. itself it acts as a notorious agent and it started causing disease. Okay, so such agent where the devoid protein, we call it as what? Viroids. They are called viroids. Viroids means they lack of devoid protein. They have very low molecular weight. So what they do? So if you just look at this RNA, we think that what it can do. After all, it is a single-stranded RNA. But this single-stranded RNA, it is so lengthy and it will fold upon itself and looks as it is going to be, looks like a double-stranded. And when it entered into a potato, okay, so normally potato shape will be like this. Either it is a spherical or somewhat like a oval, correct? But if it attack on potato, then the potato shape become like a spindle. Okay, we call it as a spindle tuber disease. So such thing was noticed by this fellow called Theodore O. Diener or T.O. Diener. T.O. Diener. Just remember, the Diener might have entered into dining table or sitting on dining table to eat potato. That means the food related to potato. So this potato, when he observed, it became a spindle shape. Then he started working on it. So it is just a trick to remember. Okay. So this is how the diseased potato looks. Understood? It is caused by who? Viroids. Viroids are nothing but single-stranded RNA. Right? So here is the text regarding this viroid. Let's read it. In 1971, very, very important, year is very, very important, T.O. Diener discovered a new infectious agent that was smaller than viruses, caused potato spindle tuber disease. Important, NEET point of view. It was found to be free RNA. Okay, it lacked the protein coat that is found in viruses. Here it is not given single-stranded RNA, but it is single-stranded RNA. As it is folded upon itself, it looks as it is a double-stranded. Okay, so it lacked protein coat found in viruses. Hence, name viroid. So viroid, oid means devoid. So the RNA of the viroid was low molecular weight. It is a statement-based question. Understood? The next one is prion. Prions means what? Virus is having two components, I said earlier itself. One is genetic material. Another one is protein part. Now, if protein part was not there, only genetic material could also cause disease. That is nothing but viroid. But now here, just remove the genetic material. Only protein. What you are saying? Only this protein could also act as a disease-causing agent. Okay. If this protein is normal, then it won't damage anything. But if this protein abnormally get folded like this, that is nothing but abnormally folded protein. So such proteins are called what? Prions. Usually, look at this image, you can understand. This is the normal prion. That means normal protein. When it abnormally get folded, it get converted into abnormal prion. So when it enters into a cow body, then it start misguiding the other proteins which are normally behaving in cow body. Okay. Imagine this protein, prion, has entered and it start looking around the proteins which are normally behaving, normally functioning. Then it start misguiding why you are behaving normally, just like a notorious boy when he entered into the class. How he is going to spoil the entire school atmosphere, class atmosphere, who will spoil the mind of student's mind. In the same way, here also this abnormal folded protein misguiding normal folded protein so that it is making those normal protein to get converted into abnormal protein. So that they also start twisted abnormally, they started behaving abnormally. So protein itself is very important to our body, right? Not only cow body, in all human body, animals body, everybody is controlled by protein only. DNA itself is converted into protein, protein is doing the action. Now, this abnormal protein, when even enters into the brain, so there also it starts behaving abnormally, then what happens? The cow started behaving like a mad. So that's why in a cow, we say that the prion causes mad cow disease. Mad cow disease. So scientifically, we have given a name called bovine. Bovine refers to cow. Spongiform. Spongiform. Why it is called spongiform? Because if you take the tissue of a brain and observe under the microscope, it looks like a spongy nature. So that's why spongiform. Encephalopathy. Encephalopathy means nothing but brain tissue. Encephalopathy, it is due to this abnormal protein, abnormally folded protein called prion. Suppose if such protein enters accidentally into human body, in human, it also causes a disease called, again, it acts on brain and causes disease called C.R. Jacob. Okay, so let's read NCERT, what I have taught just now. In modern medicine, certain infectious neurological disease, see, neurological disease because it affecting the neurons, were found to be transmitted by agent consisting of abnormally folded protein, that is nothing but prions. The agent was a similar size to viruses. It will be look like viruses only. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and an analogous variant, C.R. Jacob disease, CJD, in humans. Sometimes they may not give the full form called C.R. Jacob, CJD. If it is given, it is understood that they are talking about humans. Or BSE, bovine spongiform encephalopathy in cattle. Okay, that's it. Over. Chapter over. The last part is lichen. Already we did. Lichen, it is consisting of algae as well as, what is this? Fungus. Okay, actually fungus should be green. Sorry, white. Algae should be green. No problem. Here, the algae component is called phycobiont. Phycobiont. Only this word I didn't tell while doing. I'm telling here now. Phyco means refers to plant. Phycobiont. Whereas fungus was referred by a term called myco. So that's why it is called mycobiont. It is a close association between them. That is nothing but algae. Let's read it. Lichens are symbiotic association. We know that mutually useful association between algae and fungi. The algal component is called phycobiont. I said just now. The fungal component is called mycobiont. Again, it could be a NEET question. Which are autotrophic and heterotrophic respectively. Autotrophic means phycobiont is autotrophic because it's prepared the food. Mycobiont is heterotrophic because it is depending on algae. Algae prepare food and fungi provide, what is this? And fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. That means when you just look at the lichen, you can't even imagine the two organisms are surviving there together. Lichens are very good pollution indicator. NEET question. If you just look at lichens around your lichens around you, that means in your surrounding, you should feel proud that it is a pollution-free environment. Because lichens easily do not grow in a pollution. Okay? I mean in a polluted area. If you just go towards the forest or the place where there is very cool environment, no vehicles movement, then obviously you can find the lichens growing on the trees. Okay, that is what it is given. They are very good pollution indicator. They do not grow in polluted area. That's it. Let's check our knowledge by solving MCQ. Okay, if it is done, this part is over. Yes, match the column and find out the correct combination. Viroid, viroid is a infectious nucleic acid. Here should pair with 3. So here itself we got the answer, A3. B, virus is a nucleoprotein, B1. C, prion. Prion is an infectious protein, C2. And virion is an infectious viral particle. Right. So option C is the answer. Next question. And the antibiotics have no effect on viruses. Why? If you take antibiotic, they do not affect on viruses because they, first of all, they don't show metabolism only. Correct? They kill all bacteria which act as host of virus? No. Viruses are too small in size for antibiotics to act upon them. No. Viruses show no metabolism of their own. So, they do not show their own metabolism. They require the enzymes of host cells in order to multiply. So, this is the answer. Therefore, there is no work of antibiotics on them. Viruses produce a thick covering and encase themselves. Wrong. First of all, endospore, encystment, all these things already have told in other chapters, but it is not relevant to virus, it is relevant to bacteria. Here also I said though, endospore formation. Association between mycobiont means fungus. Phycobiont is found in lichens, correct? Not in mycorrhiza. See, in mycorrhiza, root as well as the fungus is there, but such association we won't call as phyco or myco, okay? So therefore, these three are wrong. Next question. With respect to fungal sexual cycle, choose correct sequence of events. See, it is actually related to fungus. This question I should have put before the virus itself, but it's okay, no problem. Anyhow, it belongs to this same chapter only, no? First karyogamy is given, wrong. First, which step should come in the sexual cycle? It should be plasmogamy. After plasmogamy, it should be fusion of nucleus, karyogamy, then meiosis should take place. So, you should know the sequence. This is the right sequence. Next, in a bacteriophage, select the correct information. See, here markings are given. We already know what are these things tell. This is the head part. A should be head. Here A is hand given, therefore it's wrong. Here A head is given, okay. D sheath. So yes, D is sheath. Correct. So here itself we got the answer. B is collar. Aha. I did mistake. Is it sheath? You are simply looking at me. This is what? This is the tail fiber. This should be tail fiber. So it's not our answer. B is collar. B is collar. This is correct. Then D is tail fiber. Yes. So this is our answer. C sheath is given. Therefore, C is sheath. Correct. But A collar is given. A should be head. Therefore, this is also wrong. Next. After karyogamy, followed by meiosis. See, once again, it's a fungus related question. Kingdom fungi. Spores are produced exogenously in. See, use your brain. Think calmly and tell the answer. What they are telling? After karyogamy. After karyogamy means what they are telling? Meiosis stage. That means the zygote has formed. When the zygote 2N has formed, it has to undergo meiosis so that 4 spores has to form. Now, the spore produced are exogenously in. Option A, neurospora. In neurospora, exogenously formed. Neurospora, that means we are putting in bag. We are putting in a sac because the boy is going for genetic work, correct? So, how it was forming? It was forming inside. Inside the spores were forming. When it get burst out, then it was releasing. It was not exogenously, it was endogenously. Therefore, it is wrong. Alternaria, that is nothing but cat comes in which example? Coletotrichum alternaria trichoderma. It is in Deuteromycetes. Deuteromycetes, first of all, do not show sexual cycle. Then how come you expect the meiosis? Therefore, this is also wrong. And the rest thing is agaricus. Of course, in agaricus, what happens? Here the 4 spores which are so formed, they will be pushed outside. Correct? So, the 4 spores were forming outside. It is exogenously. This is the answer. Whereas yeast is single cell. If you consider a bud as a spore, but it is a mode of reproduction. Okay? It's not a sexual. It is a vegetative. Therefore, it is also not an answer. The right one is agaricus. Next question. Which of the following statement is incorrect? Viroid lack protein coat. Correct. Viruses are obligate parasite. Correct. Infective constituent in viruses is a protein coat. This is incorrect. Therefore, this is our answer. Infectious component in viruses are genetic material, not a protein coat. Prions consist abnormally protein, folded protein. Correct. So, we have to identify the incorrect. We have identified it. Next. Potato spindle tuber disease caused by viroid, not virus, bacterium and nematode. Next. In plants, mosaic formation. See, symptoms are given. Direct MCQ from NCERT. Mosaic formation, leaf rolling and curling. Why in between I am putting such a question? Because you should not neglect, so that if all the questions will comes only in applied level or only in concept based. Because some questions will be directly picked from NCERT line also. You should be aware, you have to read NCERT textbook minimum 5 to 6 times. Okay, a good student, he will read minimum 5 to 6 times. A NEET aspirant, he will read more than 8 to 9 times. So you decide whether you are a NEET aspirant or not. Okay. Yellowing of plant parts, vein clearing, dwarfing and stunted growth, necrosis, everything. Symptoms of what? Viral disease, not a bacterial, mycoplasma or fungal disease. Okay. That's it. So this is what the end of the chapter. Hope you understood all the concept of this chapter. It is everything is perfect and crystal clear. Just read NCERT, go through NCERT, and if you like the video, just hit the like button. Okay, thanks for watching.